very efficient team of trained experts. So it has been a thrilling journey, and this experience has enabled us to work on a variety of challenges that comes with educational training and translational research, but with the agenda to bring awareness about data-driven healthcare research. Our team reached out to universities across India to make bioinformatics and high quality data-driven research accessible to absolutely everyone. So this engagement, this entire engagement has helped us to collect data and tremendously improve our learning models. That led to these many programs. So after many programs coordinated with universities and also thousands of students across India, we saw that there is a gap in skills that student effectively could integrate into research project, thinking critically about data and applying appropriate analysis methods to this right data. So to address this, during the fall, as I was telling you, that our team reached out to universities across India, and with this common purpose. So we were looking for partners who have this common purpose in the development and dissemination of awareness, of knowledge, of understanding of new technologies, importance of biomedical research. So we collaborated with Chandigarh Group of Colleges, Landrun, India, signed a memorandum of understanding which focuses on basic bioinformatics training to incorporate bioinformatics curriculum and help the final year students with their research projects, with their internships. So our team at Chandigarh facilitated the ceremony along with the administration there to start this hybrid bioinformatics training and research. So our team in Delhi signed an MOU with IIMT Meerut University. The two institutions, here the objectives are different. The two institutions have agreed to collaborate to enhance healthcare research by introducing our innovative OmicsLogic training programs for students to learn and apply computational tools for biological and biomedical research, addressing the gap between academia and industry. So this is a common objective, but here we are working on specialization where we collaborated with the scope to develop diagnostic testing kits, genotoxicity in vitro disease, and then using cell line information, designing drugs and testing on cell lines, and a lot of other future goals which we discussed and collaborated and working with IIMT Meerut. So expanding and moving from to help students from various research universities, various universities where research is now one of the key objectives as an achievement for a student to get after completion of a degree. So our team collaborated with the School of Healthcare and School of Computer Sciences at Rishihod University at Sonipat, Haryana. And then we also collaborated with an historic memorandum of MOU, which was signed between MIT University, Uttar Pradesh, and Pine Biotech for joint training and research in the areas of bioinformatics and data science. And today I'm very happy to let you all know that we've been, since the signing of the MOU, we have been working on specialized programs that we designed with the help of the faculty and our mentors. And we will be discussing more about that research training, which has all the elements in the research fellowship meeting that we have in the symposium right after the expert sessions, which we'll be going through in a while. But to tell you more about this historic agreement, it's because multiple departments with different research that they are working on, may that be cell and molecular biology, may that be biotechnology, may that be computational biology, has been a part of this collaboration with more than 300 faculties participating from different MIT universities, working on different research projects, and also students who would want to work on these research areas, which involves bioinformatics, computational biology, and applying data science skill sets to address and find solutions which could help in translational research, as well as finding commercialization in the sense of maybe finding ways to do something new within industry, creating a startup, or other outcomes which are possible through this through the research that is one of the outcomes of such training. So our team in Chandigarh has been actively involved to address the needs of the students with guidance, enabling them to understand new analysis methods, technology. So our team is also conducting on-site workshops, helping students with the very basic steps and clarifying doubts about moving forward to learn new tools and techniques and to apply this on a projects. So we conducted several of these on-site workshops where our team is available in India, in different cities in India, and also in Nigeria, where we will be learning about those sessions today, and also in the US. So addressing one of the major gaps from learning about how important it is to give the student the next step. So coming from training, coming from research, now to coming to the part where there are opportunities, which is the NGS, which is the next generation sequencing industry, which is evolving rapidly in India. We collaborated with Prema's Life Sciences India, a young, dynamic, and focused organization, which is working on technologies, niche technologies such as genomics, cell biology, helping biopharma to boost innovative research and diagnostics in India. So together with their team, we are creating these partnerships to help students who are from our partner universities and also faculty who are from our partner universities and reaching out to us to guide them from experimental designing to data analysis. So in this way, we are also helping the entire student community by conducting many of these sessions as free workshops, free webinars, where these experts from the industry are coming and helping talk about these different topics that are really relevant to the pharma and to the research community. Similarly, we discussed about NGS industry. We have collaborated with FABA, an organization, a non-government organization, non-profit society, which is the Federation of Asian Biotechnology Association, and that was created with a common platform to promote biotechnology in Asian countries by pooling resources and expertise of each nations in Asia, and then keeping the pace with advancements in the field of biotechnology and bioinformatics. So Pine Biotech and FABA discussed and collaborated to offer and conduct and promote training programs, and as well as recruitment and placements of students in the field of drug discovery, who are working and having skill sets in drug discovery. So as our programs already serve thousands of students across Asia, we are eager to develop skills necessary to work in develop skills necessary to work in the industry. So we would wanted to provide them the right opportunity and also the training that is associated and that is required for anyone to get into the pharma industry. So we collaborated, we designed curriculum, and we are working with mentors from FABA, from Science Guru, and this entire huge community of Federation of Asian Biotech Association to help students learn about these important topics in bioinformatics and data-driven research. So integrating all of this, we have had several meetings where we discussed that how these different components of education, of industry, of training, of working on different areas of research, of utilizing some of these for, utilizing some of these to solve real problems can be thought as, finding a business solutions, finding a business incubator and creating startups and creating opportunities for students who can come up with proposals that can be the part of solution by also providing them a platform such as a business incubator. So we collaborated with through the universities and other organizations where we can do these hackathons, where we can do this ideathons that can help students to express their ideas, and also we will be able to give them guidance to those to translate those ideas. So from those ideas to creating these events, this research symposium is one of such event, and I'm really looking forward to hear from all the speakers and learn about their perspective on topics that are really dear to me. So I have been a student of bioinformatics all of my life, and so I'm excited that I'm working with many of you here today on making this discipline more and more accessible. So in this next 2 days, we will hear from these speakers, address these topics we can all relate to. So how to stand out from the crowd when you apply academic programs or create a career in bioinformatics. How can university do to equip, what can university do to equip their students with relevant skills and experiences? What is this future? What is the future of this field? Where are we going with this technologies, and where will you find these cutting edge challenges that you can contribute in? So I'm really excited for the next upcoming sessions, where we have a panel of speakers from India talking about bioinformatics education, precision, how do we make advancements in agriculture by applying precision, smart crops, precision agriculture. We will learn about biomedical challenges, and we will learn by one of the top universities in India about their education model in bioinformatics, so that from education to research, we will understand that how these things are changing in India, and what are we up to. And then we have the next session after this, which would be about how anyone can be a part of this sort of research by by utilizing some of the resources that are structured in such a beautiful way that anyone who is inquisitive, who wants to do this, is being given all the resources to be able to achieve that. So that is about our research fellowship program, which is to generate new work on new problems, generate new analysis, and come up with new research outcomes. So followed by that, we have a poster presentation session from our students, and we have selected some amazing research studies which have been presented by some of our students, and they'll get an opportunity to showcase their posters with a panel of judges. And then we have the Africa panel and Africa program outcomes, followed by Latin America, and then we have the US. So with that, I would like to again welcome you all to this research symposium, and I hope that it would be an amazing journey for all of you, and we make the most out of this session. And I'll also appreciate if you can put your questions in the chat so that we can address them when we have the time. So with that, I'd like to pass this stage to Sonalika to welcome our next guest from MIT University. Over to you, Sonalika.
Thank you so much, sir. I would now like to invite Dr. Puniti Mathur. Please share my screen. Yes. Dr. Puniti Mathur is a seasoned researcher and an academician with over 17 years of experience in biotechnology. Her research interests lie in peptide synthesis, analysis using HPLC and NMR, solution confirmation, and computer-aided drug designing. She is also associated with the International Center for Genetic Engineering and Biotechnology. She is responsible for accreditation and ranking exercises, as well as industry-academia partnerships at Amit University, Noida. We are pleased to have you with us, ma'am. Over to you.
Thank you very much, Sonalika. And it's my pleasure to be here at this symposium. Should I share my screen now?
Yes, ma'am, you can.
Yeah, so is my screen visible?
Yes, ma'am, it is.
Is it in the slideshow mode? Because I am not able to see it in the slideshow mode.
No, ma'am, it's not in the slideshow mode.
Presentation, just a sec.
Ma'am, maybe you can click on the icon in the extreme right.
Yeah,
Ma'am, below, extreme right below. Where the maximization.
that is what I was.
That's not working, ma'am?
I
Yeah,
hope
it's working.
Yes.
Yes, ma'am. Thank you.
Okay. Thank you. Thank you, Samadika. Okay, so very good evening, and I would like to thank Biotech, the organizers, for this opportunity. So I was asked to talk about the bioinformatics ecosystem at Amit Institute of Biotechnology, Amity University of Uttar Pradesh, the Noida campus. So I would talk to you about what are the various programs that we have in bioinformatics here. I am currently heading the Center for Computational Biology and Bioinformatics at the Amity Institute of Biotechnology. So here, the Center for Computational Biology and Bioinformatics is a part of the Amity Institute of Biotechnology, which houses around 2,500 students across various disciplines in biotechnology. And therefore, we have 5 centers in this institute. And I would be talking to you only about one center, which is Computational Biology and Bioinformatics. The other 4 centers relate to the other aspects of biotechnology, namely molecular biology or medical biotechnology or biochemical engineering and plant biotech. So here we have a very nice amalgamation of various disciplines of biotechnology, which promotes interdisciplinary research. So I take pride in introducing you to the Center for Computational Biology and Bioinformatics. So the center in this institute contains 14 faculty members, and we are covering almost the whole expanse of bioinformatics as far as our research interests are concerned. So right from molecular modeling to drug design, systems biology, systems medicine, AI and ML, genomics, proteomics, transcriptomics, clinical research. So these are the various areas in which we have faculty members who do their research. Now, before I go further, I thought I would just introduce a slide on the emphasis of the Government of India on biotechnology in the next few years. And I wanted to show you this graph, which says that which shows you the growth of the bioeconomy. That means I'm of course talking about the Indian bioeconomy here. So the way it has improved in the last 5 years, the way it has progressed, you can see that we are aiming towards a $150 million by 2025. And therefore, if you consider all the verticals of biotechnology, that means the agriculture, the bioindustrial, the biopharma, and also the bio-IT, where we specialize. So you can see that in the last 5 years, the economy has grown almost, it has doubled over the last 5 years. And we are going to go even faster in the next 5 years. And so I think this slide itself shows you the kind of opportunities that biotechnology has to offer in general. And in particular, bioinformatics. I think this pandemic has given a boost to bioinformatics. I think every common man today knows what is a virus, and in fact, NGS, what is NGS? How are we able to get the drugs for COVID-19 pandemic? And why is sequencing important? What are various strains? So these are, this is a language that has become a common man's language today. And therefore, when it comes to identifying various strains of the virus, it is the sequencing which becomes very important. And the moment we talk about sequencing, next generation sequencing, bioinformatics itself plays a very, very important role. So you can all understand the way bioinformatics is going to progress in the next 5 years. It's going to be phenomenal. And therefore, we need manpower. We need students of today who understand the various aspects of bioinformatics, who are able to contribute to the growth of bioinformatics in the coming years. And to facilitate that, at AIB Noida, we have various programs in bioinformatics at various levels, starting from bachelor level to master's to doctoral level, and also some certificate courses. So at the bachelor level, we have Btec Bioinformatics, which is a 4-year undergraduate course. And we have approximately 100 students in Btec Bioinformatics program, which amounts to approximately 20 to 25 students per batch. We also have Btec Bioinformatics International. I'll be talking to you in detail about these programs in the next slide. We also have Btec Biotechnology with a specialization in bioinformatics. At the master's level, we have 2 programs, Tech Bioinformatics and MSc Bioinformatics. Both of them are 2-year programs. At the doctoral level, we have a PhD in bioinformatics. And along with all this, we also are running a certificate course. And the certificate course is Artificial Intelligence in Healthcare and Biotechnology. We have very recently launched this course. And it has been launched with the idea of the demand of industry in AI and machine learning in the area of healthcare and biotechnology, because there is huge industry demand for these kind of trained professionals. We launched this course, and but this is available only after post graduation. So. And all these programs that we have developed here, I must mention that the Btec Bioinformatics is our flagship program, which started way back in 2004. So our first batch of bioinformatics students passed out in 2008. So we are well established into this education in bioinformatics. And over the years, we have learned and we are continuously improving our programs. We have designed these programs keeping in view the International Society for Computational Biology, the competencies that have been elucidated by ISCB, the International Society for Computational Biology. So keeping those competencies in mind and the level of each competency at different programs, we have decided, we have chalked out the objectives and the learning outcomes of every program. So bachelor level programs, the Btec Bioinformatics program is a 187 credit unit program where we teach various different aspects. We start with a biotechnology base. So in the first year, we teach various science subjects. And then we start teaching basic biotechnology subjects. And from 3rd semester onwards, that is when we're in 2nd year, when we start introducing various programming languages, the logics of programming, when we are in bioconductor, Python, and then we go into genomics, proteomics. So that's how we have evolved our program. So the basic knowledge of biotechnology is absolutely essential for any person who's studying bioinformatics. We have another program, which is a slight twist. It is Btec Bioinformatics with an honors degree. When we say it is an honors degree, that means the student has to earn 22 extra credits. So other than 187 credit units, the student needs to earn 22 extra credits to earn the honors degree. And that honors degree is available in data science and analytics, cloud computing, AI and ML. So these are the honors degrees that we offer. So in the Btec Bioinformatics International Program, it is again one of its kind program where we are into an agreement with Indiana University in the US. So the students would spend 2 years at Amity Noida with us, and for the next 2 years they would be at Indiana University US, and they would finally graduate with a degree in biomedical informatics from Indiana University. We also have Btec Biotechnology with specialization in bioinformatics. Again, the student has to earn 22 extra credits, but you will do a Btec Biotechnology and you can study 5 or 6 more subjects in bioinformatics, so you get a specialization. So that is how, what I'm trying to tell you is, we have programs which are exclusively in bioinformatics, plus we also support other programs with bioinformatics subjects. I don't know if you're aware, but most of my Indian friends would be aware that we have a new education policy that has come up in 2020. And in that, we are now going to have all 4 years of undergraduate programs, BSc programs. So in there also, we are a support center for BSc Biotechnology. So that means that we offer bioinformatics courses in BSc Biotechnology also. The master's level programs, again, NSc and Mtech. What is the difference between the two programs? As you can see, there's a difference in credit units and also the eligibility. Because in NSc Bioinformatics, the eligibility is a 3-year BSc Biotechnology or Life Sciences, whereas for Mtech Bioinformatics, if you need an admission there, then the eligibility is a 4-year Btech Biotech or Bioinformatics program. The certificate course that I was talking to you about, it is a 6-month certificate in AI in healthcare and biotechnology. We have actually developed this course very rigorously, very extensively with the Life Sciences Sector Skill Development Council. This is a Government of India enterprise, and they are focused on developing skills with higher educational institutions. So we have collaborated with them, and it is certified. This particular certificate course has been certified by LSSDC, Government of India. So even those, we actually came across a lot of people, a lot of doctors also, who wish to do an AI course. So keeping that in mind, we have launched this kind of a certificate course, and I'm sure a lot of students, we are already getting a lot of calls regarding this course, and I hope it's going to really serve the purpose it has been designed for. A very attractive option that Amity University offers is a study abroad program. So study abroad option is available for all programs, whether it is a bachelor program or a master's program, and the study abroad programs are open to all students at various campuses of Amity at London, New York, Singapore, and Dubai. Because Amity has campuses in all these places, so students from any program, if they wish, they can choose to study abroad for a few months. Now, coming to what, other than teaching, how else are students involved? We actually place a lot of emphasis on learning by doing. So other than the theoretical knowledge that we give in our classes, we want students to be hands-on. And therefore, we have various non-teaching credit courses that we call during our programs. So when the students join in after the first year, we do a term paper, which is a literature search basically. So we teach, the student gets to learn how to, read a plethora of research papers on a particular topic, and then synthesize it into a report, right, on the student's end. And we have a strong plagiarism check for it, so the students cannot cut, copy, paste anything. So this helps the students in reading and understanding, comprehending analyzing, right? Synthesizing. Then we have a summer internship. So we have students who are allotted topics by their faculty members. Students also do it at industries and academic institutions or research institutions in India and abroad. Summer internship usually lasts for 2 months. Major project is actually a high credit, non-teaching credit course where the students have to complete a project. And this is of a large duration. It varies from 3 to 6 months. Students will be required to do a major project, and these are all credit courses. So the credits are counted towards the final grade point average. Dissertation happens for master's students. So what you see on the screen are just screenshots of what the students do. So you see that there are publications in various journals, and I do not know if you're able to see, but if you're able to see names here, these are our bachelor's and master's students who are publishing in various journals. So they're publishing in scientific reports, Computers in Biology, Medicine, in Journal of Biomolecular Structure, Dynamics, and Springer Nature, General Journals, so many journals. So using these kind of trainings that we provide to students, we encourage them to publish. And that is why our students, when they apply for their post-graduation or they want to apply for jobs, I think we prepare them well for this through this initiative of ours. So continuing on student progression, how we handle this is when the student join us, we ask them to fill a student aspirational survey, and that is what this SAS stands for, Student Aspiration Survey. So the student is asked, what does the student want to do? He or she wants to go into a higher education, or the student wants to have a startup after completing their graduation, or they want to opt for a placement. So we offer on-campus placements to students, or they want to appear in competitive exams and they want to go and change their line. So once we have this kind of idea of what the student wants to do, we accordingly groom students. So we have centers for all these things, and the students are groomed accordingly, depending upon what their aspirations are. And in that, this is an example. We have had a workshop on big data and AI in biotech and healthcare in 2019. And this was in collaboration with the UMBL, BI UK. And it was sponsored by the Department of Science and Technology, the Department of Biotechnology, which are a part of the Ministry of Science and Technology, Government of India. So this was a very successful workshop that we had, and we had a lot of participants. And after this, even last year, we had a virtual symposium, which was attended by a number of participants. We had around 800 participants all over the world for this workshop. So this actually gives a very, very good, experience to our students to see what is happening across the world when people from different parts of the world come and talk about various aspects of bioinformatics. Students get a totally different perspective. These are an example of the various places where our students do their internships, their major project. So these are leading academic and research organizations in India, along with industries like Biocon, Novozymes, Premas, Serum Institute of India. So these are the various industries that our students do their research with. And since we've also had an MOU with Pine Biotech, I understand we have around 30 students who are having their internships with Pine Biotech this time. So we're looking forward to those students and the kind of work they do. And I'm sure the Pine Biotech and Amity University together, we are going to produce some very good internships. So I also thought, let me give you a quick insight into the faculty members that are present here. As I told you, we have a total of 14 faculty members here. So we're working in different areas of computer-aided drug design, network biology, that is myself, Dr. Abhishek Sen Gupta, working in systems biology, data analytics, Bhavna Rathi in immunoinformatics, Dr. Priyanka Narad working in machine learning applications in healthcare, Dr. Hina Bansal working in network pharmacology, Dr. Sneha Santoshi working on molecular dynamics, simulations, docking studies, Dr. Mukesh Chaurasya again working in quantum mechanics, molecular mechanics. Dr. Jitendra is a statistician and has immense experience in statistics, so he's an asset when it comes to statistical analysis. Dr. Kamal Raval is doing very good work in AI and ML and clinical research. So now the faculty members that you see on your screen now, these are our computer science faculty. So we have a very good, interesting mix of faculty members with a biotechnology background, with a bioinformatics background, and the computer science background. So that gives us a very good place to be in. So that is why the quality of work that students do is really good. It is of publication quality. So we can really publish the work that we do because of the input of so many different faculty members with different backgrounds. This is to give you an idea that the research projects that go on at Amity Institute of Biotechnology, we currently we have had around 113 funded research projects from various government funding agencies in India and abroad. And the combined grant that we have received is of the order of around 44 crores. And I think more than 80% of the projects that have been granted, they have some input of bioinformatics there. So I'm not saying that these projects are all bioinformatics. No, they are all biotech projects, but we have problems which we can solve using bioinformatics also. So bioinformatics comes as a very good support to the biotech problems that come up. And we have right now the Bioinformatics Center itself has around 5 projects which are running exclusively in bioinformatics. So I was also asked to give you a little understanding of the kind of research that goes on at our center here. And this is to show you some research that we have published that we've been doing. So it is in my laboratory right now. We published this paper very recently, and this was on COVID-19 therapeutics. So we chose the protein casein kinase 2, and we already know that silmetacertib is a compound that binds to casein kinase 2, and this is we tried to modify to generate a compound which has a superior performing in silmetacertib because silmetacertib has problems of toxicity and side effects. So we used bioisosteal replacement techniques and designing pharmacophores, combining them, and using molecular dynamics to come up with promising leads. So this was one of our work that we have recently published. Another work that we did was we designed a database called as PepEngine, which we also published early this year. So PepEngine is actually a manually curated compendium of peptides which contain unnatural amino acids. So we realized a lot of work has been done on peptides which contain unnatural amino acids, and these peptides have a lot of functions also. So people have been doing a lot of protein engineering. So these unnatural amino acids basically are integrated into natural peptide sequences to give them a particular secondary structure. So protein engineering in that particular field, I think this PepEngine can be a really good resource because this is a repository of various secondary structures that have been made by incorporation of unnatural amino acids. So when a person is doing a protein engineering problem, a protein design problem, and he or she wants to insert a particular secondary structure at a particular region of the protein, if they use this particular database, they can get different structures like alpha helical structures or 3-10 helices or beater sheets or turns. So this is something that we could put together and we could publish in International Journal of Peptide Research and Therapeutics. And this could also be help us in the future, endeavors of developing some machine learning models. Now, this particular peptide-based glucokinase activator, so this is a project that was funded by the Department of Biotechnology in my lab. And here we have actually done some good work here where we could do some in silico work. So glucokinase is a protein which is implicated in diabetes. So we design glucokinase activators which bind to the allosteric site of this protein. And these activators have shown to be really important in managing type 2 diabetes. So we have again used pharmacophores, and we have used from the lean GKAs, and we have used docking studies and dynamic studies to arrive at a few peptide sequences that could act as potent GKAs. And then we went a step ahead, and with my collaborators in this institute, we actually synthesized these peptides, purified them, and we did some biological activity tests. So this was quite a complete study that we could do. And this is in the process of publication right now. So these are other faculty members, Dr. Abhishek and Dr. Priyanka. So they're doing a lot of work on metabolic model reconstruction. They are doing clinical data analytics and association using AI and ML. They also are doing multi-omics integration. So Abhishek is very good at mathematical modeling. So that's how you see that we are looking at different areas of bioinformatics and we complement each other. Dr. Kamal Rawal, as I told you initially, he is leading a group on machine learning and artificial intelligence, and he has developed various pipelines from his group. And he is also heavily funded by the Government of India, the Department of Biotechnology, the Department of Science and Technology, the ICMR have all funded his projects, and he is into immunology and vaccinology. So he's done a lot of work on immunoinformatics, vaccine candidate development. So these are the various pipelines. So if you get a chance, you must try and look at these pipelines, which are which are actually an asset when it comes to vaccine discovery. And he's done a lot of work in COVID-19 also. So during the last year, during the pandemic time, a lot of bioinformatics work, a lot of ML work was done in his lab, even on COVID-19. So this was just to give you a deep peek into what our center, the Center for Computational Biology and Bioinformatics at Amity University, Uttar Pradesh, Noida campus is doing. So I think I would stop here because my rest of the slide was just about Amity University. So I think I have just I've just crossed my time, so I should stop here. So thanks a lot for your attention. Any questions, if anybody would like to ask, I would be open to it. Yeah, so
thank you so much, Dr. Paniti. I think there are questions. If we get any questions, we'll definitely mail you and share the responses back. Thank you so much. And it was a wonderful presentation and very useful actually for the students who are right now in their bachelor's maybe and want to pursue masters, or their siblings who are looking forward for various opportunities that they could get their hands on, the various degrees that they could get into and learn from the research that is definitely going on in Amity University. And the scale that you shared and you told, and the way Amity's curriculum has been designed with such integrated programs, internships, and also the projects that the students are supposed to go through are definitely very helpful for these students as they get a good practical hands-on along with the technical things that they learn about. So it was very nice to hear about what Amity University is doing. And definitely, programs are going to help students to have a better future. Yeah, so thank you so much, Dr. Puniti. And now I would, thank you, ma'am. Now I would like to invite our next speaker, Dr. Rita Sharma. I would like to introduce her. Dr. Sharma is deeply inspired by the historic innovations that led to the domestication of modern-day crop plants and is equally astonished by the fact that out of estimated 30,000 edible plant species on the earth, we human beings only cultivate about 150 of them, with more than 70% of our calories coming from only 15 crops. Further, more than 50% of these calories are contributed by only 3 cereal crops, namely rice, maize, and wheat. These choices, driven by a simpler need of feeding the increased population in the past, have to be revised in the wake of limiting resources, deteriorating environment, and changing nutritional requirements. With an overall aim to gain deeper insights into the evolution and domestication of economically important plants that led to modern day crops and facilitate de novo domestication for the development of multi-purpose climate-ready smart crops for the future, her research leverages the synergism of plant biotechnology and computational biology to facilitate data-driven precision agriculture. I'm very much excited to listen to Dr. Rita's talk. Over to you, ma'am.
Thank you very much for a very nice introduction. Let me just share my slides first. Okay, I believe my screen should be visible now.
Yes, ma'am, it's visible. It's in the slideshow mode. I think you're good to go.
Yes.
Thank you, ma'am.
Okay. Thank you. Thank you so much. So although I've been introduced more like a plant biotechnologist using computational biology, but I have prepared this presentation in a very generalized mode because I understand that the participants here may not be just interested in the plant biotechnology per se, but would like to hear more about the scenario of computational biology in the country and how its impact is increasing over the time. So yeah, so that's how I titled my presentation, that how the boundaries between biology and computational biology is shrinking in terms of education as well as research. So first, yeah. So just introducing first how my academic journey started. I have started my journey is a very pure subject, but botany with masters in botanical sciences from Guru Nanak Dev University. And then I moved ahead to University of Delhi South Campus for my PhD program, and followed by postdoctoral research at University of California, Davis. And I returned back to India in 2014 as a Ramalinga Swami Fellow in Jawaharlal Nehru University School of Life Sciences. And in between, I also went back to UC Berkeley and Lawrence Berkeley National Laboratories in United States for a visiting scientist fellowship program. And throughout this journey, which I call as a formal training that helped me establish my own research group, I was challenged with high throughput data, which was generated from high throughput technologies. So may it be the first experiment I was supposed to do in my PhD was microarrays, and then SAGE experiments involving sequencing of thousands of bases, and then followed by computational comparative genomic studies, where we could make sense of the data that we had got using high throughput technologies. And during my postdoc also, I was involved in switchgrass and foxel-minute genome sequencing projects. And then we thought of making databases to make it available to the public community and make resources for others also to use. So this is how I transitioned from a botanist to a molecular biologist to a computational biologist. And considering that exposure that I had over the time, I was appointed as assistant professor in the UGC Recharge program in Jawaharlal Nehru University in the School of Computational and Integrative Sciences. We call it as Computational and Integrative Sciences because it's a very, very unique department in the sense that it has faculty who are coming from very different disciplines. There are physics, there are chemists, there are biologists, and they all integrate their domain expertise with the computational sciences to make sense of the big data that we were generating in our groups. And in terms of education in JNU, this department hosts a postgraduate diploma in big data analytics, which was just started a few years ago. And then we had an MTech program from 2006 to 2015, which was discontinued. And then later we started master's program, MSc in computational biology, as well as PhD, where we have computational biology and complex systems. So these programs were some of the programs which were the sort of, made the inception of the computational biology in the country. And we continuously revised those curriculum to make room for the growing needs. And right now I'm at BIT Pilani. I joined here last year. And in here we have a very single interdisciplinary sort of department called Biological Sciences with 20 faculty members, all with diverse expertise. And the boundary between bioinformatics, computational biology, and biology has totally blurred because all of us use computational sciences as a tool sort of to complement the science that we do. And that's how we identify ourselves, some of us who actually use computer science tools, as computational biologists and not bioinformaticians. Because if we start thinking about differentiating between the two, you would see varied definitions available on different sources on internet, and it is not possible for every one of us to agree with one particular definition. But the one which was given by Russ Altman at Stanford University in a blog that he wrote impressed me the most, and which connects with where I feel connected, is that computational biology is the study of the biology where you are using computational techniques. So you are essentially a biologist who is using computational techniques to assist the research. So it is more about exploratory science, discovery science, and we are using computers as just one of the tools, just like we do experiments. Whereas bioinformatics is more towards creation of the tools, where tools are created to help the biological problems. So they are created for the computational biology. So for example, there could be certain tools which are available, which are designed by a bioinformatician, but I may be the user of those to solve my own particular research problem. So I would call that person maybe as a bioinformatician and myself as a computational biologist. Although these, again, definitions are blurring with the people coming with the interdisciplinary knowledge of the domain. So anyone who has a domain knowledge of biology, statistics, mathematics, and computer science can do either into bioinformatics and computational biologists, or even both. You don't have to be one of them. But how bioinformaticians were perceived decades ago was more of people who would technically help computational biologists, or in general biologists, to achieve their research goals. So they used to work like mostly as technical posts where they would develop tools for the biologists. But that changed as we entered into the era of sequencing and high throughput technologies. So even if you see the human genome sequencing project, and most of you might already know Eric Lander, who was the lead scientist who led the human genome sequencing project from MIT and Harvard, and he was basically a mathematician and a molecular biologist. He did not have any degree in computer science, and he was the one who led the human genome sequencing project, but he had some very good scientists who identified themselves as bioinformatician to provide the support for assembly and making sense of the data which was being generated by the sequencer. So, and those people changed the scenario how we look at bioinformaticians versus computational biologists, because they started joining hands together to lead the projects together instead of one working for the other person. So this is just to show you some of the lead bioinformaticians who have gone up to the extent of leading some very huge international projects. As far as Indian context is concerned, in India, more systematic investigation of bioinformatics and education started with the inception of the BTIS network, which was established by Department of Biotechnology in 1980s. Before that, there were some people who were doing by self-learning mode some of the bioinformatics things, but there was not a formal training available for bioinformatics. So in 1986, with the establishment of BTIS network, there were DICs which were established. So basically in BTIS network, this is still existing and very much functional, and is the source of connecting bioinformatics in the country, where we have several distributed information centers, bioinformatics facility centers, which have high performance and super computing facilities available. There are 7 centers of excellence which are running the programs in masters and PG programs for bioinformatics. There are distributed information sub-centers which also include Northeastern State Facility Centers, which together make a very strong foundation for the bioinformatics in the country. So there are 7 centers of excellence, there are 11 DICs, 51 sub-DICs, and 98 bioinformatics infrastructure facilities. So you can imagine that we are really quite well resourced to lead bioinformatics project. And some of the universities which were the ones who actually took the initiative, the first ones to start the bioinformatics in the country were Pune University, JNU, Calcutta, Pondicherry, and Madurai Kamraj University. And still those programs are very well designed to meet the needs. And they're all uniformly distributed across the country so that the students would have access to these centers wherever they are located. But there has been, in spite of all these bioinformatics resources, training centers, we still had the gap that students after completing these programs are not getting jobs in the industry. So what was that gap that has to be fulfilled between the academic curriculum and the industry requirements? So there has been, the people who were leading the committees in the bioinformatics boards and BTIS centers, they thought about it, that what is missing, that is not letting our students get the good jobs in industry. And after a lot of thought and all, for last couple of years now, DBT has come up with association with LSSDC, some skill development programs, as earlier speaker was also mentioning, where they have a 6-month certification program, where the 6-month or 1-year certification programs have been started in several national repute institutes, where students are they go through the curriculum which is designed in consultation with the industry, so the industry needs are met. And these certificate courses and all can be finished after your completion of the formal degree, and that can help you to have more formal education as per the industry requirements. So usually they have some component which is theoretical courses which are done as per the standard curriculum, followed by some dissertation or outdoor project which preferably has to be done in industry, so that you get a real exposure to the industry problems, and then eventually you can apply that. So this is one step that DBT has taken to help students bridge that gap and get jobs in industry. And Professor Alok Bhattacharya, who was earlier at School of Life Sciences, JNU, he's retired now, but now still very active man. He is the one who is chairing this PTIS network and has made significant contribution in comparative genomic studies with, especially with microbial genomes, mycobacteria, and so on. And then I also put the picture of my PhD supervisor, Professor Akhilesh Tyagi from University of Delhi South Campus, who was among the first people who took initiative for getting involved into international projects involving genome sequencing. So that was where the challenge started to come, that we needed to have teams who can come together to meet those deadline-oriented projects. So Professor Tyagi was involved in first rice genome sequencing, then he took up into tomato genome sequencing, and then desi chickpea when he was director of NIPGR. So these are the people who actually took, those were not computer scientists by training, they were actually life scientists, they were biologists who took the initiative, made the teams, constructed the teams, and led those projects which were heavily computational biology oriented. And but these are few people who had taken that stance. Not everybody can lead the projects at that level. So still there were people who were calling themselves as bioinformaticians and would still just like to call themselves as biologists, and then how to bring them together and initiate collaboration. So there has been several initiatives to do that also. And now DBT has started the more of national network projects where the people who are actually not bioinformaticians or computational biologists, but would like to take help of bioinformatician and computational biologists for integrated national level projects. There are now calls for proposals where they can together propose bigger network projects to help them collaborate with each other. And some of the very beautiful examples in our country where this network projects have been very successful is the National Mango Database, where a lot of institutes came together to develop a mango database, being one of the most important fruit crop in our country. Then we have Indie Genomes project, which is completed by led by the researchers at IGIB and ECSAR with other, I think 16 or 17 different institutes together to sequence more than 1,000 Indian human genomes so that you could have a representation of the Indian genomes as well. And they have constructed a database to make it freely available for others to do and extend this research. There has also been an initiative by National Mission on Education through ICT, where they have come up with the Vidwan portal, where researchers can actually register themselves with their expertise. And then using that expertise, these databases are searchable. So if you search bioinformatician or computational biologist, you will know how many bioinformatics people are working in the country, so that if you want looking for a collaboration, you would get them domain wise as well as geographically wise, where they are located, how many of them are in central universities, state universities, private universities, so that you can find the right fit for collaboration. You don't have to just depend on your local network. You can go to this database and you can find out closest to you who are the bioinformaticians, computational biologists who might be accessible, and you would like to connect with them to initiate collaboration. So I just did a quick search before this talk, and there are about more than 400 bioinformaticians and more than 1,600 computational biologists who are registered in this portal and hopefully available for collaborative initiatives. There are student groups also who are taking initiatives to connect with each other. International Society of Computational Biology has a regional student group format where the students can come together to make a regional group of any country. So there is RSG India, there is RSG US, Australia, Sri Lanka, and so on. So these student groups, they come together and then they network and they organize events. So we have an RSG India team who is very active. I'm faculty advisor for them. And they have been doing certain events to bring together students and invite experts based on the needs of the students whom they feel they would like to listen to. And you can also network amongst each other to talk about what kind of research you are doing with each other. So I encourage the students to go and check the RSG India website and the events which they are organizing. Also, we don't have to just network in between the country. We also have to work with international levels. So there are challenges why India is it is now expanding and growing. So we have to have our place at the international forum also in computational biology and bioinformatics. And for that also, there are several initiatives which have been started. And one of them is where I was also one of the PIs when I was at JNU, is the research training group, which is mostly focused on big data research. That is an initiative between DBT and Heidelberg, Germany, for a combined graduate degree. So the idea is that the students in the participating institutes from India will go to Germany for a few months, get a training there, and then they would have a joint degree by the host institute as well as the Heidelberg host partner. So there are institutes like JNU, IIT Madras, IIT Kanpur, IIT Guwahati who are participating in this, and some of the students have already completed their training in Heidelberg for the making this program a reality. So these are some very wonderful programs that students, if they are aware, can take advantage of. Also, in terms of computing infrastructure, you might have heard about the first supercomputers being installed in IIT Delhi, but now we have several institutes of eminence, universities where the supercomputers have been installed and are in the process, so that the dream of having a national supercomputing mission can be accomplished over the years in the future, so that won't be a limit for us to think of ambitious projects. Also, there are people who are taking initiatives to bring the science in India on international forum. One of those examples is APBN Society. It's the Asia Pacific Bioinformatics Interaction and Networking Society, which is registered in Delhi. And some of the faculty at JNU and Calcutta, Boston University, and Assam University, and so on, are the founding members who came together. They're all bioinformaticians, computational biologists. I'm also one of them. We and then made this society where we can organize international conferences. The first international conference was organized in 2018 in Perth, and hopefully there'll be more which will place the Indian bioinformaticians and computational biologists on the international forum, where we can talk to each other, discuss our science, and then initiate international collaborations. And now, as I said, I identify myself as a computational biologist, but the computational biologist doesn't mean that, it's a different stream. Slowly, the gap between computational biology and biology is also minimizing. A lot of students, when they come, they say, I'm not good at computer, so I just want to do wet lab experiments. But I tell them that that's not going to sustain you, because slowly in biology also, we are moving from single gene to genome level, from single RNA to transcriptomes, from single protein to proteomes, from one metabolite to metabolome. So science is transforming very, very fast. And the way the technology is becoming so cheap, it is so cheap to study at genome level. Why would you look at just one gene when you can actually look at the whole genome? So, and then because of this, the integration of all these would require quantitative skills, mathematical skills, and computation skills. So if you want to really survive in biology, you will see that slowly these two are actually merging together to make one stream. All computational biology and biology is going to be the same. If you are doing biology, you can't say that I cannot use computers, because computers have now just become one tool in the arsenal of the biologist, not a separate science anymore. And at BITS Pilani, we have students who are doing Btec in mechanical engineering, in chemical engineering, computer science, and so on. And they come to us, and then they talk to our biological projects, and they apply their knowledge, whether it is engineering, computer science, and they do short-term projects where they to combine those disciplines together to come up with the innovative solutions. And that is how, I think where BITS Pilani leads and innovates, because it is combining together. It is not looking at these streams as separate streams. The students from any stream can go to faculty of any stream and then do the projects with them. And that is the beauty of interdisciplinary nature of science, where you can combine the knowledge together. Now, there's just the last one thing I would like to share about that. As I said, a lot of us are generating a lot of data. In my lab also, I generate a lot of transcriptomics data, and I had handled proteomics, metabolomics projects over the time. But what happens is that a lot of labs who generate this data, either they will submit it to some international repositories, if that too is mandatory by the journal. So when you are publishing a paper, some journals mandate that you have to submit to a national repository. And in those cases, you are bound to submit. But if it is not required, you may not submit. And if it is not published, then obviously most of the people won't submit and make it publicly available. So that makes a huge gap that, the Indian funding agencies are funding these huge projects, and then the data stays in one lab, stuck in one computer, and then it is never accessible to others. And so it is important for us to be able to access at least the data which is generated from Indian cultivars, Indian varieties, Indian strains, Indian populations. And so with that vision in mind, DBT has initiated an Indian Biological Data Center, which is being hosted at RCB Faridabad, and it is having clusters together at Bhubaneswar, NIC, and RCB. And Dr. Saurav Raghuvanshi, who also played a very instrumental role in rice genome sequencing project with Professor Akhilesh Tyagi at Delhi University, is leading this initiative, where they are trying to make an Indian database for all the different type of biological data that we generate in India using the public funding, and so that would be accessible by Indian researchers. And the idea is that whether the data is coming from agriculture, livestock, microbes, humans, doesn't matter. Anything which is being generated in India should be put into one database, and that it should be searchable and it should be available to the community for use and expand and build on that research. Right now, they're running the pilot project and they are working on mostly nucleotide database, but they have a plan to work on different domains, where domain specifically, there will be different domains for different types of data in this database, and that will be accessible to the community. As far as my lab is concerned, as I have already been introduced, I am a plant biotechnologist, and I'm mostly interested in crop improvement. And for that, we usually start with selection of some of the lines, some of the seeds that we want to start with, and ultimately we want to make an improved crop out of it, right? But then the work that goes in between, from selecting the germplasm to having an improved crop, is tremendous, because you have, when you talk about a genome, you have thousands of genes, and you don't know which one should be, edited to get an improved crop. And you cannot edit those thousands of genes, because as far as editing and transformation in plants is concerned, we are very, very limited in terms of technology. It takes about 6 months to 1 year to be able to have some validated transgenic lines for single gene. So this is not even possible for us to even think of characterizing thousands of genes. And this is where computational biology has always come very handy in my own research, where we use high throughput technologies to generate data at genome scale, and then use other tools like phylogenomics combined with the pathway mapping and all to shortlist the candidates, which are can then be again brought into the lab for precise editing, either using genome editing tools or the other conventional tools to improve the crop plants. So this is how my research is being also benefited as a computation with the computational biology. And by just accepting that computational biology is not a different science, it is one of my tools, and if I want to make progress, I have to learn. I think anyone can make change, and that takes your research at a very different level. So with that take home lesson, I think I'll just stop here and let the host take over, and if there are any questions, I'll be happy to answer.
Thank you so much, ma'am. So are there any questions? You can please put them in the chat box. And also thank you so much, ma'am. The way you beautifully explained how computational biology and biology are merged together has helped me a lot to make this community understand as well. Because as a community manager, I keep on getting questions from biotechnology students who say, how to get started with computational biology, or why shall we do it, or how to do it. They're scared maybe. And we keep on asking such questions because they are just confused. So your talk has definitely given such students a direction that the knowledge of computational tools is definitely very important to excel in these times. So thank you so much,
Thank you very much.
And also the information that you gave about these groups, it was very much helpful. Okay, so I have a question. What is the skill that is most needed in bioinformatics for a student? What do you think?
See, I can't pinpoint one because that depends, as I said, on your research question, right? So I know students who don't even know programming, but they are still able to handle data with the publicly available platforms like Galaxy, where you don't have to really write the programs. So, and there are certain areas where you have to write a program because there is nothing available. So it totally depends on the kind of research area you are working on and what resources are available for that particular system. If you are working on a model system, you probably would have a lot of information and you may not have to go back. And people would have already developed programs and algorithms to help you, and databases which might be available. But if you're working in a challenging new system, then you have to again start from scratch. So I think it totally depends on what you are working on. But what I would say is that I don't think there is anything that can limit. Starting from scratch just makes you confident enough that you can do it again if required, right?
Yes, ma'am. I completely agree with you. The next question is there a demand for computational biologists in agriculture in India?
Of course, there is a lot of demand. See, when you look at the demand, you would see that a lot of opportunities, they come in the projects which are mostly related to human genomes and microbial genomes, because of the amount of funding which goes in the medical sciences, health informatics. The data which is generated in animal sciences is usually much higher folds compared to plant sciences, because the amount of funding that goes in plant sciences is not equivalent to what goes in health research. So usually when we talk about big data, the big data usually comes from the human resources. So that's why you see most of the employment opportunities staggered towards those projects which are focused. If you're talking about last 2 years, a lot of funding has gone into COVID related research, which makes sense and is a sensible approach. But in case of plants, although we are not generating the data at the scale where human and microbial genomes are being sequenced and data is getting accumulated, but that does not mean that there is not enough requirement, because although We are not sequencing thousands of genomes, but then people in agricultural sciences, because of the lack of funding, are very, very selective. And the kind of polymorphism, the kind of variation which is present in the plant genomes is much more daunting. Assembling a human genome would, I guess, would be easier than assembling a complex polyploid plant genome. It takes years. I worked with switchgrass genome sequencing, and it took us 10 years from beginning to it is a allopolyploid, which means it has 2 genomes and it's a huge genome. And just coming up with the complete assembly was more than 10-year project. So there are a lot of daunting and challenging projects which are available in agriculture as well. And as we are heading towards understanding the requirements in agricultural sciences, these projects are going to increase over the time, especially with the directive that has come yesterday from Indian government, where now CRISPR edited crops are not going to be assessed for safety requirements. A lot of people would now try to put their foot into this area to be able to generate improved crops. And again, as I said, for generating improved crops, you have to take help of computational biology. So of course, as I said, the demand is there and it is increasing over the time. And we would love to have some very good, bioinformatics students coming and joining our group and contributing in the research.
Thank you, ma'am. And the next question is, what would be an acceptable level of education required for one to get a job in bioinformatics? What do you think?
In bioinformatics, when you talk about job, there are jobs at different levels. So that also depends whether you're looking for a job in industry or academia, whether you want to go into a technical team, you want to join as a research analyst, you want to join as a data scientist. Minimum requirements would actually again depend on the job, the kind of job, and the growth opportunities available in that particular area. So I don't think there's a single answer to that question as well. I'm sorry.
Right, ma'am. And Habiba is asking, does one need to know how to code to use bioinformatics tools, and which is the best language, R or Python?
R and Python, I think both are really important for any student to excel in bioinformatics, because I don't think they're as complicated as C++ and all, Java and all. If you are the starter, a beginner, there are certain things where R is more helpful compared to Python, and there are certain things where Python. So I would suggest you to do both, at least R and Python. And they're not very difficult languages. And once you understand them, probably there is nothing much that you cannot do without having to know other languages. So, but yes, these two I think are equally important.
Right, ma'am. Just one last question. Is there a repository for accessing agricultural omics data from India?
The Indian databases, so far, as I said, are either individual centric, like for example, the Mango database, which is only for Mango. And then we have this rice IRGSP database, Genome India Initiative, which is for accessing the rice database. And similarly for chickpea. So depending upon the crop plant, either there are individual crop databases, but there is no one single repository for all the plant genomes, especially for the varieties which have been sequenced in India. And this Indian Biological Database Initiative, DBT, is the one which is going to fill this gap in coming years. Otherwise, if you look at the global way, then Phytosome is one repository where you would find the plant genome sequences which have been initiated by the Joint Genome Institute in USA. By far as I know, that is the biggest repository for accessing the plant genome data specifically. But that does not go only with the plant Indian varieties. That's the varieties which have been chosen to sequence, but those are not just limited to varieties which are in United States. There are some Indian varieties also which are there in their pool, and they also have pan genomes for certain crop species. So I think for now that will be a useful repository for you if you're working on plants, but over the time, hopefully the IBDC would also have this information from the genomes which are being sequenced in India.
That's quite interesting. Thank you so much, ma'am, for all the information. And it seems there are a lot of curious people who have so many questions, and they are still some questions pending, but I'm so sorry I won't be able to take them now. I'll be mailing you, ma'am, and then I'll get back to the audience with the answers because it's time
Sure.
for our next talk. So thank you so much, ma'am, once again. Now, moving
Thank you. My
ahead, I
pleasure.
would like to invite our next speaker, Dr. Gaurav Ahuja. We'll be talking on evolving role of bioinformatics in universities and research labs in India. I would like to introduce him. He has graduated summa cum laude from the University of Cologne, Germany, with a PhD in natural sciences. And after his completion of his PhD degree, he worked as a postdoctoral fellow at several renowned institutions, including the Center for Molecular Medicine, Max Planck Institute for Aging, and Lee Kong Khan School of Medicine, Singapore. He has published 14 research articles in peer-reviewed international journals with approximately 250 citations. In addition to this, he has also reviewed articles submitted to Nature Publishing Group, Cell Death and Diseases and Scientific Reports to be more specific. And recently, Dr. Ahuja was shortlisted for prestigious Inspire Faculty Fellowship. We are pleased to have you with us, sir. Over to you.
Hi, thank you. I mean, can you hear me? I mean, anybody can please confirm if you guys can hear
Yes,
me.
sir. Yes, sir. We can hear you.
Thank you. And now let me quickly share my screen as well. I hope my screen is visible.
Yes, that's visible. I can see the Zoom. Yeah, I can see your presentation now.
Great. So I think, thank you, by the way, thank you for the whole Pine Bio team for having me today. And also thank you for all the participants to spare some time to listen to my experience, I would say rather. So when I got this invitation, I was a little bit confused. It was not a confusion like what I'm going to share with you guys, but the confusion was more like whether I could share with you the science which we are doing in the lab, which is a little bit boring for all of you. Or on the other hand, I would rather share with you the futuristic goals, like how I meant, shape up my career in, bioinformatics, or you can say computational biology, and how basically, we train in our department, because our department, Triple IT Delhi, is one of the, topmost departments where we have a dedicated programs for computational biology. So I was thinking, like many of you may be looking for opportunities or some sort of guidance from where you can do a dedicated program in computational biology, what all are the courses they teach, how it is different from bioinformatics, even if I can do a master's or bachelor's or a PhD in computational biology, what all are my futuristic, what I will do after my PhD, for instance. So there are some of the things I thought like I should rather combine all these instead of boring you guys with the science which I do in my lab. So nevertheless, once again, I'm Gaurav. I'm a assistant professor at the Triple IT Delhi. So it is a very small but a very beautiful institute in the center of Delhi. And yeah, and it primarily, as you can see from the name, it's an information technology. So it's an IT institute, but normally, we have mixed everything with IT. For instance, like we have computer science, we have computer science with biology, we have courses for the, computer science with social science. So everything is mixed with a flavor of computer science. That is what I will say is very unique to Triple IT Delhi. So let me give you a brief glimpse about who I am. What I was one among you guys. Like today, you guys are here. I mean, almost like 10 years back, 12 years back, I was sitting at your side, and I also used to listen to, not online though, but offline. But I was also one of the academic enthusiasts, like one of you, because you guys are sparing your time listening to the experts in the field. I also did that when I was a student. So I just want to give you a very brief glimpse. Like, I started with BSc in biotechnology from Jamia Millia Islamia. It's a university in Delhi. After that, I did masters in genomics from Madurai Kamaraj University. Then I got some sort of international scholarship. I went to Germany from the Institute of Genetics Cologne. I did my PhD and also a little bit postdoc over there. Then I moved to the CCAD, CMMC, and Max Planck Institute, where I did my 3 years postdoc. Then I was in a Lee Kong Khan School of Medicine, and it was in Singapore. I did a another senior postdoc, and in the middle of 2019, I started my own group. I just wanted to make a point here is when I started my group, my age was 29. So the first faculty offer, which I got it from Inspire faculty in 2019, I was 28 and a half or 29 years old, and I declined that offer because I mean, there are multiple reasons for that, scientific ones, but I just wanted to tell you guys one thing. So, you guys are building your career, or you're trying to build your career now. So if you do compromises with the science at this stage, for example, for every researcher, the faculty job is one of the dreams anyone can look for. And it was really hard for me when I declined this offer because I was not sure whether next year I will get something similar or not. But I still took the chance. I declined it because I know the power of the science. I know what I was doing. And luckily enough, within another 1 year, I got a faculty offers from 2 institutes, and I joined Delhi because I belongs to Delhi. So I wanted to be in Delhi. So I joined IIIT Delhi in July. And then afterwards, the things got changed. We got a number of, we got 3, 4 grants money wise in India. If you do bioinformatics, if you do, at least my lab is a sort of hybrid lab. We do experimental biology. We also do, computational biology, both of the, like we do 60-40. So in all the cases, like the funds is never been a bottleneck, at least for the last 2 years, I'm doing science in India. Just wanted to make another point here. From 2010 until 2019 mid, I was never being exposed to the Indian science. And I will tell you, I really thoroughly enjoying doing science in India, at least in IIIT Delhi. The reason being, the institute is supportive, the people are supportive, and the overall ambience really allows you to, flourish, to do great in science. That is something IIIT provides us. Now, without boring you much, that's the only slide about science from the lab. So my lab works on smell. I mean, the sense of smell. We use R, we use Python. I was listening to Professor Sharma. She was, insisting about learning R and Python. These are the, two big languages which are commonly being used in data science. So we use on routine basis both of them because, when in my lab, for example, I have 6 PhD students, 12 interns. So total the lab length is around, size is around 20 people in the group. Some people have an innate preference for Python, and some people have an innate preference for R. And also there are people like who are more, comfortable with Python, but sometimes for certain packages, they like R. So they do, shifting between left and right. So the best answer is to learn both of them. And there are multiple ways. For instance, you like the interface of Python and you just want to run one package of R. There are multiple libraries. You can basically use one and you call the function of the other. One example is reticulate library. You can really do, R codes, Python codes in R, and so far and so on. So you can really interchange between them. Now we also do, study about evolution in the lab. So there are some biological problems we solve related to evolution. Then we also do, as I said, I have a hybrid lab. I am fortunate to be funded by IIIT Delhi, Department of Biotechnology, Government of India, from the DST as well as from SERB. My lab is funded, so we do also do some sort of molecular biology. And last but not the least, we also do, we work with yeast as a model organism. So that is all about my science. So let me introduce you to the place which you guys may intend to do your, that's one of the, I will say one of the most, one of the most prestigious institute if in India, if you're looking for a computational biology course. Let me put it as a case study and let me you guys introduce you to our department at the Center of Computational Biology here. This is our institute. This is one of the building. I just put it like just to excite you. So we are small. This is not something we all have. We have multiple other buildings, but this is the central one, the first one and the old one. So this is our academic block. There we have lecture hall. And, at the corner of somewhere here, we have our experimental lab. And on the right side, we have a research and developmental block where we have, our dry lab, which we say, computational biology labs. So this is a glimpse of our institute. Very beautiful, neat and clean. but a little bit small compared to if you talk about IIT Delhi, if you talk about compare with JNU and other universities, we are very small. But cleanness wise and also, productivity wise, we are really, ahead. Now, this is our department. I think there are multiple people, you guys. I mean, why I wanted to introduce you to all of them, because that gives you a glimpse, like what is a core computational biology department. Now, some of the students may be thinking and may be wondering, like why there is an intermix between a computational biology and bioinformatics. I mean, I think this is always a tool which is used by many people, especially the people who are in the computational field. Some people like to call themselves as a computational biologist rather than bioinformatician, because they think, really think bioinformaticians are the ones who basically use the tools, they use the packages, they use the pre-cooked hard work of somebody else, and they just do the data analysis. So that is, I don't want to go into this fights between whether you call yourself as a bioinformatician or you call yourself as a computational biologist, because I always believe the things needs to be done, it should be done, irrespective whether you are using a package or you are rebuilding the Rome. That is all depends upon you. So we have these wonderful colleagues in the department. So I start with, Professor GPS Raghav. I think he was also one of the persons who was invited by Pine Bio for delivering a lecture. He is one of the pioneers in India in the field of bioinformatics. He has got multiple awards. He has published so many bioinformatic papers, having multiple servers, open source platforms he contributed. So he is heading our department. We have Dibarka Sen Gupta. He is a trained, computer scientist. I just wanted to say his training was, you can see like here, his PhD was in computer science with a, some portion on the biological data, but he's trained as a computer scientist. Now we have Arjun Ray. He is also my colleague. He is basically a biophysicist. He works with the protein modeling. So this is another dimension. So think about it, like this is our department. So Professor Raghava being senior, being experienced, he is basically mentoring everything. We have Dibarka Sen Gupta, he has expertise from the computer science. We have Arjun Ray, he's a biophysicist. Same with Ganesh Bagler, he is more into network biology, and he's also a physicist by training and doing biology. Now it's me. I mean, my PhD is in genetics. As I said, I'm a trained genomist. That is what me, filling the gaps. Now we have Jaspreet. She works on chemoinformatics. We have Vibhor Kumar. He works with, so he's MTech from IIT Delhi, PhD from Helsinki, and he is basically experienced with, your epigenetics. We have Tapritesh. He's a medical doctor, and he also did PhD after MBBS, and he works with the healthcare system and all that. We have Shriram. He is a mathematician by training. So he used mathematics to make models to understand the biological system. And now recently we have been lucky enough to get Professor Murugan, who was a associate professor in the KTH in Stockholm and recently joined our department. Now, having said these things, so, and he's a PhD from IISc Bangalore, and he works with, chemistry, pure chemistry. Now, this is the vision of our department. So the vision of our department is to become a leading center in the country for developing the mathematical and the computational techniques for understanding the biological system. So think about it, like we are not that sort of department where you have, faculty members who are trained in biology, learned bioinformatics and teaching you. No, the department is crafted in such a manner. Each and every person in the department is bringing an orthogonal dimension, which basically is necessary, super important to do interdisciplinary research, which is the prerequisite for computational biology. Now, so these are all the names. These are basically from where these people are. Now, just quickly, so what we teach, what courses we cover as a part of our computational biology course. So we have, this is something which I'm showing you for, the Btec and the MTech. So we have foundations of biology, because there are multiple people we get in our institute. They are not from biological background. So, but they still want to learn, computational biology. So we have a foundation course so that they can have a feeling of biological system. So that we cover in the foundations of biology. We also introduce them to the quantitative biology, a little bit mixture of mathematics with biology so that they can better train for the next step. We also have a hardcore cell biology and biochemistry. We also have genetics, and we also have molecular biology for them. We have algorithms. So we also teach. So there are dedicated core courses for the algorithms in the computational biology. Because you know that, in case you are using a tool, it's fine. But in case you want to make a tool, you should have a better understanding of the algorithms. So we teach them the core algorithms which are being used or which may be used in computational biology. So we have a dedicated course for that. We have a network biology course. We have chemoinformatics. We have systems and synthetic biology course. We have computational neuroscience. We have a course in the data science in genomics. We have dedicated courses for the machine learning for the biomedical applications, big data mining in healthcare, simulations and systems biology, network science, biostatistics, and statistical computation. So these are some of them. Once you are in our department, every student, irrespective whether you are a bachelor student, you are a master student, or you are a PhD student, you will be given the choices to attend any of the elective courses. So IIIT hosts a lot of elective courses. We have hardcore mathematical courses. We have applied mathematics. For statistics, I can say we have 5 different courses for statistics. Each of them is very specialized. For instance, I used to teach, biostats. So it is more about, using R to, do descriptive statistics as well as inferential statistics with the biological data. But in case you are more interested in more hardcore statistics, you can easily, opt for some other courses. So every possibility is being provided by the department to the students. Now, moving a little bit more forward, what courses IIIT Delhi Computational Biology Department offers you? So we are a very young department. We started in 2017. And, we have a PhD in computational biology. We have Mtech course in computational biology, which is funded by DBT and AICTE. So which means that if you are opted for a Mtech in computational biology, so DBT funds you. DBT funds you for your, you get some sort of small stipendia or stipend. And apart from that, you also get, some contingency money for your thesis and all that. So this program is supported by the DBT. And you also get a little bit money to do some sort of experiments. You want to buy something. So all this support we get from the DBT and AICTE. Now, we also offer a Btec course, but there is a caveat. The caveat is the Btec course is not computational biology. We call it as CS plus B. Plus B is, normally it is a tailored course for computer science. So all the courses which a computer science graduate is basically, or the undergrad is basically learning, you will also have the same courses. But at the same time, you will have additional courses, which is the bio-related courses. So that's why we call it as Btec in CSB. CSB is for computer science and biology. So that is the Btec program. Now we have Btec in computational biology minor. If somebody has questions, we can take into account. So we also provide postdoctoral training. A lot of postdocs are there in our department. We also have external interns. For example, in all my colleagues' lab, even in my lab, we get people from other institutes. So if any one of you looking for internship, want to contribute to or want to learn, contribution is too early, I think, it depends. But if you want to learn, get your hands dirty with, computational biology, you are most welcome to work as an intern. We have the PG diploma students as well. IIIT has a joint data science program with IBM. So we also basically the faculty members from the department also teaches to the these PG diploma students. So this is the curricular activities provided by the department. Now a little bit more forward, what is the strength of our department? So the core strength of our department is we offer holistic, interdisciplinary, and active learning approach. We are very intensive for computationally and programming. All our courses are really intensive for computationally as well as programming. So why I'm saying this, because for instance, in our bachelor's and master's program, before the students joins the department, we normally have, a training, like we call it as preparatory module. We really teach them the programming skills so that whenever the real session starts, semester starts, they are well equipped with the jargons of programming. We have a lot of mathematical courses, as I said, to make the students comfortable and strong with basic mathematics, which is required to do any cutting edge work. We also have intensive biology courses, including the laboratory, because as Dr. Sharma was also mentioning, like every computational biologist, they also produce data, because that is something the need of the hour is. So the department recognized over the years, it's easy. I mean, it was all good. Like our students were analyzing data. They were, building algorithms. They were basically making softwares. But at the same time, it was really important to teach them, because, Btech and Mtech programs are the learning programs. So during this time, it will be really nice if they can know, I mean, they can have some images, like, some sort of microscopic images. Our students take few minutes or 2 days to develop a very sophisticated architecture to do deep learning architect. But the point is, if they don't understand from where this image is coming, then there is no sense of doing that. So that's why the departments thought it's better to have a dedicated laboratory, and from the help of DBT, we set up a fully functional experimental laboratory in IIIT Delhi. We also, as I said, we do special preparatory modules for biocomputing, so we call it as for biocomputing. We also provide research and industrial internships. Now, apart from this, we also organize a lot of scientific talks, blah blah blah. There are so many things. The department also offers core courses, a lot of elective courses, and also departmental courses you can opt. For instance, you are an MTech student in our department, and you are also interested in a special course, for example, that is provided by the social science department. If you have the liberty, being in IIIT Delhi, you can opt for cross-departmental elective courses. You are allowed to do that. Now, we also do PG diploma. Now, our faculty expertise, as I said, it's very diverse. We have faculties experts in bioinformatics, mathematics and computing, chemoinformatics, medicine or health informatics, structural biology, genomics, molecular biology, biophysics, AI, ML for health. So quickly think about, research, innovation and extension. Very slowly, I'm telling you. So it is the data for the last year. So the last year, the total citation come to our department, we are 10 faculty members. In total, we got around 4,000 citations. There were 63 publications came out, from our department. This is not the total impact factor. This is the average impact factor. It is not total. It's wrong. So now, on the other hand, applied research. So we have developed 7 databases, 26 web servers, and 3 mobile applications within 1 year. In terms about innovation, our department has made a technology transfer, which means, we talk about bioinformatics research. So me and my colleague, Dibarka Sen Gupta, we developed something back in 2019, and we give it to the we patented it, and the patent, we sold it to a private company. I will be talking about it next, like how you make a transition. Being a data scientist, you develop something, and then you basically, there are people who are interested in that product, and then you can do the technology transfer. We did that. And during pandemic, there are multiple things the department has done. 4 of them are there. Now, there are multiple faculty colleagues because they are very aggressive in their research. We have 2 startups from our department, and we also have a PG diploma program, which is IBM IIIT Delhi. So these are some glimpses about our department, how vibrant, how dynamic we are in terms of computational biology research. Now, as I said, we also have a lot of infrastructure. So infrastructure-wise, it's not a problem. I just give you one example. So my own lab, it is one among 10 labs in IIIT Delhi. My own lab has 4 personal servers. I have my 4 servers, which has if I tell you in terms of RAM, we have more than 1 TB of RAM for like 6 PhD students. So we are very generously funded by Government of India, different agencies, also by the institute. And these are the common facility which we have. So the upper one and the lower one, these are the institute. It's not an institute, department department also have their own servers. So these are the departmental servers, but each and every faculty member have their own personalized servers as well. Now, as I said, these are just the equipment. Some of the equipments I put it from the experimental lab, which we have qPCRs, Western blots, we have spectrophotometers, multi-channel fluorescent plate readers. We have, I mean, you think of something except sequencer, we have most of the equipments. And mass spectrometer, we have most of the equipments, and we are planning to purchase that as well in the near future. Now, I just want to make a case study, of computational biology in India. So as I said, we have made a technology transfer, and it was brought up. It led to the, generation of Care Onco Biotech. So this was the research carried forward by Dibaka Singh Gupta, my colleague in the department, and myself. So we did that. We, patented this technology, and it is now basically being validated for more than 400 patients. And we are soon going in the Indian market as a liquid biopsy test for cancer. So let me introduce you guys, and there I stop. So what basically happened is Dibaka, one of my colleagues, so he basically came to me when I recently joined IIIT Delhi with some of the complex problems, because he was trying to solve the tumor-educated platelets. So each one of you know, like your body has platelets, but people eventually over the years recognize the platelets in our body get tailored or get transformed if there is a cancer in the body. So the point is, if there is a cancer, the cancer can be sensed by the platelets in the body, and you can isolate the platelets, and then you can screen for the biomarkers in the platelets. You can able to detect the presence of cancer. So this is called liquid biopsy. Like we do solid biopsy, the doctor do the surgery, they take the piece, they take a piece of the tissue, solid tissue, they call it as tissue biopsy. In the same way, either you can take urine, you can take saliva, you can take blood, and then you are basically looking for cancer. It comes under liquid biopsy. So we developed this tumor-educated platelets, and now it is a part of Care Onco. We are one of the founding, the scientific co-founders of Care Onco Biotech Private Limited in New Delhi. Now, so this is the team. This is Dibaka Singh Gupta. So he is the one, and it's me, Gaurav, it's Ashok Juneja, Pallav Mitra, our CEO, Avinash Malhotra. We have wonderful ladies in our, these things. So this is all about us. Now, what is the burden of cancer? So the late detection. So one of the problems with the cancer is we detect the cancer late, and late detection leads to high mortality. Now, the cancer burden is, we know that there are 19.3 million people die because of cancer every year. And the reason for that is the cancer is detected late. 75% of the cancers is always been detected late. And since it is detected late, there is a high mortality. A lot of people die. And also, if you detect the cancer late, if you want to cure the patient, you have to do chemotherapy, radiations, and immunotherapy. So the overall cost is really high. So what we developed in IIIT Delhi. So in IIIT Delhi, we developed an assay which can be done in 2.5 hours, and there is a lot of AI involved. So doing a qPCR is not a problem, because qPCR is just a simple steps. Any Tom, Dick, and Harry with a training of 2 weeks can able to do qPCR. And thanks to this COVID, because everybody's know about qPCR, how it is done. So we have not done any rocket science in this part. We have done the rocket science mainly in AI. So we have developed, with the help of computational biology training, we have developed the AI-based platform where you project the CT values coming from the platelets of the any patient, healthy patient as well as the tumor patient. We applied a algorithm called Copula. You can read about it, and it is one of the nicest method to basically do the classification as well as to do the batch correction. So we applied that, and then we basically implemented the AI algorithms, and we basically can classify the tumor patients as well as the healthy cohorts with the accuracy around more than 90%. And to be honest, there is no method to date which can achieve this much high sensitivity as well as high specificity. So we are one of the upcoming methods which are astonishingly very high sensitivity as well as specificity. This was our paper. So the entire, you give us the blood, we do the testing, we give the report, and the whole procedure takes around 2.5 hours. That's all. And if you see the other players in the market, like there are people who do liquid biopsy, but they require 25 ml of the blood. The problem is the cancer, if you are a cancer patient, and if you are trying to measure the relapse of cancer, you cannot give them 25 ml of the blood. So all the other methods available in the market, they require high volume of blood, 15 to 25 ml. But our method works with the 6 ml of blood. It can detect 10 plus, cancers. You don't need anything. It just basically works with the normal molecular biology lab. We can detect stage 1 cancer as well. Accuracy is around 94%, and it's very affordable. So it's $100 to $150. You can really do the testing. So, I haven't talked about science. Sorry for that, but these are all just a glimpse of papers. I was able to get it from IIIT Delhi in the last 2 years from my lab, as either the senior author or the co-corresponding author from the lab. So these are the journals where we published in the last 2 years from IIIT Delhi. With that, I would like to thank my team, my PhD students. A lot of my, we have a junior research engineer who is basically doing the lab management, Nilesh. We have the previous students from the lab, for example, Siddhant Kalra. He was the MTech student, so now he's in the US doing his PhD. We have Ria, she was a Btech student, did her project with me, and now she got a job in Google, working for the Google India. Vrinda, she is also a Btech student, worked in our group. Now she is working in Microsoft India. Sidra, she got a PhD. She was a MTech student. Now she is a PhD in Aachen in Germany. Radhika, she is doing job in India. She was my MTech student, all my collaborators. And thanks to the funding agencies, and thank you to each one of you. Thank you. I'm open to questions. I'm sorry I took a little bit more time. Thank you.
Thank you so much, sir. It's perfectly fine. And if you have any questions, please put them in the chat box, and we'll be happy to take them. And to add here, we definitely knew about the laurels of IIIT Delhi, sir. But today you have told us about it in more detail. So we are glad to know about all the efforts that IIIT Delhi has been working on and is undertaking. So every bioinformatician does know who Dr. Raghav is and Raghava is and what is he doing. Yeah, he's the father of bioinformatics, I'll say, for most of the bioinformaticians in India.
Yeah, that's true. But I think many of the other members are there. So I thought it's better to introduce the department. And also there are many students are there. So that's why.
Yes, sir. So it was very nice and wonderful to know all of you over there and know about the team that's working really, really hard.
Yeah, thank you.
Okay. Thank you, sir. So we have a question. Okay, how can someone join your research projects in your lab that you're working on?
So I think in case you want to only join for the projects, there are two. I don't know which level you are asking for. For instance, if you are a Btech student, then in that particular case, you just drop and you go to our departmental website. You can look for the faculty colleagues with whom you want to work with. Drop them an email. In 90% of the cases, you get an email reply within 2 days. So, and in case they really find you to be motivated, they will take you. Now for the MTech, if you want to do the MTech thesis with them, you can write to them. You can ask them whether they have a position for the MTech thesis. We do have positions for the external members. We can have them as well. And there are also JRF positions we do timely advertise from our institute website and also on social media. So they can apply. So there is no stoppage to apply to our department.
Okay, sir. So I think, Ojasi, you have your answer. And the next question is, what are the most promising directions now using bioinformatics? What are your takes on it?
So, if you really ask me, I think it's better to get specialized a little bit later in your career than too early. Because I think the fundamentals needs to be strong. That's my advice. So guys, whoever asked this question, pardon me if it doesn't suit your if you are not anticipating this. But to be honest with you, I will say just make, I mean, if anybody asked me, I would say the bachelor should be in a fundamental science, like, either computer science or core biology. Whatever the transitions you should make is in your master's. That is where you need specialization. Otherwise, if your fundamentals are weak, because the more specialized you go from the beginning, you will miss a lot of fundamental things, which normally it's detrimental, So the best thing is you do, I mean, so the question was about when they've, sorry, can you repeat the question, Sonalika?
The most promising directions now using
Okay. So if you are already dived into computational biology, that is the answer for those people. So if you are already doing that, I would rather say everything is exciting, but the most hottest nowadays is AI ML. Everybody know about it. But also my advice is don't basically do AI ML just like, go to sklearn, get the code, just run it, or you make a classifier. Don't do that. Because, nowadays, there are multiple softwares. You can easily build model. Making a model is not something exciting nowadays. Getting a new algorithm, getting how to, even if you are not capable enough to make a new model itself, like the new algo for the binary classifications or classification or regression. But in that particular case, at least the architecture which you can design, just design elegantly. So don't do the tutorials and make a model. That is something I will say. And the exciting thing is taking the genomics data, use AI ML. That is the hottest thing nowadays in the market.
Right, sir. Absolutely right. I completely agree with you on this, sir. So students are asking, these are PhD students and Btech students, about how can they join the department as an intern, like what are the internship opportunities in the department for CSE students?
For CSE, I mean, the point is, there are no restrictions. I will answer this question the other way around. In our department, there are no restrictions. There are a lot of students coming from other universities. For instance, like last semester, this semester, I got students from Central University, Bihar. They come on campus. So the institute also provide them with the hostel facility. So they stay there. So it all depends upon the tuning between the incoming student and the professor within the department. And there are no restrictions, no rule as such in the books, which either stops the professor to recruit somebody from outside or to restrict anyone. So there are no rules. The only thing is the alignment between the incoming students as well as the faculty members. That's all. So it's better to write them an email, send your CV, show your enthusiasm in their research. You will get a positive reply. That's what I basically do. For instance, sorry, I will, Sonalika, I take this very, one small thing. For instance, it's a very funny thing. Normally faculty, I don't know about others, but to be honest, I don't get enough time, even for anything. So there was a student who was from the CS background from our own institute. So he dropped me an email 1 week before. I didn't reply. After 1 week, he sent a gentle reminder to the same thread. I haven't replied. And to be honest, yesterday he was standing outside my door and he said, "No, I want to work with you." So I cannot say no to him. You got my, that is the excitement we need in the students or anybody who wants to learn. So I think this is the question is answered. So.
Yes, no, we completely understand how busy researchers are with their research. And yes, sir, but definitely who's interested can reach out to the mentors there and they'll definitely reply you, I hope so. If they don't, you can keep on bugging them and as sir said, seeing your motivation to work, you will get a chance, right, sir?
Yes. So, I mean, there are a couple of more questions. Could you please let us
Yes,
get
sir.
this?
There are a lot of questions actually. I can see the curiosity. I'll just take one more question, sir, and I'm going to mail you the other questions because we are running late on time.
Yeah, please drop me an email and please tag the person so that they can get the answer as well. I can also, see guys, you can drop me an email. Chances are high I may not reply, but if I really like from your question, like you really need that answer, trust me, I will give you a very detailed response. So don't afraid, write to me. Yeah, please, your last question, please, Malika.
Yes, sir. So, Saloni is asking, in the liquid biopsy technique, we just get to know about whether a person has cancer or not or any other details as well, like what liquid biopsy is all about?
So, I mean, to be honest, liquid biopsy is very, at its initial stages. So there is a very big company supported by Illumina. I think everybody know about RNA sequencing. So everybody know about the giant Illumina. So Illumina basically provides you the platform to do RNA sequencing. There are other platforms as well, but Illumina is so-called the giant, the king in the field. Now, Illumina is backing a company called Grail. So you can read about it. So they are basically working on early cancer detection using blood, and their method is different. So the point is, the company has worth around 12 billion USD recent valuation, and they have not even come up commercially yet. In last year, they just bring their first product. From the last 8 years, they were working on the research. So just wanted to answer this question. Liquid biopsy is in a very early stage, especially if you ask about our technique about TEP scan. In TEP scan, you can normally, up to now, we have solved between whether the cancer is there or not. Even if, for example, a patient is there who has undergone chemotherapy, radiotherapy, and you don't know, the cancer always relapse, it comes back again. So at least now you have a test where you can donate, you can give your blood every 6 months or 3 months. You don't have to open up, the doctor don't have to open up your stitches and see whether the tumor is there or not. Or you don't need to do PET scan, for example, which is like 70,000 rupees, 40,000 rupees, 40 to 70,000 rupees in Delhi. And there you have to remove your clothes, you have to go naked into the machine. And so it's a lot of uncomfortness you will face. I mean, I think liquid, so we just able to solve up to now the presence and absence of cancer. And we are aggressively working on from where the signal is coming from in the platelets. So we are working on that. So it is, the research is going on. We are very far away.
Right. Okay, that was quite interesting. So thank you so much, sir, for such a wonderful session and so many insights about IIIT Delhi. And looking forward to meet you soon and getting in touch with you once again. Thank you, sir.
Thank you. Thank you, everybody. Adios.
So now moving ahead. So far, we learned about bioinformatics education and research training, emphasizing the role of bioinformatics in every research lab and the evolving role of bioinformatics in universities and research labs in India. Now that we know about all the opportunities around us, the question arises, how do we prepare ourselves for the research labs? How do we get the required training and enhance our skill set? Not only this, but also work on a research project and get a good hands-on before we actually hit the industry. To get all your answers, let's learn about OmicsLogic Research Fellowship Program. The Research Fellowship Program has been designed to help young researchers and students take advantage of the bioinformatics resources for analysis of complex life science data and become versed in bioinformatics. Research fellows participate in cutting-edge bioinformatics research led by expert mentors. The fellowship program will offer a combination of online resources and mentor guidance to prepare you and help you complete a bioinformatics project. The platform is a cloud-based analytical server, that is a T-Bioinfo server, which uses research labs and independent scientists around the world, the data generated by these labs. In the Omics Research Fellowship Program, the research fellows will get a chance to develop an independent research project, learn to utilize big data analysis tools, get guidance from experienced mentors, get an exposure certification and feedback, and get an access to OmicsLogic courses. Now, these courses are there on the portal. We'll be just taking you to the portal in some time, and we'll showcase you all the resources that are up there, which you can learn from and leverage the most. So these courseworks are on multi-omics, which includes genomics, transcriptomics, metagenomics, epigenomics, data science using R, Python specifically for bioinformatics and machine learning. And also you will find various OmicsLogic project examples to understand how the things that you learn in the coursework could be applied to answer real-life research problems or research questions maybe. And all this will be done through T-Bioinfo server, which is an AI-based web server for multi-omics big data analysis. So now talking about our community, we leverage publicly available data, online tools for big data analysis, and a network of mentors to help students learn bioinformatics, apply their skills to meaningful research projects, and work with mentors on turning their projects into publications or research posters. The programs we offer provide training, access to high quality tutorials and tools anyone can learn to use independently. The program is offered at university, high school, or community colleges levels, as well as directly to citizen scientists around the world. We have associated with industry and academic mentors with years of expertise in computational approaches for basic and translational research, biotechnology and pharma companies, as well as emerging domains like space omics and translational cancer bioinformatics, infectious diseases, vaccine research, computational neuroscience, AI-driven cheminformatics, and drug discovery. Now, to talk about the structure of the research fellowship program, the program is structured in such a way that the fellows are supposed to go through an extensive training process in the beginning of their fellowship period, where they learn about the key concepts of whichever specialization track they pick. It could be oncology, infectious diseases, neurosciences, space omics, agriculture, etc. They learn about various data extraction, analysis, visualizations, algorithms involved, methodologies followed. They practice it on the T-Bioinfo server, and finally propose a research question when they all are geared up with the knowledge and expertise in hand. Throughout this process of training and working on an independent research projects, the research fellows interact with the mentors and OmicsLogic community through online Zoom meetings and communication channels like Skype and Slack. They help us stay in touch with our fellows anytime they need assistance. And once the fellows have completed their project, then they can present their work to the community in the form of a poster presentation or a research publication, or even submit it in their institutions to fulfill the requirement of a minor, major, or dissertation project. So now let's look at some of our research fellows projects and also learn more about the outcomes of the research fellowship program by one of our mentors, whose name is Dr. Raghavendra. So over to you, Dr. Raghav. Dr. Raghav, we can't hear you. Okay, just a second, sir. Can you unmute yourself now? Ojasvi, can you make Dr. Raghav host? Let me, so try it now. I've changed the settings.
Yeah, can you hear me?
Yes, sir. Perfect. We can hear you.
Yeah, I think you can hear me. Thank you very much, Sonalika. And let's find ways to share my screen. Can you see my screen? I think you can see my screen.
Yes, sir. We can see your screen.
Thank you. It's still not responding to me. Can you see my screen? Yeah.
Yes, sir. We can see your screen.
Okay, thank you very much. I think I got it. Yeah. Thank you, Sonalika. And it's great to be here with everybody. Yeah. So, what happened? See my screen again?
So it was visible earlier, but I think it's not up here now. I think you need to reshare.
Yeah, I think I need to reshare, but it's not. Okay, let me check again. My computer. Let's try one more time. No, it's not working. Okay, I think I got it now. I think I got it. My Zoom just got frozen for some period. Sorry for the hiccup.
It's perfectly fine, sir. Yes.
Okay, thank you, Sonalika. And it's one more time, it's great to be here. So today I'm excited to share with you about how this research fellowship program works and how we have structured this research fellowship program. So the goal of this program is to provide training in bioinformatics to enable independent research, which is guided by mentors and with peers to develop or to develop a research project, including using big data bioinformatics analysis on your own, project on your own. So we actually enable students from all backgrounds, like from undergrad, from masters, and from PhD level, and even faculties, to develop a novel and independent research using the latest technology and the lessons that we have in Learn OmicsLogic platform. So, yeah, so this program is actually structured that so that the students get guided through various areas of bioinformatics data analysis using practical examples. So during this program, we go through several high-quality publications, and to learn about the applications of these methods in projects that these publications describe. So this kind of hands-on experience will allow beginners to try computational biology techniques on public domain data, and it will also make everybody to work with large files and extract meaningful information from, let's say, data that includes patient samples, data that includes animal models and cell line models, microbiome data, and etc., etc. So this program actually provides support, mentorship, and extensive training to involve or to implement all the skills that you have learned from the Learn OmicsLogic platform to the proposed research ideas and the topic on your own. So at the key basis of this program is to develop a project or to develop a title of your project. And the project development for this research program involves these 7 steps. So for every project, we follow these well-defined steps. So you start by framing the question, and which is followed by extensive literature review. So once you have identified a topic of interest, then we collect the data from publicly available resources that will validate your hypothesis, or that will help you design a more interesting question and explore for possible patterns. And from these exploratory analysis, from these hypotheses, we can actually decide on the type of in-depth analysis to be performed for the data. So after performing these analysis, we can then interpret the results in the biological connotation or the biological relevance for the samples, and by connecting the big data omics, and by connecting the patterns that we find from this omics data to the phenotypical characteristic of the biological sample. So in the end of it all, after completing your project, then you are encouraged and you will be required to submit your research projects and communicate your results to the OmicsLogic platform, and you will be as a part of students project. So let's take a couple of steps from this project development as an example, and discuss in little bit detail how we are actually trying to frame a question that involves or that helps you to start your project effectively. So at the basis of all, we have to start from a topic that you care about, or start from a topic that interests you the most. And you can actually start with a topic that you have some prior knowledge about, and then slowly expand to include topics that could be of interest not only to you, but to general public or to general audience, to generalize your interest in these topics. And then you can proceed to design or proceed to develop more refined hypothesis or exploratory questions for your specific project. And we also have to find, once you have a set of questions that you want to explore, then we have to find the availability or the suitability of the data that exists in public repository that can help you answer your question and identify a perfect title or a specific aim for your research project. So yeah, so as you explore the topic and make note of hypotheses and questions and the data sets reference, many questions will come, or many questions in will come to your mind, and you have to pick and choose that is going to fill the gap in the literature. So when you find something really interesting, you have to find the suitability of that interesting thought to be translated into formal research statement or formal research problem, and that will form your topic. So obviously, collecting raw data or primary data for your research and finding these sources are very important. So once you have once you have your hands on some of the data that is available, you can perform some exploratory analysis and try to find if the data has some pattern that will eventually answer your question. And if that does not, then you go back to the step again, refine your question again, and try to include more data, or try to be inclusive of expand your research question to include more data and more types of data, and then thus you can refine your hypothesis as such. So so of course we have a course designing a bioinformatics research project, which details on how to proceed with all the steps discussed above, and it also goes in detail about how to start with the literature review and go on about each and every steps of those 7 steps to research project that I discussed about. So you are encouraged to check this course, which will be extremely valuable for budding students like you. And let's wait for my PowerPoint to respond again. Is it? Yes. So let's take a few examples of student submitted research projects on a variety of topics that we have already. It's again not responding. Let me, for a short while, switch off my video. Maybe it's eating up a lot of bandwidth. I'm not able to stop my video also. If I'm still audible, I would pass it on to Dr. Mohit and come back to a couple of slides where I'm going to discuss some of the students' project that is submitted to our portal. It looks like my computer is not responding for some reasons.
Thank you, Dr. Raghav. Yeah, I think we are running out of time. So thank you for your presentation and describing the structure of the program. And maybe now also we have the student presentations coming up. So I'll request Persh to also reach out to those students who are already here and get them ready. Meanwhile, I'll just share a little bit about the research fellowship program that we have been discussing in this session. I hope that my screen is visible to you.
Yes, sir, your screen is visible. Now it's not.
Just a second. Yeah, it was was not playing, so I think it's fine now. So as you learned about the OmicsLogic Research Fellowship, similarly, the collaboration, what we are trying to do is that the collaboration between industry and academia is the key. That has been being discussed in the symposium, and we were going to learn from a lot of speakers who are coming from industry as well as from academia. So we believe that this collaboration between industry and academia is the key to catalyzing innovation and growth. So industry partnership is instrumental in advancing research and in the creation of skilled force work. So this is something that we are looking at as a community, as a company also, to address this huge gap between data and the information that we have been talking about. I wanted to share how our OmicsLogic community is growing with young researchers who are contributing by working with experts and generating these amazing resources and projects and outcomes as a part of this experience. So I was like really very excited also. I just wanted to include that in this presentation, that I was really excited to check the results from the symposium registrations form submission. So the results suggested that 30% of all of you, the participants, would like to learn about genomics and its application. 20% of all the participants said that they're interested in transcriptomics. And then 21% of all the students said that they're interested in data science and machine learning. And then there was an interest in epigenomics, which is 13%, and then metagenomics 10%, and structural biology 8%. So I'm really happy because many of you are inquisitive to learn more about these new technologies that we have been discussing, like precision medicine, precision agriculture. So as many of students and faculty are already are part of this OmicsLogic research community, in my presentation, I will cover that how university research program, what is this research university program, research fellowship university program, and how it is structured. So just to quickly tell you a little bit about it. So what we did is like a collaboration with several leaders in this area, where they have an experience of over 30-40 years in education, discussing about this about the structure of how we can help students and enable faculty and create a pipeline to make the skilled workforce do translational research, to do research which includes these resources that we have created for the students to utilize. So as I was saying, that these problems that we are discussing in our research fellowship program and these research areas that we are discussing is just not related to bioinformatics, but application of bioinformatics in different fields. So we have collaborators in the US, Africa, and India, and other places. So they are working on medical biotechnology, plant and environmental biotechnology, biochemical engineering, cellular and molecular data generation, and computational biology and bioinformatics. So this enriched community where experts are from almost all the fields of biology and expertise of data science and machine learning, if we incorporate that and with the help of industry, I think you can envision a complete pipeline for students to learn all of this. So this is the pipeline that we created for a student to go through a very basics, like basic training, to utilizing some of the advanced tools that we've been discussing, like AI and machine learning and all of that. And then how this is used in terms of application, like how this skill sets, how these tools, how these algorithms can be used to crunch biological data and to gain insights which are really valuable for anyone to know. So that value obviously is going to help you to get it maybe published or get grants and all of those things that you learned from a previous speaker. So this is the pathway that we are focused on, where we have incorporated different elements that we have prepared and created and put together on OmicsLogic Learn and T-Bioinfo Server. So as I was telling you, that the first step is to learn about the basics. Start with the OmicsLogic basic curriculum, and which covers the basics of NGS and also these important technologies where we are focused on analysis, downstream analysis, and also understanding that how can we replicate this while using coding or a non-coding environment. So after that, we learn about the projects that are available, which is to learn about the application. So you have to understand that how can an actual problem is conceptualized. So many students learn about these different techniques, but when it comes to like applying this technique, there is a huge gap also. So many students, when they go to a research lab and they write it in their CV that they know genomics, and they know these tools and that tools, and that you will see many times that we get in the CVs. But what recruiters are more interested is in the application, not only just the tools, because in the end you have to have that thinking about how to solve problems. That requires a lot of experience working on different types of project examples, data sets. So this is what we are doing in step 2. Then we have these all different areas where student now goes and chooses which area is what interests the most, and develop research ideas and research projects. And in this intermediate step, we have training programs such as infectious diseases, precision oncology, precision medicine, which is very much specific about these application of these technologies, which we run throughout the year. It includes also the upcoming areas such as space omics, and then understanding the three-dimensional data, which is what we have seen in the last presentation, that how these different areas of bioinformatics research are combined together, are interconnected to help in the process of finding a solution such as drug discovery, and helping for human healthcare. So these internship tracks are divided. So we have research fellowships where we help students and faculty to collaborate and develop these projects. And then once the student is capable and know how to do this analysis, they are more they get opportunities such as industry internships, where they are getting paid for knowing and doing these analysis for specific programs, for specific groups, and specific research labs. So similarly, like the step involves integration of the faculty, which is very critical at this point of time, because as a university ecosystem where we are trying to enable students to work on research projects that are defined by faculty who are interested in incorporating these bioinformatics tools, these bioinformatics research, utilization of the big data, utilization of the public repositories into their research, so that they can enhance their research, so that these research have high value and good outcomes. So our team, we work with the faculty to help them develop these projects further, and then the students to work on these projects. Similarly, we have an initiative called Omics. It's a free student club where we invite the faculty and also help the local community, or also help the international community, online community, to learn about these specific areas of research, and to learn about how research is done through in different research labs via these researchers, faculties, and what are the different areas that they can contribute it, similar to the session that we that we had. So all of this involves research consultation, where we are talking about these different areas of research, working on these different areas of research, utilizing public repositories, utilizing our online tools, utilizing the tBioinfo server, and coming up with projects and outcomes for students, helping them to get scholarship, helping them to do this on their own, supporting them throughout their period of internship, and then that leads to this amazing results when these students from undergraduate and postgraduate degrees come up with a really noble project. Not only that, they get a profile and certification to showcase to the world, and also the experience to work on a real research project with real data sets. So we have several of these students who were able to complete this and will be able to present their results in the upcoming session. So without wasting much time, I'd like to pass the session to Sonalika to introduce the judges of the session of the poster presentation, where we will have the opportunity to learn from these students who have completed their project, completed entire analysis on their own, and have shared their experience as well. Thank you so much.
Thank you so much, Dr. Mohit, for shedding some more light on the Research Fellows projects and progress. So it's time for the poster presentation competition. And I just quickly take you all through the judges for the poster presentation competition. We have with us Dr. Vivek Das. He's a senior research scientist at Novo Nordisk. Then we have with us Dr. Verena Raz. She's a training coordinator at H3A Bionet South Africa. And Julia Panov, Dr. Julia Panov, she's an associate director at TBRC. Dr. Priyanka Narath, she's an associate professor at Amity University, Noida. And finally, we have with us Taco Midden. He's a data scientist, ML and AI specifically in the EDNAS group in Japan. So it's time we begin with our poster presentations now. And the first person who will be presenting his work is Andrew Lee. Let me make sure that Andrew is a co-host.
Yes, he is.
Okay, perfect. He is. Andrew, can you share your screen? Yes, we can see you. We can see your screen as well.
Can you see my screen?
Yes, we can see your screen, and the time given to you is 5 minutes. Okay, you may begin. Thank you.
Hello, I'm Andrew Lee, and my project is a cell type specific expression and protein interaction approach to identify autism-rich genes. I'm an OmicsLogic fellow and a data science intern at Columbia University. Firstly, there is a high prevalence of autism, as 1 in 44 children are afflicted by the disorder. The clinical diagnosis of autism has been limited by the lack of conclusive biomarkers and the high genetic and clinical heterogeneity in regards to autism's etiology and hallmark symptoms. Early diagnosis of autism is crucial, as the intervention loses its effectiveness early in the adolescent stages of development. And there are 2 main biological reasons for this. The first is that neuronal plasticity declines, and secondly, the opportunity for complex neuron growth is lost. There are 3 cases for autism diagnosis. The first is autism confirmed cases, which would then follow a behavioral and developmental monitoring approach. There are also negative autism cases, and there are a variety of other neurological tests to test for other conditions. However, for autism speculation, this is highly dependent on genetic testing through the discovery of new autism-rich genes. The Simon Safari database classifies known autism genes based on their strength of association into gene categories of syndromic, high confidence, strong candidate, and suggestive evidence with gene scores of 1, 2, and 3. However, there is a growing lack of consensus, as only about 100 high confidence genes have been found out of the estimated 1,000. In the recent years, there have been 2 main advancements in autism genetic understanding. The first is that autism genes have high expression specificity, as 88% of the known genes have been found in the early tissues. The second major finding is that autism genes have conservation in disease-implicated pathways, and genes that confer risk for autism are functionally related. There are 3 main biological pathways for this. The first is neurotransmitter transport, seen in figure 2A, synapse regulation, seen in figure 2B, and 5 classes of autism dysregulated receptors, seen in figure 2C. This brings me to my first purpose, which was to visualize single-cell RNA-seq autism candidate gene expression across 26 cell types. But first, I had to test the accuracy of the analysis in separating known autism genes from the non-associated genes. My second purpose was to identify novel autism-rich genes through expression profiling. And lastly, to identify biological support for the candidate genes through kinase and transcription factors for known autism associations, and also correlating these genes back to the 3 major biological processes I had explained in figure 2. For my methods, I obtained a single-cell RNA-seq dataset of the human midbrain, a training set of 91 high-confidence genes from the Safari database, and a testing set of likely gene-disrupting variants. I then developed a cell type specific gene expression analysis and confirmed the accurate stratification of the autism and non-associated genes through 2 main tests. The first was to calculate the average expression of all 19,532 genes in the 26 cell types and compare this average to the Safari autism high confidence genes. I also compared the hierarchical clustering separation for autism genes of varying association confidence levels. I then identified new genes through a gene expression analysis. All the cell types I used in this study have been correlated to autism, and I did a literature review to find how these cell types have been linked to the autism symptoms. For my first results, I found that more strong candidate genes with gene score 2 were hierarchically clustered with the Safari high confidence genes compared to the same set of high confidence genes with suggestive evidence genes at gene score 3. And this means that increased autism association is correlated with increased expression similarity between the Safari high confidence genes and the testing set. And this is especially seen in the low expression and high expression regions. For this second test, I also compared the Safari high confidence gene average and found that they have higher expression levels, 2 to 3-fold, compared to the average of all genes, especially in the dopaminergic and the GABA neurons, which have been highly correlated to autism in the past. And this supports that expression profiling can differentiate between autism and non-associated gene sets. I first found differentiated clustering of known Safari genes from the likely gene disrupting testing variants on the principal component analysis. And also on the heat map, we can see that the autism genes have high expression levels and similar expression profiling. However, 6 genes labeled in blue on the heat map, ARH GAP21, SLIT1, ATP6V1D, APPBP2, SNAP91, and SMAD5 have been identified as potential new autism candidate-rich genes. For my second method, I compared the autism RNA-seq expression levels using expression t-distributed stochastic neighbor embedding and ridge plots. But first, I compared a Safari high confidence gene, a non-associated gene, and a candidate gene. First, CHD7, a high confidence gene, and SLIT1, one of the candidate genes I identified in this study. You can see that these genes share high expression levels and similar expression profiling. However, SDF4, a non-associated gene, has little to no significant expression. I did this process for all 6 candidate genes and found that all 6 candidate genes had high expression levels in greater than 10 cell types, especially ARH GAP21, SLIT1, and ATP6V1D, consistently 10-fold higher expression compared to the average. A similar trend was also seen in the Tisney plot, as it showed that the candidate genes were widely expressed in multiple cell types, as evident with the multiple red regions. For my third method, I wanted to provide more biological support for the candidate genes simply beyond gene expression. I did this through identifying new protein-protein interactions, transcription factors, and protein kinase associations for the candidate genes. I found that the candidate genes have been correlated with past autism-associated protein interactions, especially the MAPK1 and MAPK14 protein kinases, which have been linked to synaptic dysregulation in autism in the past. And there were 5 additional associations for these candidate genes. I then performed a literature search on all these 6 candidate genes, and you can see that out of these 6 candidate genes, 2 of them had little known past correlation. So these could be identified as new candidate genes. The other 4 have little evidence to suggest that they have association to autism. So there's more studies that need to be conducted on these genes. And linking them back to the pathways correlated with autism, you can see that 4 out of the 6 genes have been correlated back to these 3 major biological processes. For my conclusions, I produced a new alternative approach for identifying autism-rich genes. I identified 6 potential genes to use in genetic testing. My study also supported previous research, as it identified the midbrain as an autism-rich gene hotspot, and it also showed that autism genes have high expression levels during the early developmental stages. And with increased discovery of autism-rich genes, more individuals with autism can be diagnosed earlier and with greater accuracy. My future research is to use molecular RNA-seq inversion to analyze mutation inheritance and genome-wide association studies to analyze the mutation prevalence in autism cohorts. These are my references, and thank you.
Thank you so much, Andrea. And next we have with us Risi. Risi, over to you. Yes, we can see your screen. You may begin.
Thank you so much for the introduction. My name is Risi. I'm currently an MSc student of Genomics and Precision Medicine, and I'm interested in the clinical genomics of rare diseases. I'd like to take this opportunity to introduce you to my work, which involves an assessment of traditional and genomic screening in newborns and the applicability for Africa. Let's move on. Did you know that it is expected that in the next few years, each baby born will have their genome sequenced at birth? And as Hippocrates once said, it is more important to know what sort of person has a disease than what sort of disease a person has. And that forms the basis of newborn screening, which has been shown to improve the overall quality of life of neonates. This includes, this is through detecting congenital conditions that occur either in early childhood or manifest during the late adult stage of the child. So these include hemoglobinopathies, metabolic disorders, some rare diseases, and some assortments. So the purpose of this particular survey was to document the currently existing efforts in the use of traditional and genomic newborn screening with a special focus in Africa. When it comes to my methodology, as you can see from the decision tree on the right, my filtering criteria is actually highlighted here, whereby, for instance, the publication date was set to a maximum of 10 years. What this means is that if a study was important but it was published in 1986, it would automatically be exempted from this particular review. When it comes to my findings, we would say that Africa is still at the initial stages of implementing traditional NBS, largely as a result of limited access to resources for GNBS. However, based on literature, we can see that it is important for Africa to move from the traditional methods of screening to the newborn genetic screening, whereby when it comes to cost, you find that the cost of buying a traditional sequencer goes from $400,000 to $600,000. However, when it comes to the cost of buying a next generation sequencer, we're looking at a price range of about $160,000 to about $165,000. So the initial cost of establishment of the traditional methods of screening is just way too costly. And then also when you compare the maintenance cost, it is way cheaper to maintain the NGS as compared to the traditional one. And when it comes to the traditional methods such as tandem mass spectrophotometry, it takes a lot of time to develop this expertise, unlike in NGS. Then my second finding was that sickle cell disease is the most screened for disorder in sub-Saharan Africa, which doesn't really come as a surprise, given that this part of the continent bears over 70% of the burden of the disorder. And it is estimated at the moment that over 300,000 babies are being born annually with this condition. So when it comes to genomic newborn screening, most developed countries have already set aside funding to conduct research on the potential clinical utility of genomic NBS. However, as at Africa, nothing is currently going on. So you see, these people, they are strategizing. They're just not expecting to go there. Then when it comes to the challenge that is currently arising is that there's a great discordance on the selection criteria for the gene disease inclusion in currently existing panels. So each panel has different set of diseases, therefore you can't really compare them. And when it comes to Africa, a few efforts have been made where a few countries like South Africa and Ghana have sequenced singular disorders. Then our findings do reveal the unpreparedness of both resource and under-resource settings to implement GNBS. When it comes to the future of this particular project, I'm currently involved in ongoing work, whereby we have developed a gene panel based on this, on this survey, and this panel has a range of conditions and their corresponding variants, and a subset of these conditions actually are African specific. And I'm currently also working in collaboration with bioinformaticians and computer scientists to try and validate the panel. And we're also exploring some algorithms from the project. So thank you for having me, and I look forward to hearing from you.
Thank you so much, Tracy. That was a nice presentation. Next, I would like to invite Bridan Ganam. Sorry if I'm pronouncing her name wrong. Yeah, over to you, Bridan.
Hello, everyone. Is that my screen is visible?
Yes, it is visible.
Yeah, we can see it.
Hello, everyone. I'm Brinda, from a research scholar from Department of Bioinformatics, Pondicherry University. So I'm going to present my work on microbiome of 4 different lung diseases, a comparative 16S rRNA metagenomics to understand the diversity and functional profile. So in this study, we collected 4 different lung diseases from NCBI, such as pneumonia-associated HIV infection, lung cancer, saccharoidosis, and COPD. So these raw data were subjected to taxonomy assignment, followed by diversity, statistical, and functional analysis. The aim of the study is that first, to compare the microbiome of different lung disease sample with healthy sample, and to show the effect of cigarette smoking in male and female patients by evaluating the abundance and diversity across the sample. Second is that finding the possible biomarker to treat or to monitor the disease severity, and also to treat the co-infections. And finally, profiling the functional properties associated with the diseases. So here in material methodology section, we can see clearly what is the overall workflow. So we were having some set of criteria to collect the sample. First, the sample with lung diseases, and it should be collected from the respiratory tract, and then sample with the known gender and smoking history, and it should be 16S metagenomics and padded. Then we were having set of two different set of pre-processing. One from Silva NGS, which is having inbuilt quality checker, Silva incremental aligner. Another one we used Galaxy, which is having inbuilt FastQC and Trimmomatic. After pre-processing, we went for dereplication. In the dereplicated sequences were subjected to reference database mapping alignment, which is called that Silva database. So after blast against this database, that sequence with more than 93%, sequences were classified and clustered together, and the OTU was assigned. After assigning the OTU for each sample from different data set, the tables were consolidated and normalized using TSS method. That normalized consolidated table was subjected to diversity, statistical, and functional analysis. So here in data pre-processing, we can see what are the samples I collected and how many samples were there. And with respect to male and female and smoking habit, how many samples are there and what are the control samples, all the informations are given in here, and how much the quality. So almost all the reads were having more than 20 Fred score. And here in rarefaction plot also we can see what is the sequencing depth, and then how many OTUs are there in each bio project. Then I'm moving to the result section. So here I started with diseased lung microbiome diversity and abundance to check the microbiome diversity. I used PASS4 tool. This is specific to ecological parameter analysis tool. Here it is having the set of parameter to find which microbiome it is most diverse. So compared to all, by seeing the parameters, we come to know lung cancer is the most diversified among all. Then we went for a relative abundance and taxonomy analysis. So based on the taxonomy analysis, we come to know from phyla to family level, actinobacteria, proteobacteria, firmicutes, and then bacteroides were the most abundant phyla. Then at genus level, specifically we went for Venn diagram representation. Why? Because what are the genus were common to all the microbiome, and what are the genus were specific to each microbiome, that we want to know. So after doing this analysis, we come to like 211 genus were common to all the microbiome, and which was having SK pathogen as well. SK pathogens are the nosocomial pathogen, which are common in all the microbiome. Then we compare this particular SK pathogen against the control. Then we found like some of the SK pathogens were specific to all the microbiome and which were abundantly seen in each microbiome. Then after studying the pathogen, then we were checking whether the probiotic also any changes there or not in all the microbiome we checked. Then we come to know like compared to control sample, in all the diseased one, the probiotics such as bifidobacterium, lactobacillus count was very less. Then we went for network analysis also. We use past correlation of compositional data network analysis. In this, what we wanted to look for, like what are the hub genes, what are the hub pathogens or hub species. Here in ecological term, they are telling this hub pathogen as a keystone pathogen. So we using this particular compositional data package, we come to know like Haemophilus was the most common hub pathogen in all. Then along with that, Staphylococcus, Acinetobacter, and then Klebsiella also the hub pathogen. Then through NMDS plot, we come to know except COPD samples, remaining all the microbiome were showing significant difference between smoker and non-smoker. Then finally, we went for functional analysis. For this, I used Megan, the Ultimate Edition standalone pipeline. In this analysis, I come to know like transporter, specifically membrane transporters are abundantly seen in all the microbiome, and the two, while comparing to the control sample, it was 10 to 15 fold higher in all the diseased microbiome. Then we also went for CARD database analysis. CARD is a comprehensive antibiotic resistant gene prediction database. In this also, we found like tetracycline resistant genes were abundantly seen in enriched in all the lung diseases compared to control sample. Then finally, we went for pathway analysis, which is also inbuilt in Megan tool only. So in CAGE pathway analysis also, we come to know beta-lactam resistant pathway was enriched in all the lung diseases. So while analyzing that pathway, we will come to know like beta-lactam resistance is mainly due to RND transporter. RND transporters are the transporter present in the bacterial membrane. The main mechanism behind RND transporter is that it will efflux the antimicrobial agent from inside the bacterial cell to outside the cell. So targeting that membrane transporter, specifically in this escape pathogen, and also along with that respiratory pathogen, which is Haemophilus, will be the better way to reduce the severity and also to reduce the co-infection. Along with that, supplementing with the probiotic, which is less in all the lung diseases, are also one of the way to reduce the severity. So these are the findings we come to know in this analysis. Thank you, and these are the references.
Thank you so much, Brenda. Quite interesting it was. And the next participant is Nabita. I'm just going like so quick because we are short on time, and also we have to maintain a time slot. So after we are done with the presentations, then we listen to the judges for an overall review. Okay. So the next presenter is Nambita Sahi. Nambita, are you here with us?
Yes, I'm unable. Okay, I can screen share now. Hello, my name is Nambita, and I have done a project on developing novel drug candidates for MDD, MMD, and hypertension, and I am part high schooler at Crystal Springs Upland School. So the second messenger cGMP is extremely important target for disorders such as heart failure, atherosclerosis, and cardiac hypertrophy, disorders affecting millions. The SGC protein is a site that converts GTP to cGMP, a small molecule controlling vasodilation. Currently, very few to no therapeutics target the SGC cGMP pathway due to many unknowns about the protein structure and qualities. It has a role in rare diseases like MMD, and it is extremely important to target cGMP to find treatments. cGMP can not only treat cardiovascular disorders causing up to 31% of deaths worldwide, but also have a role in cancer regulation, major depressive disorder, and more. Understanding the SGC cGMP pathway with evolutionary tools is the utmost important to target these deadly illnesses. So this study seeks to first categorize a protein through models created from databases and deep learning software. Then multiple protein sequence alignment is used to determine that the HNOX domain of the protein has a high concentration of conserved residues, the parts of the proteins that stay constant across evolutionary lineages and are therefore most important for protein function. A blast genetic sequence analysis is done to compare SGC to proteins such as hemoglobin, and it is determined that the SGC is a druggable target. Finally, a virtual screening of thousands of ligands are done in Autodoc Bina from a small molecule library. After reviewing SGC structure, it is visible that it is likely a membrane protein due to the helix. Many conserved residues are concentrated in the HNOX domain, so it is confirmed that the domain is functionally important for SGC, and it contains a heme site, which is important for signal transduction. The only issue is if hemoglobin and SGC have similar structures as small molecules may harm hemoglobin, a protein vital for carrying oxygen and carbon dioxide. By superimposing the two structures with a multi-aligned viewer, it is found that the heme sites occur in different locations, and in targeting it would not be damaging. For the genetic analysis, RNF213 gene co-expression, HLZ2 has a Spearman's correlation of 0.769. It is a co-actuator for the peroxisome proliferated activated receptor alpha. And OAS3 regulates the expression of chemokines and interferon responsive genes. Similarly, for the gene GUA1B1, MAP9 has a Spearman's correlation of 0.746 and is required for spindle function. So after the genetic analysis and structural analysis are done, binding affinities are collected through docking, molecular docking. So the 5 ligands with the topmost armistine binding values have their pharmacokinetic properties analyzed, and their chemical moieties were analyzed in PyMol to reinforce if they were good candidates. By gathering this important structural information, drug candidates are proposed after conducting a virtual screening of thousands of ligands. Drug candidates, namely C15H24N2O2, C9H11NO, and C16H20N404, were identified. The drug targets identified can move on to lab testing on animal models to be developed further into medications that are lack of FDA-approved treatments. Finally, a combination of invasive treatment and medication can be used to reduce the chance of stroke or high blood pressure. A soluble guanylyl cyclase contributes to a variety of conditions from hypertension and MMD to even cancer. Further research in drugs targeting SGC would be imperative. So here are my references, and thank you so much.
Thank you so much, Nandita. That was real quick, and thank you. Now the next presenter is Zainab. Zainab, are you here with us? I can't. Yeah, you are here. Okay.
Hello?
Yes,
can you hear me?
We can hear you. Yes.
Okay. I shall share my screen. So can you see my screen now?
Yes, we can see your screen.
Okay. Okay, hello everyone. This is Zainab Al-Sheikh, a teaching and research assistant at Zuwairi University of Science and Technology. I'd like to present my bachelor thesis project titled Predictive Analysis of SNP Disease Association for Alzheimer's Disease. I'm sorry, I have this issue. I'm sorry, for just a minute. For some reason, it keeps scrolling. Okay. Yes, okay, pardon me. So Alzheimer's disease is a slow progression of memory failure and language disturbance, poor judgment, and ultimately death. According to World Health Organization's report published in September 2021, 55 million people have dementia worldwide, and it's predicted to reach 78 million in 23 and 139 million in 25. So in 2050, I'm sorry. The pharmacologic current AD treatments do not necessarily slow or stop the damage and destruction of neurons that cause Alzheimer's symptoms and make the disease fatal. And that's why future medicine's focus shifts from treating existing diseases, typically late in their progression, to preventing diseases before it sets in. The elevated risk of AD, along with the potential AD preventable strategies that are recently invented, create a crucial need to discover and validate prognostic biomarkers that may identify subjects at great risk for accelerating and testing preventable strategies. So, although, I'm sorry, again, it happens. I don't know why. Okay, that's it, my aims. Okay, although genome-wide association studies have been successfully employed to interrogate the genetic architecture of common and complex diseases, they are limited for being based on single locus analysis, and they fail to detect the epistasis interactions between different loci and their effect on the appearance of the phenotype of interest. So our main aim is to evaluate the impact of different selection techniques for selecting significant single nucleotide polymorphism associated with diseases. We assess two methods to choose a subset of SNPs for better and more accurate prediction. The first method is the p-value SNP selection technique, which is based on the statistical significance of SNPs. As a second approach that we actually are assessing depends on interaction selection technique, which actually depends on the ability of machine learning based feature selection technique to capture the cumulative effect of small effect size SNPs, which is valuable in complex diseases. By assessing the best model, we will reach best machine learning model, we will reach a better predictive accuracy and hence revealing the genetic basis of Alzheimer's disease. Okay. So this is an overview of our proposed framework. First, our data set was obtained from National Alzheimer's Coordinating Center. It consists of 364 unique samples. Each sample contains almost 380,000 SNPs. The framework applies a pre-processing steps according to specific quality control steps to ensure the data meets certain criteria, such as missing rate test and imputation, minor allele frequency test. Then we do SNP encoding to represent SNPs as numeric values that can be read by machine learning models. We divided the SNPs into 4 batches according to their p-value. Evaluating the p-value SNP selection technique, the only SNPs that should be used as features for our machine learning models are the ones in batch 0. That includes SNPs whose p-value are less than 10 to the power of -5, while other batches were used in testing the second selection technique. In assessing the interaction technique, we consider a higher number of SNPs to capture their cumulative effect. However, we have to keep the number of SNPs comparable with the sample size to assure that the training model will not overfit the data. Hence, working with a finite number of data samples in a high dimensional feature space, we need to eliminate some features using feature selection method before using another model to fit the data. Here we try to propose machine learning-based feature selection techniques that relies on different consideration than the phenotype association. We search for a feature selection technique that uses the least number of features to avoid overfitting and achieve the higher accuracy as well. The feature selection techniques we tried are mutual information and linear SVC with L1 penalty. We used support vector machine as a linear machine learning technique, and we assess the accuracy achieved by different kernels. And then we compare them. The kernels use a linear kernel and RBF kernel. Okay, so as illustrated in the left graph, the best accuracy was achieved by batch 2 in both linear kernel and RBF kernel. This batch 2 contains 441 SNPs. All of them has p-value less than 10 to the power of -3. Batch 2 achieves higher accuracy than batch 0 and batch 1 using both linear and RBF kernel. The second approach is that combined both p-value technique and machine learning approaches achieve even better predictive capacity using both kernels. So we mainly depend on mutual information as a machine learning-based selection technique. The best accuracy achieved, we can see on the right graph, is accuracy when we choose 300 SNPs. So out of the 441 SNPs in the batch 2, we used this batch in mutual information after applying mutual information, so and reach 93.5% accuracy. So we can notice that the higher the number of features we set, the better the achieved accuracy, which supports the first hypothesis of interaction. However, the model accuracy starts to decrease at some point, which may be due to some false positive SNPs that were taken into consideration in the model. Okay, we tried to, we investigated the Alzheimer's disease correlation with those 300 SNPs that were computationally used in decision-making to find out if they are experimentally supported. Using Nomad database, we found that 180 SNPs of them are located in 152 genes. By looking them up in literature, we found out that out of the 152 genes, there are 104 unique genes related to Alzheimer's disease or other related diseases, as well as 11 Alzheimer's disease correlations with the SNPs themselves. Pathway enrichment analysis revealed that there are 146 pathways associated with these 300 SNPs, with 26 correlated with neural system. We tried to do network analysis using cytoscape. We graphed the SNPs along with the affected genes. Nodes represent both the SNPs and genes in a network. An edge between any SNP and gene indicates the biological association. Then for focused analysis, the nodes with more than 3 edges are filtered, which means the genes affected by more than 3 SNPs. The remarkable genes in the network are shown here. Those 3, by looking up them in literature, we found that they are experimentally verified to be related to Alzheimer's disease. So to conclude, the application of genome-wide association studies, I'm sorry.
Yes,
okay.
Anna, please conclude.
Yes, the application of genome-wide association study in case control setup have resulted in plethora of significant genetic variation associated with complex diseases phenotypes. Integrating machine learning-based feature selection technique outperformed the p-value selection technique that is traditionally was used. So finally, our recommendation are to increase the sample size under examination in terms of having more patients and to apply the same pipeline to samples taken from Egyptian cohort. So with Alzheimer's disease, because the SNP print differ with different populations. So we are trying to enable the creation of personalized health report service, which will enable Egyptians to obtain detailed information and reports based on their genome and to take appropriate action, which might be applicable. Okay, so these are my references. I don't know what it's not working. Yes, and thank you.
Thank you so much, Anna. Next, we have with us Karam B for his presentation. Karam B, can you? Yes, we can see you have started sharing your screen. All right, we can see your screen, but we can't hear you. I think you need to unmute yourself. Yeah, thank you.
Hello everyone, this is Karamveer, a research scholar from Department of Bioinformatics, Pondicherry University. So today I am going to present my work on oncogenic and non-oncogenic class prediction from human papillomavirus genomics data and using machine learning. So human papillomavirus is a double-stranded DNA virus which belongs to Papillomaviridae family, and till now more than 200 types of HPV have been discovered. So based on the genotype of HPV, it may cause carcinomas, or it may cause warts also, and sometimes it may be asymptomatic also. So it has been reported that some of the this HPV is involved in different type of cancer like penile cancer, head and neck cancer, and anogenital cancers. However, HPV and cervical cancer relationship is well studied and it is well established. Even in 95% of the cervical cancer cases, you will find HPV is responsible for that. Then nowadays, as advancement in sequencing technology is happening, so it is adding more HPV genotype every year to the databases, and therefore it is necessary to predict based on the genomic information whether we can predict the type of HPV, its phenotype. So in past few years, people are using genomic composition and codon usage data with machine learning to predict, to find the pattern in biological data. So here we tried to use RSCU information with machine learning models. So RSCU is a relative synonymous codon usage. So it is used to find the biased codon present in our sequence. So we have used RSCU table we constructed for each gene of HPV. So HPV is having around 8 to 10 genes in different variants, but mostly in mammalian lineage you will find 6 core genes which are E1 and E2, mostly involved in viral replication and transcription, and L1 and L2, these 2 genes are coding for the code protein, and these 2 genes are oncoprotein. They used to cause the cancer by regulating p53 and retinoblastoma. So we have constructed RSU table from these 6 genes only. So how we constructed the feature table is we have taken these 6 genes CDS sequence, we have constructed RSU table, and then we have generated feature based on the vowel codon position. So if RSCU value is 1.6, we are considering that codons only biased codon. So in biased codon, if the third nucleotide is whatever it is, based on that only. If it is adenine, So we will sum up all codons RSCU value. So if these are the codons which are having Wobble nucleotide, whatever the nucleotide is there. So for each nucleotide at Wobble position 8 EGC, we constructed 4 features for one gene. So in this way, we were having 6 core genes. So we were having total 24 feature for this. 210 HPV we have taken total. So with that feature table, we went for machine learning, building machine learning model that was able to classify the class of HPV, whether it belongs to, it can cause the cancer or not. So the total number of sample we were having were in training data set, which were 18 carcinogenic and 129 non-carcinogenic. As we can see, it is an imbalanced data set. So we did oversampling using SMOT, and we synthesized this minor class, and we created a balanced data set, and we were having test data set with 6 carcinogenic and 57 non-carcinogenic HPVs. So we compared different linear and non-linear models where it can perform best. So we have used logistic regression, linear discriminant analysis, KNN, and naive and SVM. And as you can see here, it is above 98% in logistic regression and SVM. It is on training dataset and it is on testing dataset. So in both these two algorithms were performing nice. So we went for hypertuning this SVM model, and we find out that linear kernel with scaled gamma was performing good on our test dataset, and we got 98.4% on training dataset and 98% on test dataset. The MCC value for test dataset was 91%, and specificity and sensitivity of the model was 100% and 98% respectively. The balanced accuracy, as our data set is imbalanced, so the balanced accuracy was 99%. So you can see the final result here. So out of 6 carcinogenic, 6 of them we predicted correctly, and out of 57 non-carcinogenic, 56 were predicted correctly. So what we concluded here is we have constructed the ROC curve, and we found out like the 100%, we got the ROC value of 1, which means 100% classification. And what we concluded is linear models such as SVM.
You can proceed, Karanvi. Participants are requested to please keep themselves on mute. Yes.
Okay. So from here we concluded like linear models such as SVM with linear kernel or logistic regression, which are simple linear model, can be used to HPV class prediction, and genomic composition information such as codon usage information can be integrated into those models to classify newly discovered HPV phenotype. And these models can be really helpful to the clinician for early diagnosis of novel HPVs. So these are my references, and I would like to thank DBT for providing being philosophy. Thank you.
Thank you so much, Karanveer, for your presentation. Next, I would like to invite Ruthvi Vaja.
Is my screen visible?
Yes, it is.
Okay. So, hi everyone, I'm Rita Sharma, and I'm currently an undergraduate student in the field of biomedical sciences and research. Today, I would be presenting my research on gene expression meta-analysis of colorectal cancer tumor cells that reveals genes in association with tumorigenesis. To start with the introduction, more than 12 million people are every day diagnosed with colorectal cancer, and 600,000 people die from it. It's very deadly disease. However, the pathogenesis of colorectal cancer is still not known. This work identifies 13 gene signatures representing 4 CRC tumor gene signatures and 10 other glandular tumor gene signatures that are colonic by origin. Gene set enrichment analysis approach have been used to define positive and negative CRC gene panels from GSEA identified leading edge genes. Leading edge genes were identified and later used to verify enrichment and significant edge gene membership of CRC panels in different types of glandular tumors in origin. GSEA-based approaches here used to analyze the diseases and mRNA expression data of tumor and normal colorectal cancer tissues derived from human colonic biopsy samples by defining and comparing gene expression signatures. In order to examine the gene expression changes associated with tumor formation, in CRC, differential gene expression analysis was carried on using Welch t-test two-sample analysis. The t-test score of clean and normalized value was used in order to define the gene panels. The t-test scores later defined 13 gene signatures which were of statistical significance. The signatures are formed from the gene list ranked from high to low differential gene expression between tumor versus normal samples. The signatures derived from the same data set use the same control samples. Now, leading edge genes from these analyses were examined, and shared leading edge genes were used to define two CRC tumor versus normal gene signatures, whereas another panel was normal versus CRC tumor gene signatures. Out of this, in the methodology, we had gene verification and gene comparison data sets also. So for gene verification, in order to assess if the results generated from GSEA could be achieved randomly, 1,000 gene panels consisted of 175 genes to match the average number of genes for positive and negative CRC tumor gene panels. So the two identification data sets were used, and later on from them, the gene signatures were derived that could be used for gene verification and gene comparison. GSEA was again used to compare the identified CRC gene panels from the original identification data set and the gene signatures derived from the samples of one of the 6 glandular tumors, and later on were used to compare and contrast between glandular tumors such as hyperplastic polyps, polypsia, adrenal tumors, and further on. Talking about the results, the leading edge genes from GSEA across other glandular tumors were examined to identify genes of potential interest. In this study, out of 55 genes from the positive CRC gene panel, and 54 of them were continuously expressed among the all other glandular tumors in origin, which is a statistical significance as well. Similarly, in the negative gene panel, out of the 77 genes, 75 of them were constantly expressed in all other verification and comparison data sets. Now, in order to analyze that are these genes that are expressed are significant statistically or not, we again did a gene comparison study and did a t-test for this gene panels. Now, in this t-test, out of the 55 genes, 50 of them were of statistical significance, that is p-value less than 0.001, and out of 75 genes, 71 of them are of statistical significance. Moreover, this work also identified certain kinds of solid carrier family genes which are associated with tumorigenesis and the process of colorectal cancer, which could be helpful in identifying how this tumor spreads, and it could also be used as a therapeutic target for the people who are suffering from this disease. This work also had several kinds of limitations, such as biological implications. For example, the gene expression changes that were associated with certain kinds of genes were not observable, that were mentioned in the literature review of the data sets. Platform variations in both the gene inclusion and primary nucleotide sequences can substantially affect our results, and we have seen that in the statistical significance results as well. However, if the desired outcome is potentially prioritized and the list of the leading edge genes can be further used for laboratory or clinician examination. A lack of direct experimental approach is still lacking in order to find out therapeutic targets and certain kinds of predictive biomarkers for the colorectal cancer. However, our work that identified certain kinds of solid carrier family genes could be useful, and it needs to be proved by wet lab research or Western blot techniques or qRT-PCR techniques in order to validate that these genes are involved in colorectal cancer disease tumorigenesis, which could assess that the GSE-based approach that we used is more useful in identifying certain kinds of genes which normal bioinformatics tools like fail or are not that efficient to derive. So thank you so much.
Thank you so much, Ruthvi, for your presentation. Next, we have with us Dr. Mohana and team, and from a team, we have Dr. Ankita, but I see Dr. Mohana joined as well. So I'll pass on to the team. Dr. Mohana, over to you. Dr. Mohana, I can't hear you.
Hello, Ojuswi. It would be really great if you can make co-host to Dr. Ankita as well.
Yes, Dr. Ankita is the co-host now. So for all the judges, Dr. Ankita and Dr. Mohana worked on the same project, and Dr. Ankita will be representing the poster. Dr. Ankita, can you unmute yourself? We can't hear Dr. Ankita, Dr. Mohana.
Yes, hello.
Yes, we can
Am I
hear.
audible?
Thank you.
Yeah,
Yes, you're audible.
Am I audible?
Yes, ma'am, you are audible.
Okay, you can see my screen, right?
It's coming up.
Okay. Okay, can you see my screen now?
No, I can't see your screen. Yes, I can see your screen now. Thank you. Can you go in the presentation mode? We can't hear you. Are you speaking something?
Yeah, I'm just starting my video, but I think it needs permission.
Okay, so if it's not starting, maybe you can start with your presentation. I think it's on from your end, but it's not coming up. Yeah.
Okay. By the time it starts, okay, I'll start presenting.
Yeah, sure. Thank you.
Yeah. So, good evening, everyone. I'm here to present my review article on oral potentially malignant disorders and oral squamous cell carcinomas.
Dr. Ankita, you're not audible. Can you confirm if it's not audible to you as well, so that I know it's not an issue at my end?
Yes, Ankita is not audible to me as well. It's another one.
Okay. So I think we can have, okay, so we have the recording by Dr. Mohana and team already, right? On the poster presentation page. So I think the judges can go through the recording by Dr. Mohana and also look at her poster and assess the same. And so Dr. Mohana was our last poster presentation competitions participant. Actually, we had a lot of submissions for the poster presentation competition, and these were by two categories, by OmicsLogic research fellows and also non-research fellows. So we gave a chance to non-research fellows to present in the presentation competition, and we have the presentations by our research fellows on the page that has been given to all of the judges for assessment. It would be good if I could also pull out that page and showcase to all of you. And meanwhile, I do so, I would like to hear from the judges. So Dr. Vivek, I would like to hear from you. What is your thought about the presentations?
Hi, this is Vivek here. Sorry, I'm not able to put on
video, but thanks for organizing this. It was really, really great, and I am very happy to see some very young emerging talents. In fact, it was very nice and crystallized to have people speaking about the overall scientific process, the overall thought process, the hypothesis, like what went behind it at a very early age. So I'm very pleased to see that the training is going on a very early age. Very nice talks and very nice poster presentations. I've gone through some of them, and I'm very glad that this was organized well.
Thank you so much, Dr.
Thanks
Vivek.
for adding me. Thanks for having me.
It was a pleasure having you in our panel of judges. And now I would like to hear from Dr. Varina Ras. What is your take on the presentations we have had so far? And also, I think you have been going through these presentations on the page as well. So yes, over to you.
Yes, so I have really enjoyed looking at all of the presentations. I've also really enjoyed listening to everybody today. I must say I'm quite impressed at the caliber of the presentations, but also of the work. And it's really great to see some early career researchers really tackling some real world problems. And that's been very exciting. All of the talks today did some hectic analyses, and I think you should all pat yourselves on the back. And I think you should also be very, very proud of your progress in such a short span of time. It's been truly impressive, and I'm really, really enjoying it. And I hope to be here for a lot more of today's session, so that I can hear some more from everybody else. But it's been really, really wonderful, and I've really been liking the posters as well. The posters are fantastic. So just well done, guys. It's really good.
Thank you so much, Dr. Varina. It's very motivating, actually. So thank you so much for your kind words. And yes, these young birds are doing amazing job. That's true for sure. And this is the poster presentation page, by the way. So I'll just share the link of it in the chat box for all of the participants and the listeners to look at it as well. We have had a lot of poster submissions, and it was really tough to choose the best out of them. So out of the 50 submissions that we received, we have put together some nice presentations that you all can go through. And also the others were amazing too. But yes, we definitely had to pick. So we chose a few of them. Next, I would like to hear from Jaco Maiden. What do you think about the presentations we have had and also the ones that are there on the page?
Thank you very much. I think the presentations were fantastic by all the participants, using different types of advanced methods to answer different research questions, understanding the domain knowledge, and in my opinion, most importantly, working with different types of data and understanding how can this data be used to answer those research questions. I think all the participants did a really fantastic job, and that's it from me. Thank you.
Yeah, well, thank you so much, Jaco, for your views on the posters and the presentations. I would now like to hear from Julia. What do you think about these presentations?
Thank you, and thank you for the invitation to be at this event. And I really enjoyed it. I really enjoyed all the projects, and there's a lot of them. So it will take some time to go over, I think. But yeah, just as it was said before, it was really, really interesting to hear the different data types that people were using and the different tools and software, and sometimes also something new. So it was really exciting to hear the students and the presenters. I really enjoyed it. So thank you.
Thank you so much, Julia. And on this note, we are now good to start with our OmicsLogic Africa team and listen to their speakers and their programs and progress so far, what they have been doing all around the year. So on this note, I'll be now handing over it to Ayan Faye, Community Manager, OmicsLogic Africa. So I think it's Erikunle who has to begin. Erikunle, are you here with us? Okay. Ojasvi, can you make Erikunle and Ayonfe co-hosts so that they can start with their sessions? Thank you so much. And meanwhile, Erikunle and Ayonfe begin with their part. I would just like to notify the poster presentation competition participants that we'll notify you soon about the results once the judges have gone through all of these presentations. It'll take some time to review all of them, and we'll let you know the results in the session that we'll be having to announce the results.
All right. Hello, can you hear me?
Yes, we can hear you.
All right. I want to confirm if Adekunle is on board. Hello, Adekunle, can you hear me? Hello, Adekunle, can you hear me? All right. I believe he's having some technical issues, but I'm going to share my screen now. So okay. Okay, I think Adekunle is on board. Hello, Adekunle, are you there?
Hello, can you hear me?
Yeah, we can hear you now.
Great. I'm here with you. Good afternoon.
Yeah, so can you share your screen or should I just share it from here?
Okay, let me do that, please.
Okay.
All right. Can you see my screen, please?
Yeah,
Yes,
I can see it, but you need to show the presenter's view. Yeah, okay, I think it's perfect now.
Yes, thank you very much. All right, good afternoon, everyone. I welcome you all to the African OmicsLogic Research Symposium with a thematic focus on bioinformatics education and training around the globe. I'm convinced we've been having an enriched and stimulating scientific discourse over the course of the time these symposium has started. And at this point in time, we are moving on to the African OmicsLogic Africa section, where I will have the privilege of introducing us to the OmicsLogic. Okay, great, yes. Where I'll have opportunity of introducing OmicsLogic Africa. And as part of that, I'll be talking about the programs we've had here in Africa in the past few months, as well as some of the partnerships that is in the works for us in advancing bioinformatics education here in Africa. Moving on, I'd like to introduce myself. My name is Adekunle Adeluoye. I'm a faculty with Lead City University here in Nigeria, and I'm an associate of MLSC, and that is the Medical Laboratory Science Council of Nigeria. Importantly, I'm a mentor and facilitator with OmicsLogic trainings of the Biotech Incorporation USA, and I double also as the program director for OmicsLogic here in Africa. So the OmicsLogic sets of bioinformatics and big data analysis training programs of the Biotech, which has been globally proven by peer reviews and curriculum experts in our partnering institution, is being inventively explored in closing the need gaps that we have identified here in with regards to bioinformatics education and training globally. But of great importance for us is Africa, where we are located, and what we are doing in terms of these scientific in terms of these is with respect to building scientific capacities for researchers, for students, for professionals, practicing on the field and across the continent. Our postulation, of course, is that increasingly, by advancing the knowledge of bioinformatics and its use in obtaining scientific insight, from sequence data, there will be a significant growth and development of the bioinformatics community here in Africa, and its considerable implications, of course, will be for translational science, from understanding of the gene functions, as well as its role in biology, in agriculture, and in medicine, as well as several other, peculiar space. So with this, we are guided by the OmicsLogic goals, okay? And so we are pursuing this with the goals set out by Biotech for the OmicsLogic programs, and this includes the provision of training in bioinformatics that is geared towards enabling independent researchers to be able to follow peer examples and get guidance from mentors to carry out their research. We also seek to develop a growing community of bioinformaticians with a shared passion for data-driven research and acquisition of citizen science. Likewise, our goal is to enable students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology, in life and data sciences. And so, working with these set goals, OmicsLogic programs that we bring to our participants across the African community attempts to introduce participants to big data and NGS bioinformatics training, which is designed for everyone interested to learn and apply data, data science skill sets and computational biology skill sets to biological data sets. And so the OmicsLogic programs boast of everything a participant actually needs to get started and grow in their bioinformatics and big data analysis space for both research as well as in their practice. So just as we know that the generation of biological data has witnessed significant growth in recent times, interpretations of this process data now presents a huge challenge to everyone, requiring, of course, most times multiple steps, bioinformatic tools, human capacity that are required to generate, final results from those raw data. And so, while the growing interest in bioinformatics and big data analysis continues to grow here in Africa, many of these keys are far from being able to, many of these scientists are far from being able to utilize or gain access to this key set for utilization in their scientific research as well as practice. And so these gaps are what we at OmicsLogic Africa, we are bridging with our training programs for students, faculties, and researchers, across the continent. Similarly, the lack of dynamism, when it comes to continuous reviewing of curriculum, or as well as updating of curriculum for our educational system by the educational regulatory bodies, also further means that many are being disadvantaged in terms of learning these essential bioinformatics knowledge and skills in tandem with what is obtainable in the global scientific space. And so our approach at OmicsLogic Africa is exploring university partnerships so as to integrate our hands-on, mentor-driven, globally proven training methods and resources into university educations, both at undergraduate and postgraduate training levels, which, of course, we believe will help scale rapidly the growth and capacity of bioinformatics, within the continent. And so, in the course of the last 12 months, we have organized, a number of programs to bring bioinformatics training with project-guided learning and resources, to a cross-section of participants. That ranges from beginners who are totally new to bioinformatics, as well as students, and many other faculties, researchers alike, to introduce them to bioinformatics and help develop their skill set and research capacity in order for them to be able to explore the world of omics science and using bioinformatics and big data analysis knowledge and tools. So this high impact training includes, of course, basic OmicsLogic training. We also organize OmicsLogic metagenomics training in time past. Also, we have conducted the data science for biologists training, and likewise, we organize introduction to bioinformatics and computational biology bioinformatics for biomedical drug discovery. So these are series of programs that we have, been able to, package and bring to a cross-section of participants in the past month. And also similarly, just as I mentioned the other time, our goal is to explore partnership with notable institutions, universities here in Africa, in order to be able to mainstream this training, this OmicsLogic training into their curriculum for their undergraduate and postgraduate students, as well as faculties alike. So a few of such notable partnerships we've been able to forge towards achieving our bioinformatics education and training goals includes these with Lead City University here in Nigeria. We also have Crescent University also on our partnership currently. Likewise, we have the multi-research laboratory of Osun State University here in Nigeria as well. And lately, we had a working partnership with the University of Yaoundé in Cameroon, where we also brought our OmicsLogic training, to the cross-section of participants from these institutions. And I'd like to, at this point, share some of these notable, sites from some of those partnerships with us, which ranges from the multi-research laboratory of Osun State, as I mentioned earlier on, and include, on to, Redeemers College of Technology and Management here in Nigeria. We equally have Adeleke University here in Nigeria, Lead City University, as I mentioned earlier on, as well as Crescent University, where we have, interacted with experts and, management faculties to bring these, trainings across their students. And I'd like to, share at this point some of the reviews, notable reviews we've heard from our previous participants. Here's Odinaka Kingsley Obeta, who once told us, it's my first time learning bioinformatics, and I enjoyed the course a lot, looking forward to more opportunities to learn more. Odinaka is a medical laboratory scientist, is a practitioner who sees the needs for him to gain these essential skills. Equally, we have undergraduates, and here Adetutu Oluwasanmi is representing this group of participants. She has to say, everything was new to me, obviously. However, it was easy to understand. So we're happy that we're able to carry along the likes of Adetutu Oluwasanmi in our training. And equally, we have this coming in from Samuel Emeka Peter, who is a postgraduate student from University of Lagos here in Nigeria. It was a great opportunity to gain an experience I'm glad to have. I garnered some information that the amount I paid for the program isn't commensurate to. It was an eye-opening period I would be glad to recommend by my friends to equally experience. So these are some of the notable reviews we've gotten from our participants, even as we get on in our efforts to bring these trainings across more participants within the African space. So I'll conclude by adding that African continent has been regarded as a cradle of modern humans, and also the African genomes has been reported to contain more genetic variation than those from many other, other continents. And so this, of course, presents a rich focus for scientific studies. Hence, the need not to, to carry along the African continent when it comes to genomic research, and discoveries. So as we know, bioinformatics has become an indispensable tool, for management of this hugely generated data in modern biology, medicine, and other related fields. However, it is still something to grasp with. The fact that infrastructural gaps exist here in Africa and other challenges as well with our educational system, particularly with the curriculum and integration of this bioinformatics into the four walls of the university. All right, so hence the need for such approaches, innovative approaches like what we are doing with OmicsLogic Africa in advancing bioinformatics education and training across the continent. So this is what OmicsLogic program of the Biotech is set to achieve, playing a strategic role in scaling the bioinformatics education and training needs, as demonstrated so far. So on this note, I'd like to appreciate every one of us, but then permit me to mention that shortly from now, as part of the African presentation section, we will be having a panelist section where the panelists here will be doing justice to the discourse around bioinformatics education. So I encourage us all to stay tuned. And so on that note, I want to thank you for listening, and you can reach us through Africa@pi.bow. Thank you very much.
Yeah, thank you so much, Mr. Adekunle. That was a very brilliant presentation. Yeah, I really got a lot from what you actually had there to share with us. I will be sharing my screen now. I hope you don't mind.
Yes, please.
Okay, thank you. Please confirm if you could, if you can see my screen clearly. Yeah, so once again, I want to welcome us all, and we have heard so far about the progress of what the OmicsLogic Africa team is actually doing in Nigeria. In a bit, I will be calling on few persons. I hope we are testy enough to look out for these persons that will be joining us very, very soon. Yeah, my name is Ayonfeoluwa Alabetutu. I am a molecular biologist and geneticist, and I'm a PhD candidate. I hail from Nigeria. I got my higher education in Penza, Russia, and I am a faculty with Rosom Academy in Russia. I'm a blood pathogen certified expert, and I am also an enthusiast of data analytics, data science, population genetics, bioinformatics, regenerative medicine. I am the OmicsLogic Africa Community Engagement Manager. So it's so lovely to be here. On this note, I would love to prepare us. Please, I would love that Professor Charles Oluwaseun, I would love that he's given a co-host right now as I read his profile. So we will be having a talk session now where Professor will be bringing us some insight into bioinformatics research in Africa and the impact that it has on the world. Professor Charles Adetunji is presently a faculty member at the microbiology department. He's the acting dean of the Faculty of Science at Edo State University in Nigeria. Where we utilize the application of biological techniques and microbiological processes for the actualization of sustainable developmental goals and agrarian revolution through quality teaching, research, and community development. He's currently the acting director of intellectual properties and technology transfer. He is the AG Dean of Faculty of Science and Department of Microbiology in Edo State University. He's a visiting professor and an executive director at the Center of Biotechnology, Precious Cornerstone University in Ibadan, Nigeria. He has won several scientific awards and grants for renowned academic bodies like Council of Scientific and Industrial Research in India, Department of Biotechnology in India, the World Academy of Science in Italy, Netherlands Fellowship Program, and so on. He has won grants from the Agency of International Development and Cooperation in Israel, Royal Academy of Engineering in the UK, among other places. He's published many scientific journals and articles and conference proceedings in referred national and international journals with over 370 manuscripts. He is ranked amongst the top 500 prolific authors in Nigeria between 2019 till date by Scopius and Cyber. His research interests include microbiology, biotechnology, post-harvest management, and nanotechnology. He has recently been appointed as the president and the chairman, the governing council of the Nigerian Bioinformatics and Genomics Network Society. He's presently a serial editor with Taylor's and Charles USA, editing several textbooks on agricultural biotechnology, nanotechnology, pharma foods, and environmental sciences. So he's an editorial board member of many international journals and serve as a review of many double-blind peer-review journals like Elsevier, Springer, Francis and Taylor, Wiley, PLOS ONE, Nature, American Chemistry Society, Bedlam Science Publisher. Is a member of many scientific and professional bodies, including the American Society of Microbiology, Biotechnological Society of Nigeria, Nigerian Society of Microbiology. He's presently the General Executive Secretary of the Nigerian Young Academy. He has won a lot of international recognition and has also acted as the keynote speaker, delivering invited talk, position papers for various university research institutions and several centers of excellence, which span across several continents of the globe. He has over the last 15 years built a strong working collaboration with reputable research groups in numerous and leading society. At this point, I would love to call on Professor Charles to give us, to take over. So Professor Charles, are you there? Can you hear me? Yeah, Professor Charles, can you hear me? We can't hear you. Professor Charles, are you there?
Good evening, everyone.
Yeah, good evening. We can hear you now. You may need.
Can you see my screen?
You may need to switch your screen to the display. I mean, to the.
Yes, we can.
Yeah, can you hear me? Can you see me?
Yes.
We can hear you. We can see you.
Okay.
But continue, sir.
Okay, good afternoon, everyone. Can you see my screen?
Yes, we can. Yeah, we can see your screen.
Hello?
Yes, it is visible.
Okay, good afternoon, everyone. I'm so grateful, Dr. Ayofe Labetitu, for bringing me to this great conference. I found it as a great privilege to be talking to you. Today, I'll be talking about some recent bioinformatics innovation at Kinase known for numerous global challenges. And today, as we know, around the world, a lot of people are going through a lot of challenges, a lot of problems. So it is now high time we come together as a scientist in order to solve the global problem. Now, COVID-19 was not envisaged in some years to come, and bioinformatics has really played a lot of key role. Now we are in post-COVID era, which shows we need a lot of things. So this is, I serve as the president of Nigeria Bioinformatics and Genomics Network, and a distinguished professor of biotechnology, bioinformatics, microbiology, Aspiration, and also I'm the executive secretary of Nigeria Young Academy and senior coordinator of West African Microbiology of Nigeria. So before we go on, this bioinformatics of the thing we are talking about, where does it come up? It stems up from the Word of God, which shows everything encompassing about God. So which from the GTCT of everything, no matter whatever we want to discover, want to grow about, everything is all about God. So what are we saying in essence? Bioinformatics have become a sustainable tool that will help us to achieve the digital revolution about the sustainable development we have been talking about the sustainable city, sustainable food, water, health being, well-being of mankind, energy, decarbonization, and talking about education, gender, and a lot of other stuff. So we show circularity on a lot of things and a lot of place to play. Now, the whole world is going through a lot of low carbon economy and a lot of other things. So bioinformatics have become a sustainable tool that help us to have a kind of economical, eco-friendly, to manage a lot of things, talking about metabolomics and a lot of other products. This are not shows that bioinformatics have become a kind of eccentric cycle that encompasses around innovation, sustainable environment, and circular economy. Now we are trying to digress from linear economy. Now the whole world is talking about circular economy. Now, which shows that bioinformatics have a lot of roles to play around the whole globe, most especially during the era of post-COVID. Now, talking about Industry 4 have a lot of things to play with the help of bioinformatics. We're talking about 9 pillars of technological advancement. Now we are talking about 11 pillars of technological advancements, and developments of bioinformatics have played a lot of crucial. We're talking about industrial stream, industrial electricity, industrial coupling, and industrial intelligence have come to play today. That's what we are talking about. Now, I would like to share some of my experience as a distinguished professor in the area of bioinformatics, biotechnology, and bioeconomic biologists. Now, before I go on, there's a particular model that has really helped me in achieving some of these sustainable development goals. Most of my theory based on how to emphasize, define, ideate, prototype, and test. Before bioinformatics can become adaptable everywhere, we first of all need to go back to our local community, identify the major challenges. We have to emphasize, define, ideate, and test the prototype. Now, know that many of us have not done a lot of things, but we are more key into what is called translation research. So there is difference between research translation and translational research. Now, before we are talking about Millennium Development Goal, but now all of us are talking about Sustainable Development Goal. We form a kind of impetus for promotion and defense of efforts as well. So bioinformatics as a tool have become a crucial tool for us to have this diverse set of development goals. Now, it has been stipulated that the whole globe is going to increase drastically to 9 billion in the year 2050. As we know, FAO estimated that there is going to be almost 60% transformation and in terms of agriculture for us to meet the band of the ever-increasing population. Now, the issue of climate change is there, so which shows definitely bioinformatics have a lot of role to play. Now, there are 4 major goals of our time. How do we achieve food security, issue of climate change, health, and high rate of unemployment? Now, my research group have been able to discover a new bioherbicide to the help of bioinformatics, which we have established the molecular basis, and we have done a lot of sequencing, and they're showing the phylogenetic tree, which shows the relationship between various strands of the bioherbicide that could be used to form a kind of permanent replacement to chemical synthetic herbicide. And this is showing the publication, another publication that shows the stability and the active compound, and through the use of some bioinformatical techniques in order to establish the most active ingredient present. And further, we went further to show the mode of action that also published a paper and documents. This showing the term and the biochemical activity that join together with some bioinformatic results, which I will still explain further. We went further to see the metabolomic aspects and carried out the structural elucidation using the HPL, CFTR, and NMR, and caused the 45, 95, 105 caused it to show and establish the relationship between the genomics, bioinformatics, metabolomics, and the proteomics of the herbicidal agents. Not only that do we stop at the research level, we went further to document the patents in this regard, and we have done it. Now, during COVID, we do a lot of works that have to do with artificial intelligence, edge, and a lot of Internet of Things-based smart agriculture, because we discovered that bioinformatics have come to be a sustainable tool that we could use. So what do? We research into the use of agrobots that can become a kind of robot, most especially in unaccessible area that we have challenges of some infrastructure, especially in Nigeria. So some of my students are recently working on that. We went further, I was talking about climate change earlier. So we went further to see how climatic conditioning could be monitored and automated system could come to place. Not only is that we went to start on social commercial implication of social security, then we went further to show and establish Internet of Things in monitoring aquatic environments and a lot of other stuff. Then we went further to see how we could design a kind of intelligence system that could enhance a saltwater stream production using OmicsLogic. Not only that, during 2021, when there was COVID, my research team that is being headed by Dr. Wadazani from India. We went further to show evolution of protein structure, analysis of various cassava mosaic, because cassava form a kind of staple food that is being consumed and used by many people in a lot of industry. And it starts as a source of carbohydrates, but the yield loss ranges around 1.9 to 2.9 billion per annum in the eastern and the central world of Africa. Now, protein called virus we're researching to because they have a kind of multifunctionality for us around the virus and all stages of various cycle. So what do we do? There is a lot of information on whether the SCM cassava mosaic virus isolates have a kind of relationship that relates around the protein codes, and we get a kind of new biopesticide that could be used to manage. So what do? I employ our research group use a lot of bioinformatics techniques to achieve this, and this has been established, and this is showing a lot of protein code families around it, and showing the phylogenetic tree around the protein code, employing the neighbor joining method using Mega S software. We went further to show the graphical alignments of the COVID-19, and we went further to show the distribution of the conserved muscle region in the cassava mosaic vira. Not only that, we were able to establish the three-dimensional homology mode of the cassava mosaic virus. And we were able to compare the biological process ontology, the cellular product component ontology, and molecular function ontology, and the graphical representation of the active gene. So during this, we discovered that the protein codes could be used and serve as a kind of new bioprocess that could be used in regulation of management of cassava mosaic virus. So this is showing the gene reports where we publish the article. Also, our group headed by Dr. Razani also projects a lot of activity on activity, and we did a lot of robust profiling of the cytochrome P450 in agriculture, Melvica, and industrial notable Aspergillus in order to understand more insight into the evolutionary origin, family, expression, and protective function. Now, what do we do? This work will also receive a very big award, but was sponsored and given to the leader of the group, Dr. Rosali. He got a grant by American Society of Microbiology and Federation of European Microbiology Society, and Melinda Gates that gave us a travel award to America to present this particular one. So what am I saying? In essence, we were able to establish the taxonomic distribution of the protective in various Aspergillus strains, and this showing the evolutionary trends. We're able to establish the prediction of the subcellular localized analysis of the protein gene in the several Aspergillus, and this is showing some of the secondary metabolism gene cluster for the various Aspergillus that we tested. Also, we went further to establish the wide genome of Saticoma Athanaria species, and this is showing the evolutionary relationship between the P450 gene in 13 Athanaria species, and this shows their subcellular cytochrome profile in various species, and so it's the secondary metabolite in the 13 Aspergillus. And this is showing the publication with recently group, our group, that's where we have established the role of the Aspergillus, and we went further to establish the evolutionary relationship between them, and this is showing the various genes. Also, in Alternaria, we have been able to do that, and also we've been able to establish comparative phylogenetic analysis of the cyclone paper for monogenesis from Fusarium species. Also, this is under consideration because Fusarium and some other sponge species like Aspergillus parasiticus because a lot of problem of aflatoxin. So our group have been able to establish through the help of bioinformatics tools to establish some autosogenic strains that could be used and establish a kind of jointly competitive with toxigenic strain. That's this they could use to eradicate toxigenic strains Aspergillus that is responsible for formation of cancer and metabolism. So my group also work on crystal regular interface short pattern spread called CRISPR, and we have used it. And recently we won a grant from USA, EU, Africa, Asia, Pacific, and Caribbean Initiative. And in order to see with the help of nanotube and genetic engineering and some other thing, how we could easily pass genetic material into the, and this is showing the graph. And we have done a lot of work in application of bioinformatics to establish active compounds for microorganisms, and this shows some of the results of the blast we've been able to get and establish. And presently, we now discovered some vegetable entail beneficial of our microorganisms like endophytic, established very safe comparative in lab, and they could be used towards the progression of a drug that could be used for management of several diseases. Now, also, we also use some bioinformatics and natural resources to see in silico screening of selected medicinal plants could serve as a potent innovator of transmedia prophase, necessary for SARIN2, implication for COVID, whenever they understand. So in this particular study, over 132 phytochemicals from Zinnia venalis and Aspirational annual and Moringa lovera was screened as a potential innovator of transference membrane serine. And we were able to establish using silico method to establish, and this showing the sequence alignment of the protein sequence, and this is showing the protein plots for the variation of the modern disruptive protein. And we went further to show the result of the modern traits of polysynthesis we use in our program. And we also show the 3D counter-trypsin protein, Marvin, and a lot of interactions with the residue of tryptophan and scolarin A. Hanum, and this showing for the Moringa lovera, and this showing the publication where we have documented our findings. So CRISPR, we've used CRISPR also in our group for a lot of things for management of COVID-19 disease, and we have done a lot of work on our machine learning and deep learning in management of COVID, and also we've established a lot of work in the area of function extended reality in healthcare, machine learning, a lot of virtual augmented and mixed reality. And we went further to biomedical data and transfer learning, high alternative of a thing in robotic healthcare, and cyber integration in American application of 3D enabled prototype customized research and method healthcare. And CRISPR has been used by some other group, not my group, and management of sickle cell disease, in gene therapy, and in other gene therapy, and they've used it in management of liver disease, and also for sabotage of organs in pigs, and they're reducing malaria. They're using reproductive medicine, and CRISPR baby also is going on therapy gene. A lot of work has been done in that, and HIV management, cancer management, in fixing a lot of hematological disease. Other CRISPR been used for genetic screening and feature propagation also, and a lot of application for a lot of things. A lot of scientists have done a lot of work and application of bioinformatics in production of vaccine, and a lot of work is still also going in Nigeria. Also, during this, we've used a lot of diagnosis techniques for management of AV, machine learning for management of COVID, Internet of Things, Internet of Air Things, for COVID management. Smart Sensing, now my research group, some of my PhD students are working in the development of biosensor that could sense, and we use a lot of that for biomedical in producing of this biosensor, and we use a lot of data techniques for diagnosis also in treatment of COVID-19. Now, on the whole, for me to round up, it has come to a point that we, in kind of multidisciplinary approach, need to integrate whatever we are doing and encompassing, as a university said, making bioinformatics as the model. So that means we have to allow transdisciplinary, interdisciplinary, multidisciplinary, and multidisciplinary needs to come together. We need to work together for us to form and solve majority of the global problem. Now, I will say that we need teamwork. We need to carry the students at secondary school and that. I remember some of the time the CBN government came to my university now to see whatever we do. So we need to carry government along. We need to involve people. So at this point, I would like to invite many of you to join my series at US. I'm a series editor to several textbooks. So you can join me and you can see some of the projects that they switch by bioinformatics and other biotechnological advances that we have done. You can join them. And this is my company. You are welcome. And I would like to invite all of you to recent advances in biotechnology, one behind the program with it, and this shows some of the programs we have held in the past. And definitely, as a bioinformatician, why am I showing this? We need awareness. We need to sensitize people. We need to conduct training. This is some of the training where we invite expertise in the area of bioinformatics that they visit Nigeria, and we can still collaborate for more of this. Not only that, we need to talk to the government. I was invited by the Senate of the Federal Republic in 2016 and last week to come and talk about science. So we need to talk to the government about what we are doing. So in view of this, let me say thank you for all the opportunity. These are some of the professors that have really helped me in some countries, and also looking forward to collaborate with you and talk for more opportunity.
Thank you so much, Professor. You've actually touched a lot of these things, and you were able to go through 175 slides within that period of time. Thank you so much. We've seen how that professor has touched on different aspects of research, even from the microbiology perspective to integrating genomics and doing quality research. Thank you so much, Professor. It has actually really been a great time being here with you. It has actually been a very, very great time being here with you. On this note, I would love to say that our next presenter, she is on ground, and it's so lovely to really have her here. I'm going to share my screen now so that we will be able to see the next person. Can we see my screen?
Yes, we can.
So can we see my screen? Can you hear me too?
Yes, yes.
Oh,
go ahead.
Okay. So real quick, I'd just love to introduce Dr. Vinira Raz. She just have a simple introduction because of the time. She is a training and outreach coordinator with hereditary health and what's the third age? You're gonna get to know Bio Network South Africa. So Dr. Vinira Raz, are you there?
Yes, I'm here.
Okay, please, can we give her the co-host right so that she can share our screen?
She's already a co-host.
All right, please just go into the full screen mode such that we can see your screen maximally.
Can you just confirm that you are seeing my screen and not the presenter view?
Yeah. So we can see you now.
Great. Okay, perfect. So hi everyone. Yeah.
We can see your notes.
Presenter view, yeah.
Okay, sure. Let me just see if I can switch.
It is kind of in a presenter mode. We can still make use of this. Yeah, perfect now.
Sure. Okay, sure. You can see it now?
Yeah.
Perfect. So hi everyone. I am Rita Sharma. I package. So I just want to thank everybody for firstly inviting me to be with you all today. It's been a really wonderful day so far. Actually, I've really been enjoying all the presentations, all the talks, and learning from everybody else's experience as well. So I'm getting a warning that my internet is a little bit unstable. So if you all don't mind, I'm actually going to just switch off my video so that my screen stays up and you can hear my audio. But I'll switch it on a little bit later if my internet stabilizes a little bit. Right, so just to give you an overview of what I will be just touching on briefly with this talk, I would like to give you all a brief overview of HCA Bionet and who we are. I'd like to cover very briefly as well some of the HCA Bionet training and also our training models. I will discuss our multiple delivery training model in a little bit more depth than some of the other things I'll talk about, because this is a model that we really are proud of as an organization. And a lot of development went into this model, and I'll talk to why it's been such an amazing model for us. And then I'll also talk to how we've adapted this model more recently for much more advanced bioinformatics training. And then I'll touch on some of the other mechanisms that we are using to share our training and make it a little bit more reusable, a little bit more findable, and interoperable. So I'll speak to FAIR and bioschemas a little bit, and then I'll speak to the DSI Africa initiative, but more specifically ILWAZI, which is the Open Data Science Platform, and the group that I'll be forming part of going forward. So for those of you who might not be too familiar with us, I just thought I'd do a quick introduction to H3A Bionet. So H3A Bionet is a Pan-African bioinformatics network, and we are part of the Human Hereditary and Health in Africa Consortium, also known as H3 Africa. And at the moment, we are currently comprised of 28 partners, and we are spread across 17 different countries. And so we have quite a nice representation across Africa already, which I'm quite happy about, and I'm sure everybody's quite proud of. And we have over 200 members at the moment, so we are quite a large consortium at the moment. But H3A Bionet was established to develop bioinformatics capacity in Africa, and more specifically, to enable genomics data analysis by H3 Africa researchers across the continent. And that was our initial goal and aim, and it's expanded a lot more since then. But mainly, basically, H3A Bionet is developing human capacity through training and support for data analysis, and also facilitating access to informatics infrastructure by developing or providing access to pipelines and tools for predominantly human microbiome and pathogen genomic data analysis. And so the major goal, of course, of H3A Bionet overall is then to increase the number of qualified bioinformatics graduates on the continent, but also to create research opportunities and also, very importantly, provide some financial support for promising new graduates in the area of bioinformatics. One thing H3A Bionet really wanted to do was to kind of slow the brain drain in Africa and attract some Africans back to the continent who are studying abroad. So to make Africa more attractive as a bioinformatics, leader across the world. And so one of the ways that we, of course, do that is through training. And that's predominantly what I do as part of H3A Bionet. I work with the training work package, and we work on, designing inclusive training models and just unique training modalities so that we can reach as many people across Africa as possible. And so H3A Bionet does training in a number of different ways. So we have run over the course of the last decade or so, a lot of face-to-face workshops. We've done a lot of trainer style courses. So we focused on developing capacity in trainers. We've also awarded quite a few internships. And so internships have obviously slowed down a little bit during the COVID years now, but before that, we did try to move graduates around so that they could get hands-on or experience in different labs. And that's also been something that we try to push quite a lot. We've done a lot of live and online training, and we also focus a lot on hackathons and jamborees, and each of these components are done with a different end goal in mind. And I want to say that when H3A Bionet just started, obviously the focus was a lot on face-to-face workshops. And so with that, we ran many face-to-face workshops, but we realized very, very quickly, we've got a huge network across Africa. And when we run face-to-face workshops, we are reaching 20 to 30 people per course or per workshop. And so that was really just not good enough. And so we wanted to create models that could reach more and more people across Africa simultaneously so that we could actually train more people simultaneously, and in that way also increase capacity for a lot of this type of work. And so one thing we really worked really hard on is developing this sort of multiple delivery mode training model. And so it uses a lot of different approaches to teach particular courses. The first course that we ran, and one of our flagship courses, is our Introduction to Bioinformatics course. And this course was run because, of course, there was a need for just foundational training across Africa. When H3A Bionet just started up, a lot of people wanted foundational training, but we couldn't run, 100 face-to-face workshops in a year. And so what we did was we decided, well, let's try this multiple delivery mode training model. And it was designed to reach multiple classrooms, multiple sites across Africa at the same time. And how the model works is that we would basically have institutions apply to host the classroom. So we'd have a call for classrooms. They went through a rigorous selection process. And if we if we felt that they had the capacity to run the workshop, they were accepted as a host. We then allowed a round of participant applications, and each classroom basically had their set of participant applications. They did their selections as well, and these participants then joined that classroom. But what made this model really, really unique is that although it was a distributed classroom model, within each classroom we had a coordinator, we had a teaching assistant, and we had a systems admin for on-site support. And then we had all of us in the background as the core team, also supporting those classrooms with an added level of support. And so what this allowed us to do is to reach hundreds of participants across numerous classrooms every year now. We're running IBT actually now for the 7th time this year. And last year we had something like 50 classrooms running at the same time, spread across about 15 or 16 countries in Africa. And we had close to 2,000 people taking the course at the same time. And that was really fantastic for us. So the whole course we feel is quite scalable. We started with about 26 classrooms and it grew organically to about 50 classrooms. I don't know if we're going to have more than that this year because my capacity is wearing a little bit thin to manage everybody. But yeah, for all intents and purposes, as long as you have more hands on the core team, the model can continue to grow on end, really. But what we did with this particular model is we made use of a lot of online tools initially because we wanted to remove that infrastructural burden on the classrooms because they were spread across Africa. They all had different issues at the time. Sometimes they were low resourced in terms of capacity. Some classrooms had intermittent internet. And so we all are aware of a lot of the problems in Africa. It's a very, very heterogeneous sort of environment. And so this kind of made the environment a lot more homogeneous because now everybody was using the same online tools. And so all you really needed was a computer and a browser and you were good to go. But then we started training thousands of people across Africa. And so now everybody was coming back to us and saying, well, this model is wonderful, but how can we do something more advanced? Or can we do something more advanced? And so at some point, myself and another training person at HA Bionet went, well, let's try and do something more advanced and let's see how it goes. And that kind of spurred and sort of led to the development of an adapted multiple delivery model. And so what we did with a 16S course that we ran for a couple of years now is we actually had a bunch of people from different domains come together. So we had us as the trainers and the curriculum developers. We had developers come together, sysadmins come together, and we decided, well, let's try and package all the tools, the data sets, software, dependencies, packages, everything into a container. Let's actually now try and get these classrooms to pull that container, install it, manage it, run it locally, and then see how they fare. And so what we did with this particular model is we had a smaller cohort of classrooms because now the infrastructure became very important. HA Bionet has invested a lot in infrastructural development across all of our nodes across Africa. And so we were quite confident that the computational resources were there in most cases. And so we also wanted to have them develop their own ability, both for 16S analysis, but also for running these kinds of courses and workshops on their own, even if HA Bionet disappears for whatever reason. So what we had them do was pull these containers, give all these students access to their servers or clusters, wherever the container was hosted, and they actually needed to run analyses in the cloud for the course. And so this, a lot of people kind of told us, you're a little bit crazy. We don't really think this is going to work. But I'm really proud to say that it has worked and it's been working for a few years now. And we've run, I think, about 3 or 4 iterations of the 16S course now. And at the last course, we had about 30 classrooms who all ran this course in this sort of model. And it worked really, really well. One thing we also do with the model is we actively manage everybody with the learning management site. And so we actually track every single participant all the time. And so we give them graded tests and assignments. There's lots of feedback tools, forums, and we manage literally every single participant quite actively. And we also grade everything that they do. And so this was a very exciting model for us to see work. It also then caught the interest of the international community, really. And last year, for the first time, we teamed up with the Wellcome Trust, and they've got a wonderful NGS course that they run. But their course is run obviously with a slightly different model. So they use a VM. And so we tried this model last year for the first time for that NGS course. The VM was incredibly large. It was about 40 gigs unpacked. When it was zipped, it was 20 gigs. And so each participant had to pull this VM and then, work with us via the learning management site and online lessons because of COVID. They couldn't join their physical classrooms anymore. So the whole course became virtual, but it still worked. It became a truly blended learning experience. But it was really exciting for us to see that whereas the Wellcome Trust could reach 20 people per workshop, and they ran the workshop maybe twice a year. Last year alone, I think we reached, over 300 people with that training, and it ran for about 3 months. This year, we have about 600 people on the course and 39 classrooms doing NGS analyses and really advanced analyses. So they're doing everything from QC to RNA-seq, ChIP-seq, genome assembly, really the whole shebang. They're working their way through the content and they're managing. And so that's also been really exciting for us. And so I won't speak to a lot of these topics in too much detail, just in the interest of time, but I did want to just touch on a couple of other things that HA Bionet has been doing to just increase and grow the impact across Africa as well. And so one thing we are focusing on quite heavily, not just in training, but overall, is the concept of FAIR. So making everything findable, accessible, interoperable, and reusable. And so we are making sure that we are really hitting all of those FAIR indicators, whether it be with our software and tools or with our training materials. And one of the ways that we are making things more fair is also by implementing bioschemas. And so we are currently testing the implementation of bioschemas. So bioschemas allow you to mark up your website with some semantic text. And so it allows obviously scrapers, web scrapers especially, to find your content quite easily, at least in the case of training materials. And so there are 3 different schemas that relate to training. And so we're trying to implement them. And what this will help other web scrapers and people who host catalogs of training, for example, to do is to automatically pull in any training we've ever done once these schemas are implemented. And again, in that way, we are making our training materials very much more findable and accessible. And it's also being duplicated in a lot of other repos, which I think is very exciting. We also have embarked and assisted with a lot of competency development for bioinformatics. So the International Society for Computational Biology has suggested a core set of competencies that they would like bioinformaticians to have. And so what we do is we actually use those competencies to drive all of our curriculum development. Even workshops and courses are mapped to competencies because it's very vital to us that we actually develop these competencies at an appropriate level for the audience or the user, the end user. So what the ISCB competencies have done in the past is that they've come up with a whole bunch of personas. I've just taken a snapshot of a couple of the personas. There are very many more than these. And then what they've done is taken their competencies, their suggested core competencies, and they've used Bloom's taxonomy to map out, at what level does all of these different personas need a particular competency. And so what that does is really just help drive the development of your course and make sure that you're hitting the competency at the correct level for that particular persona. And so I've just got a couple up here on the screen. You can see I've got a physician, a lab technician, an ethicist, and a biocurator, for example. And then I've just pulled out just a snapshot of some of the competencies. These are not all of the competencies. There are more than these, but these are just the first few. So you can see here that basically we use Bloom's taxonomy to map the level or the depth of knowledge that they would need or ability or competency for these competencies. So just for example, statistical research methods, we would basically suggest that all of them basically have knowledge to comprehension level in terms of Bloom's taxonomy, but a biocurator might not have to, so a biocurator would have to have this competency at this level, whereas the others could have it at a lower level. But we use it in that way to just kind of develop curricula. And it's been very interesting the way that our curriculums have actually changed as a result. And so these competencies are actually being further developed now. There's a paper coming out, I think, in maybe about a month's time, maybe less. where we basically are expanding these competencies to include more data science competencies, but also adding knowledge, skills, and attitudes. And so we hope that that will also assist everybody in using the competencies. And there's a lot of other developments happening around these competencies. So lots of tools that the ICB, for example, are working on, automated tools where you can go to the website, for example, and say, I am a physician and I want to learn X, and they will pop out what competencies you actually need to develop to be considered competent at a particular level. So that's been quite interesting for us. One other thing that HA Bionet is focusing on quite a lot are guides and SOPs. So if you visit our website, possibly after this talk, you will see that we have a lot of SOPs, a lot of how-to guides that we've been developing that's based on a lot of our work. I'm not going to talk to obviously all the others, but I'll talk to the training guide. We've developed quite a comprehensive training guide as well. So if you do not have experience in running bioinformatics courses, we've put together just a very short guide. It's not very comprehensive, but a quick guide to guide you through the main steps that we typically take when we design our training. And these steps are on the slide. So you can see that we think quite thoroughly about the way that we do training. These are our steps just for workshops. Courses sometimes can be a little bit more complex, and of course degree programs a lot more complex than this. But this is our basic training workflow. And so we follow this for each and every course that we design. And what we've done is we've packaged that into a little, just a simple PDF document that also has a whole bunch of templates and little help guides that can help you do training in the way that we do it, if you're interested in that, of course. But we've made lots of resources available. Everything's been created under Creative Commons licenses. And so you're welcome to take it, adapt it, reuse it as you see fit. And so that's also something that we're really excited about. And all of these resources are loaded onto public repositories. Excuse me. The one that we use quite a lot is Ziva Hub. So Ziva Hub is the UCT instance of Figshare. So it's a local instance of Figshare. And so we use this quite a lot to upload our materials and to make it more findable. It attaches a DOI, it versions the materials. And so it's really quite lovely in that way. And we actually upload all of our training materials now onto Ziva Hub. And so if you went to Ziva Hub now and you search for HA Bionet, you'd actually see all of our older course materials that we've managed to curate so far available there as archives that contain workflows, slides, often videos, data sets. So just whatever you would need to actually be able to do that training, hopefully on your own. So that's been quite great. Now, in the interest of time, I'm not going to speak to the DSI Africa initiative in too much depth, but I just thought I would tell you a little bit about the DSI Africa initiative. It's quite sad for me to say that HA Bionet is actually soon coming to an end. We are ending our funding cycle. We've been funded for, I think, more than 10 years now. So it's time for something new, I guess. And so we've been funded by this new NIH DSI Africa initiative. And basically, the whole idea behind this initiative is that infrastructure and capacity development are a big focus, of course, of boosting health research on the continent. And so the work will be facilitated through 5-year grants that have been awarded by the NIH, by the Common Fund, which is called Harnessing Data Science for Health Discovery and Innovation in Africa. And they've awarded actually in the region of $75 million to various research hubs that are in various places and are focusing on different things. So some research hubs are obviously data intensive, so focusing on the science, but then there are also training groups that are focusing on increasing just capacity for analyses. And there are also things like LC research groups, so groups exploring the ethical, legal, and social implications. And then there's the Open Data Science Platform and Coordination Center, which is the group that I'll form a part of going forward. And really, this group just wants to facilitate the development of a trans-African network of data scientists. So just to tell you just briefly about the ODSP will, in essence, develop a flexible, scalable, open data science platform for the DSI Africa Consortium to find and access data, select tools and workflows, run analyses, but very importantly, on a choice of computing environments, all through easy-to-use workspaces. And then administration and support of the core resources and consortium will be kind of coordinated by a coordinating center. And so those of us at the central HAI Bionet team, a lot of us will be forming part of that coordinating center going forward. But together with—
Please, can you just round up quickly so that we're going to have the next panel? I think we just have 10 more minutes for the African session. So please continue.
Sure, I thought I had 30 minutes, but no problem.
All right.
Okay, sure. So let me just actually wrap up then. So this is just a quick snapshot of what that platform is going to look like. We'll have all these workspaces where you can basically choose how you want to run, what you want to run. And it's going to be federated, so you can either bring the data to the tools or the tools to the data. And so we're very excited about this whole initiative and where it might lead African science. And I'll stop there.
Oh, great. Thank you so much. I believe that there are some questions that are coming in, and I will be calling on you as soon as possible so that you could help us with that. So a quick question, Dr. Binera Raz, are you there?
Yes, I'm here.
Okay, so the question that just got to me now says that is there any opening presently for an undergraduate student, maybe a postgraduate student that wants to join the program, maybe from somewhere from West Africa? Do they still have the chance to be trained by H3 Bionet? Yeah, that's the question.
Yeah, so definitely. So HA Bionet will still be continuing for about another year and a half. So I think until the end of next year. But we are also working on sustainability funding where, some things like our core training and things like that can also, just put my video on. So we are working on some sustainability funding that we hope will help keep some of the core services going, some of which will be training, but a lot of our training is actually going to transition over to the ODSP. And so we'll still have the opportunity for many scientists and students to join us for training, definitely, yeah.
Yeah, thank you so much. So for those that are just joining us, you have been speaking with Dr. Binera Raz. She's a training and outreach coordinator with H3 Bionet. She hails from the University of
Hello, can we hear you?
Activator, the person of Adekunle Adeluoye, which is about medical scientists, for her to invite to the stage our panelists. I believe that they are on ground. Please, am I audible? Can you hear me? Am I audible? Can you hear me?
Yes, we can hear you now.
Okay, so I'd love to call on Mr. Adekunle Adeluoye, which is a biomedical scientist, and our program director, OmicsLogic Africa, for him to take on the spot for the panel discussion. Please note that that you have 17 minutes for this section, so that the US team can come around and take over. So over to you, Dr. Adekunle. Please, I would love to see the faces of our distinguished panelists.
All right, thank you very much.
Will you be sharing your screen, or I should just?
Okay, just let me share my screen, please.
All right, all right.
Okay. Okay, allow me to go to the presentation mode.
All right, I bring an apologies from Dr. Volke, just coming in right now. The internet is not stable, so she may not be able to join us as I mean, like right now the message is just coming in, so like she's able to hear and not hear. So I think we have somebody short of the panel discussion. Yeah, please carry on, sir.
For the introduction. So can you hear me, please? Can you hear me? Hello, can you hear me?
Yes, we can.
Oh, great. So thank you very much. Quickly moving on to the panel discussion, I have with me our erudite professor, Dr. Charles Oluwaseun Adetunji, whom we listened to earlier on, giving us an overview of researches across Africa and in the entire globe, particularly utilizing bioinformatic tools. He is a distinguished professor of microbiology and currently the acting dean of Faculty of Science at Edo State University. So it's my pleasure having you on board. Dr. Charles, are you there? Hello, are you there? Hello, can you hear me?
Yeah, please carry on. Once it's around, we will hear from him. Okay, great. All right. We just got a message from the coordinator regarding Dr. Voketoye. However, hopes she's able to join us afterwards. But I'll quickly move on to introducing Dr. Elijah Kolawole Oladipo, who is the acting head of department for Department of Microbiology at Adeleke University here in Nigeria. And he is also the principal investigator and lead partner for Ellis Biogen. Dr. Elijah, you're welcome on board. Are you there? Dr. Elijah, are you there with me? Hello, Dr. Elijah. Dr. Elijah, are you there? Okay, sorry. He's just writing me that he's not able to talk because the host is disabling him from talking.
Oh, sorry. Let me see to that. All right. Could you please help with that to activate him? Okay. Hello. His audio. Oh, okay, great, Dr. Elijah. Pleased to really have you here.
Okay, this is Professor. I think Dr. Elijah is going to join very soon.
Oh, yeah, thank you. Professor Charles, you're welcome. You're welcome.
Yeah, thank you. Thank you very much.
Great. So we hope Dr. Elijah also joins us. And of course,
I think Dr. Walker is around. She will be unmuted in a bit once her network is all right. Yeah, continue, sir.
Okay, so thank you. We just heard from the leader of the HRBL Net, the coordinator, the training and outreach coordinator. Points, I would like to welcome on board Dr. Werner Ras. Are you there, Dr. Werner Ras? Can you hear me?
Yeah, I'm here.
Oh, great. Thank you very much. So what we hope to have Dr. Volke as well as Dr. Elijah join us during the course of this discourse. I think it will be...
I'm around now.
Oh, great, Dr. Elijah. Good to hear your voice. How are you doing today?
I'm fine.
Great, great. So we want to continue with this panel discussion now. Of course, as we can see on the screen, we want to essentially discuss around these thematic areas, starting with bioinformatics education and training in Africa. So I remember very well. Sorry, my internet is also, I hope you can hear me. Can you hear me, please? Hello, am I audible?
Yeah.
Yeah, you're audible.
Okay, great. So looking back into about 2 decades ago, of course, when we look at bioinformatics education and training in Africa, we know that at some point we needed to see most of experts from America, Europe coming to Africa to, essentially help, our researchers in the early days. But we've come a long while, starting from the formation of the H3A Africa, beginning with the H3A Bionet project that started back as a consortium in 2003. We've come a long way. So looking back, I'd like to start with the first question, talking about bioinformatics education and training in Africa. How far have we come? Where are we at the moment? And I think I would like to start by asking Professor Charles Adetunji to take on that question. Bioinformatics education and training in Africa, how far have we come?
Yeah, thank you very much. Actually, we are really doing fine, but we still need a lot of sensitization and awareness. And not that people are not doing much, but much is not yet being done. How do I mean? We still need to revisit our curriculum. Yeah, whereby we need a lot of awareness and sensitization starting from the younger generation. Now, so many people that have exposure to visit many labs are doing bioinformatics, but I can battle with you. I showed some picture in my presentation, some primary school students, secondary school students, and tertiary. So the awareness is just at the top, to the bottom top approach. So not everybody knows bioinformatics. And the most challenging thing is just that we have the bioinformatics around us, but many people don't even know that is what is called bioinformatics. Some people live even every day. So not understanding the basic of the bioinformatics is a problem. So in general, the training is good, the education have been spawned, but the awareness and the sensitization is very low.
Okay.
Thank you very much.
Thank you for that. I think from that response, we can see, a kind of a dichotomy between education and the training part. So essentially, if a key takeaway from what you said is that the trainings are there. There's a lot of work going on. Dr. Venera spoke to us quite a lot regarding several efforts at training, which has come to play in recent times. But then, essentially, it seems there is a gap when it comes to the educational aspect.
Yeah.
Of course, education includes the awareness. You can imagine even there are undergraduate or even postgraduate students, researchers who are getting to know or hearing about bioinformatics for the first time. Anyway, I'd like to move on. I would want to pass that same question now to Dr. Elijah Oladipo, sorry, permit me. All right, so Dr. Elijah, from your own perspective, having been involved also at creating opportunities to bring trainings to and bioinformatics education to the populace, to researchers and scientists, particularly students. How well do you think we have fared? From this, coming in from this angle, training perspectives.
Yeah, I think we are not doing bad, and there is always room for improvement. And I can say that what I am particularly interested in now is the quality of the training, because I surf the internet a lot these days, and you see a lot of trainings coming up from individuals that they don't know what they are doing in bioinformatics. And so, which is just a major problem. Like from our own angle, we are trying to make the bioinformatics so cheap and coming down to the grassroots. And I thank God Verena is on this call. I've been hosting for E3 Bionet since I was lecturing at Adeleke University, and the institute that I founded too, we have been trying to do something like this. But I'm very, very sure that we have a lot of ground to cover, like comparing ourselves to the Western world, like what my collaborator on the platform has. So we still have a lot of ground to cover. Though we are not doing bad, we are improving. That's why they are, even from the E3 Bionet, the numbers of classroom is increasing and the number of participants increasing. And even from our own side, to the level of training we are doing, we see many people are doing it. It's only that we have some challenges that is facing us in Africa, like the issue of internet. Even you, when you are presenting now, there was a time your internet was freezing, and those are major things that, bioinformatics is dependent on internet. And I'm very proud for Verena in the last part of his presentation, having taken about open source tools that can be used for people coming from Africa. There are some other tools that even if you are from Africa, your IP address does not allow you to access, because and those things are affecting the rate at which we are moving. So we are looking forward to having a platform that Africans will be able to have access to, so that the little things they are being taught, they can go on their own and redevelop themselves more. And another challenge that we have, even me personally, I'm facing the challenge as a life scientist. I graduated from a technology school that I did computer programming at 100 level and 200 level. You discover that at high pace of computational level, you need some programming language. And most of these life sciences, this programming is not included in their curriculum. So some we tend working with only GUI, and when it comes to coding, they have to have a lot of ethic. So we have to look into that aspect too, in how to train students at early stage of their life. And I'm happy to tell you that we are taking that step in Helix Biogen Institute. Like one of our research assistants, he won a grant from Open Science for taking bioinformatics to secondary school. That's high school level, in order to sensitize them about what bioinformatics can do and what genomics can do. So in the nutshell, we are not doing bad, but we still have a long way to go in order to fill in the gap that we have within Africa and the Western world.
Thank you very much.
Sorry for interrupting. We just have 3 more minutes, so I will be throwing the last question to Venera Raz. We have 3 more minutes for the next session startup. The next question I'm going to be throwing to her at once, and if it is possible, others too can answer, is what are some obstacles to collaborations for various organizations, universities, and citizen scientists in the sphere of bioinformatics in Africa? So that question is going to you, Venera Raz.
So, sorry, before you come on, let me add this. I think Professor already mentioned that, and it came up also during Dr. Elijah's response. So please, can you please share with us also what you think will be done in terms of the curriculum development for bioinformatics, particularly for our various universities, now that we are taking these projects to primary, secondary schools to get them aware. Unfortunately, many of them cannot come into the universities to have these trainings or exposures to bioinformatics because of the lacuna that is in existence in our curriculum. So what do you think has been the experience with you interfacing or conducting the E3 Bionet project? Over to you, Dr. Vera.
Sure. So I want to start by saying that HA Bionet has been working quite diligently with a lot of institutions across Africa to actually do curriculum development. And I mentioned competencies because we've actually fed that into the curriculum development as well. So we've developed a lot of predominantly postgraduate curriculums, but we have a few groups now actually working on implementing undergraduate curriculums, and we're also trying to do that locally. Whether we can implement a whole curriculum or not is a task of its own, but at the very least, introducing some formal modules in bioinformatics at the undergraduate level, we're really pushing for that, because I agree with Dr. Aladipo that it is vital that we reach everybody at various levels, and Professor Charles brought that up as well. And I think a big obstacle in Africa is, like everybody has mentioned, it's just access to high quality training from experts. And I think a big obstacle as well is when I interact with people and I talk to them about bioinformatics, it's almost like they are shy to speak to you because they feel that their knowledge isn't, good enough to even engage with the content. And so how do you change that perspective? How do you change that perspective of, I can't actually do this work, I don't know enough. How do you help them know enough? And so that is the sort of angle that I think we come from in terms of training, is building up that pedagogy of, understanding, of knowledge, of ability. And so that's one way that we've been working on combating some of those obstacles. We also use our training sort of with the dual purpose of also forming a community. So we try and connect not just students with each other, but researchers with each other. So a lot of our participants are former researchers and scientists. And so we use that training as a mechanism actually to start forming a community. And we try and get them to connect, get them to collaborate, continue working. But I think a lot of people think that they can't do bioinformatics, or that bioinformatics always has to take a lot of money or has to have a lot of skill and expertise. And yes, it does, but there are things that you can do to get yourself there. You can start very small and grow as a lab, for example. There are things that you can do. And so I think that's been a big hindrance, is just the willingness of people to work on bioinformatics because they just feel like they don't have the resources. But then resources themselves is a problem. Infrastructure, a lot of the time. Sometimes infrastructure is so expensive. Internet is shockingly expensive in Africa. And so how are you gonna be able to compete with other institutions who have that money available to them? How do you make yourself competitive for grants? And so that's been a major focus of HA Bionet as well, is developing that capacity, developing that collaborations of going, okay, I don't have the bioinformatics expertise, but I've got the data. You've got the bioinformatics expertise. So let's come together and we submit that grant and we both get awarded and we both develop as a result of that. And so I'll allow some other people to come in there so I don't monopolize all of the time, but I think there is a lot of work that needs to be done. But I think as a continent, we have come a long way in a very short time, and I'm very excited about where we're going to go in the next 10 years.
Oh, great. Thank you very much. Of course, it's time to move on. I want to apologize to our panelists. We do not have enough time to go into this, but please, in few words, as a closing, comment, what do you think are those strategies to involve students more, especially from the training perspectives? We've seen students come on to some of these programs, and some of them, despite they've paid or join some of these free programs, they get to drop down the line, maybe because something is becoming too complex for them. So what are the strategies do you think we can use to get students involved the more in bioinformatics? In few words, please. Let me start from Dr. Veneras, quickly.
Yeah, I think it's just sort of changing the ethos of what you believe a bioinformatician looks like. A bioinformatician looks like many of us. And so I think for me, that's, that's for me, the perspective I come from is that anybody can do anything if they train properly. And so I live by that across the board.
Thank you for that. So I'll move on to Professor Adetiji to have a say into that. How do we get more students involved, particularly at the very early stage into bioinformatics? Are you there, Prof? Okay, let me move on quickly to Dr. Elijah. Dr. Elijah, what do you think?
Well, the only thing I can say is proper mentoring. So if the students can be well mentored, and I believe that they're going to be the change of the game. And though mentoring is not easy, it's a great task, but if they can see older scientists to mentor them, to guide them to the process, even the process that we felt is very hard, you're going to see that it's very simple. Just show them the way and stay with them, then they get to the good destination.
Thank you very much. Thank you very much. Is Professor Adetiji there?
Yeah, actually, I want to go in line with what the coordinator for I3 Biomet have just said. Also, we need to synthesize from grassroots. Now, we can create a program, talk to your professor, talk to your scientists, create a kind of club within university, catch them young initiative. That is going to go a long way. Then the government needs to key into what we are doing. The policymaker, the industry, the academia, that is when the whole thing is going to go a long way. Thank you very much.
Thank you very much. On that note, yes, I want to just give a wrapping thought on most of what we've said. I love the way Dr. Venera started by talking about making bioinformatics as simple as, what we do, everything, every day, what most of these students have already started to learn, and seeing it as something they can build up their skill sets in that domain to further advance their scientific practice. So yes, that is very important. And I love what also Dr. Elijah said, talking about, mentoring. Of course, I'm glad to say that this is very much available also with the Pine Biotech platform, the OmicsLogic programs. Mentoring, we take it very, 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 we play a lot of importance on mentoring. And so it's a key thing we need to do, because I believe once the students see themselves making this progress, they are kind of inspired to go further down, and they become a good example for other colleagues who are also showing interest in bioinformatics. So that is very key. And thank you, Prof. A question I was hoping I would have opportunity to ask you was how we can engage more of our dons in our universities, in our educational system here in Africa, to get them interested and have a buy-in into bioinformatics, because definitely one of the missing pieces I would say that we are having at the moment is with our government and our management, even at the university level. Some of our management, our vice chancellors, they are drawing back when we have few researchers and academia pushing for involvement and inclusion of bioinformatics in the training curriculum and programs. So I want to thank you. I hope we'll have opportunity to do more discussions around this subsequently at other times. And on that note, I will want to hand over back to the US team now. I believe we've taken...
I will be calling on Sonalika, but before I call on her, I'd love to appreciate everyone that was present here, from the professor to Venerable Raj to Mr. Adekunle and the OmicsLogic African team. You have been very, very wonderful. Quick feedback said that thanks for the nice presentation. We hope we have more of these. So thank you very much, everybody. You have been so lovely. Sonalika, I just saw you. Over to you.
Yes, thank you so much, Eonfe, and also everyone from the OmicsLogic Africa team for having these wonderful sessions and talks, and also very interesting panel of questions you all had. So after having two wonderful, amazing sessions, first by OmicsLogic Asia and then by OmicsLogic Africa, now we are all set to start with the talks by OmicsLogic USA. Now I would like to call on stage Mr. Ilya Brodsky, CEO Pine Biotech. Mr. Ilya Brodsky is one of the co-founders and CEO of Pine Biotech. He has facilitated and managed collaborators with researchers at Stanford, UCSF, Louisiana State University, Tulane, and other academic and pharma R&D teams. Mr. Brodsky oversees the research projects and manages the development of the prototype, preparation of the relevant data projects, and implementations of the systems for the needs of students and teachers that the project is designed for. Over to you, Mr. Ilya.
Thank you very much, Sonalika, and thank you everyone for joining this session. So I really enjoyed the conversation so far. It was really exciting to hear about different initiatives in India and Asia in general and in Africa. I think there's a lot of great initiatives kind of taking place. And especially, I would say it was exciting to hear the presentations by research fellows and students that worked on independent projects. And I think that's really the reason that we're all here. We want to see not just, teaching bioinformatics, but use of bioinformatics. And we want the students to be successful pursuing their careers, both in research and in industry. And so as a company, we are very in tune with the need and the growing appreciation for the bioinformatics skills because I think that as much as research has benefited from bioinformatics and developments in bioinformatics, we can definitely see how translational bioinformatics that is able to take the findings from research projects and translate them into products that people use, into new medicines, biotechnology, and others, that really, I think, has been illustrated by this pandemic. So what is always amazing to me is how before the pandemic, you would ask someone to explain what a mutation is or what a variant is and what is sequencing. And very few people would actually be able to clearly identify these terms. And so today, because of media, because of television, because of just awareness of what everyone had to deal with close to them, I think 95% of the people would be able to accurately identify what these things are. And so as a result, I think that we are seeing how awareness about what bioinformatics is and the need for bioinformatics is growing. The industry is able to leverage these methods and build infrastructure to make it more effective. And so I think what we are excited to do is kind of try to link all of these pieces together. Our goal is to work with some of the people that we've seen up until now to learn from these initiatives that have been successful and to try to develop a scalable infrastructure so that this kind of training can be available to as many people as possible, including people that don't know what bioinformatics is. I think that's one of the major objectives here. So again, thank you for all of the panel members, for the speakers that have participated up until now. I really enjoyed the talks. Thank you for all the students and the presenters that presented their projects. I think really exciting work that they have done. I would like to now pass the presentation on to Laura Murray. Laura has joined us recently as a bioinformatics program coordinator. She has her bachelor's degree in biology, in microbiology, and she then worked at a cancer pathology lab, really seeing how diagnostics happens in cancer, and then decided to pursue her degree in bioinformatics. And I think what she is trying to help us with and what she will be speaking about is kind of where we are going with the OmicsLogic training and how the resources and the different types of aspects of learning, like education, mentorship, implementation of modules about bioinformatics in non-bioinformatics curriculum is what we're going to be doing in the next 6 to 12 months. So Laura, I'll pass it on to you, and thank you again for everyone for joining.
Thank you, Elia. Let me just share my screen. Okay, can everyone see my screen?
Yes, we can.
Great. All right, so welcome to an introduction to OmicsLogic. A little bit about me. So my name is Laura Murray. As Elia mentioned, I am the Educational Program Coordinator here at Pine Biotech. I have a background in gaining my bachelor's degree in microbiology, and I'm currently finishing up my master's degree in biology and bioinformatics. My focus and areas of expertise are in metagenomics, transcriptomics, and genomics. And my most recent research was in analyzing the metagenomics of deep sea hydrothermal vents and which bacterial communities reside there. So in this presentation, I will explain the current challenges and limitations in bioinformatics. I will describe how we aim to address these challenges with the OmicsLogic portal. I will describe Bloom's taxonomy for learning progression and how it can be used in bioinformatics coursework. We will explore the OmicsLogic portal and how it utilizes Bloom's taxonomy in structuring its courses, pipelines, and projects. And finally, I will describe several outcomes of the OmicsLogic portal. So what are some of the challenges that the field of bioinformatics is currently facing? To begin, this field is constantly growing and developing new tech and software to improve processing time and power. The data sets that are analyzed are also usually very large and take immense amounts of computing power and memory that many personal computers just don't have. Also, despite being publicly available, data are also usually complex to access, and coding knowledge is usually required to do so. Similarly, analysis pipelines and programs require coding knowledge that many biologists don't have. In a university setting, students have the opportunity to learn the background information of designing and implementing a bioinformatics project, but don't receive practical hands-on experience with the full workflow due to the limitations mentioned previously. Similarly, faculty faces challenges with being able to cover a diverse range of topics, especially if they are specialized in a certain area of bioinformatics, or if they are unfamiliar with bioinformatics in general. For example, a professor whose research focuses on oncogene expression might not know much about the metagenomics of seawater. So in order to overcome these challenges, the OmicsLogic portal was developed. This program aims to standardize, organize, and systemize bioinformatics training in order to make these analyses usable for any biologist or clinician. There are some traditional problems with the standardization of bioinformatics coursework. Materials for courses are usually developed in-house, taught sporadically, and are rarely updated. The OmicsLogic portal introduces new material that keeps up with new technology. These courses also need to be organized. Because the challenge is to combine several different topics together, the flow and transitions between subjects needs to be developed. This is addressed at OmicsLogic through introductory material being woven throughout the course. The last challenge we face is systemization. How can you know that someone is stuck and needs help, or allow someone to run forward? To address this, OmicsLogic uses standardized modules and interactive sessions, beginning with mastering terminology and concepts to implementing these concepts with real data sets in a project-based approach. So here's an overview of the learning process. So in order to create a clear path of skill development, OmicsLogic employs the Bloom's taxonomy levels of learning. This approach starts with the basics. Students recall facts and terminology and are able to repeat and memorize the material. With this base, the learning builds to conceptual knowledge, being able to understand concepts and apply information to new situations. The student will then be able to analyze data by drawing connections and evaluate when to use certain methods. This leads them to the highest level of learning, the ability to design and create original work. So how do OmicsLogic resources help advance bioinformatics teaching? OmicsLogic is a portal for practical and theoretical learning of bioinformatics. Composed of courses designed to take you through theoretical knowledge, OmicsLogic takes a combination of training modules, data analysis tools, curated project datasets, and interactive sessions with mentors to give a student a clear path up the Bloom's taxonomy pyramid. No matter what the user's initial experience with bioinformatics is, OmicsLogic is a useful tool for gaining a baseline knowledge of the theory behind bioinformatics, analysis of large datasets, an introduction to basic coding languages like R and Python, or beginning a data analysis project. So these 6 courses were taken from the core OmicsLogic curriculum available online. Beginning with an introduction to the applications of bioinformatics and an overview of data types, the student is able to learn terminology and factual knowledge to build upon. From there, genomics, transcriptomics, metagenomics, epigenomics, and data science are introduced. Each course starts with important terms and concepts and then introduces case studies and pipelines related to each one. This allows the student to gain the conceptual knowledge in order to begin their own projects. So OmicsLogic also expands upon its basic core curriculum with specialization track projects. Bioinformatics methods are best learned in the context of real case studies prepared by our network of mentors and collaborators that illustrate how various bioinformatics methods and omics data types are used in current research, both in medicine and in biotechnology, agriculture, and even astrobiology. The projects are prepared by taking publicly available data sets and using them to explore and understand both the research challenge and the analysis path to extract meaningful insights from complex omics data. The OmicsLogic portal gives students options for analyzing a given data set, either a code-dependent path or a code-independent path. These datasets and associated publications can be used in several ways. So like I mentioned, you can learn bioinformatics without coding at any coding experience at all. For that, in each project, we explain the analysis pipelines on the user-friendly Bioinfo platform for big data analysis. You can also learn the scripts behind many of the steps, learning to wrangle, clean, and visualize and analyze data in popular languages like R and Python in order to create your own analyses. Finally, you can hear about the projects and their research applications in our mentor-guided programs where experts share their experience and support research projects developed by you. So here's an example of the code-independent pipeline. The pipeline builder interface on the Bioinfo platform is a color-coded pipeline that helps establish a logical connection between the technical names of data analysis methods and the logical steps needed to build the full pipeline. Data is put in as an input file, and then analyses can be chosen based off of the type of research. Each analysis is separated into 3 colors: blue for pre-processing, red for mapping, and green for quantification. Each time you click a button, a pop-up will contain additional information useful for building the pipeline. The ease of the color-coded buttons allows programs to be run without any coding knowledge. Interactive outputs allow for easy visualization of the data. Several demo pipelines with real data sets are available for students to practice the workflow, including demos on genomic variant calling of cancer cell lines, 16S amplicon analysis on diet and its effects on the microbiome, and a pipeline for examining a phylogenetic analysis of Ebola virus. So after using the Bioinfo platform to visualize the pipelines and practice analyzing, students are then able to take their theoretical knowledge, the remember and understand level of Bloom's, and apply that in a project, the apply, analyze, and evaluate level of Bloom's. Then students are able to check their comprehension with levels and badges, certificates of completion, and tests. Similarly, faculty should be able to assign the OmicsLogic portal to their students with the understanding that it will give them holistic and comprehensive knowledge of bioinformatics, beginning with the Bytes and Molecules course and on to designing their own question-based project. So OmicsLogic employs a hybrid approach, combining learning and research. To ensure that students are building off of the first level of Bloom's taxonomy, OmicsLogic tracks progress with activities like pipelines, coding, and literature reviews, quizzes on topics, key terms and important questions, and covers a broad range of topics like biomedical research, biotechnology, and agriculture. Then, students are able to continue their knowledge base with a research assessment, making sure they are able to understand methods, challenges, and results, as well as being able to comprehend primary research literature and recreate the methodology. They should be able to ask questions based on data exploration. This leads to the ability to encounter errors in their analysis, apply theoretical knowledge of why those errors are occurring, and find a solution. Bioinformatics uniquely combines life science terms with data science skills, which allows researchers to answer life science questions with reproducible data analysis methods. So OmicsLogic Research Fellows are able to ask research questions about any bioinformatics topic, leading to a diverse range of projects. From analyzing affected biological pathways in multiple sclerosis to brain metastases due to small cell lung cancer, these students are able to apply what they've learned in the coursework, work with a group of professional mentors, and submit their projects on the portal for review. So students should then be able to reach the highest level of Bloom's taxonomy, create. This is achieved through the creation of their own analysis project. For example, Semih Dolaniye, an undergraduate researcher at Bahçeşehir University in Istanbul, Turkey, who finished her OmicsLogic training, was able to work on a project involving chronic lymphocytic leukemia, a type of blood cancer that has a very heterogeneous biological background and diverse treatment strategies. From her work with OmicsLogic, Semih's work was able to be published with the Columbia Undergraduate Science Journal. Here at first, she elucidated 870 significantly differentially expressed protein-coding genes that were involved in the biogenesis and processing of RNA. Consequently, these findings led our study to investigate non-coding RNA. 33 long non-coding RNAs were found to be significantly differentially expressed among 2 conditions based on differential gene expression analysis. So not only does OmicsLogic collaborate with undergraduate students, as seen with Sime, but projects with graduate students and other researchers are common as well. Here is a poster presentation for a project done in collaboration between Noguchi Memorial Institute for Medical Research and Pine Biotech. Here's our team at Pine Biotech who are involved with the project: Dr. Mohit Mazumdar, Dr. Harpreet Kaur, and Elia Brodsky, as well as our collaborators at the University of Ghana, Dr. Anita Gansa and Dr. Benedicta Mensah. This poster was on Plasmodium falciparum, the parasite causing malaria, and its apical membrane antigen, the Roptree neck tube protein binding, which is a critical interaction for Plasmodium falciparum merozoa invasion of the human host red blood cells. Several proposed malaria vaccine candidates are designed to prevent such binding and limit merozoa entry, which have been studied with variable success at in vitro and early clinical trial stages. By performing haplotype network analysis of genomic sequencing data from sites with variable infection rates, comparing our data with malaria gen repository from another region in Ghana, and mapping genomic variants onto the protein structures, they arrived at preliminary conclusions about local variation and immunogenic epitope affecting variants for consideration. In this poster, preliminary findings were shared that suggest that genomic data can be used to develop a local strategy for efficacious vaccine design against P. falciparum blood stage infection in Ghana and surrounding regions. So OmicsLogic is proud to collaborate with many diverse programs across the globe. We also work with over 15,000 students at community college like Glendale Community College, top-tier universities like Georgetown University, as well as 20 universities around the world. OmicsLogic is also proud to partner with NAPI, the National Association of Idea Principal Investigators. NAPI seeks to elevate bioinformatics research in areas of the US that are traditionally underfunded, for example, in Puerto Rico, Louisiana, and West Virginia. NAPI puts an emphasis on inclusion, making sure that there are as many people have the opportunity to get involved with bioinformatics research as possible. OmicsLogic is proud to mirror that emphasis of inclusion, with adding many free course options, giving students asynchronous resources to complete whenever their busy schedule allows, and pairing them with professional mentors who can offer guidance in getting involved in the field of bioinformatics. So in this presentation, I explained a bit about the current challenges and limitations in bioinformatics. I described how we aim to address these challenges with the OmicsLogic platform. We discussed Bloom's taxonomy for learning progression and how it can be used in bioinformatics coursework. We also explored the OmicsLogic program fundamentals and how it utilizes Bloom's taxonomy in structuring its courses, pipelines, and projects. And we finally described several outcomes of the OmicsLogic program. So if you're wanting to know how to get started, the OmicsLogic portal is easy and free for anyone to register, with many courses available at no cost. There are also several upcoming programs that, as a student, you can get involved in. This is a program for data science for biomedical data. There's also one for genomic data analysis, and one on an introduction to big data bioinformatics. As a member of faculty, please reach out to us about learning more on integrating OmicsLogic courses into your own bioinformatics curriculum. So thank you all. Are there any questions?
Thank you, Laura. And I think the link was pasted also in the chat for anyone who wants to visit. I think for the sake of time, we're going to move on quickly. So again, thank you for this overview. I want to briefly introduce our next speaker and kind of speak about what are the messages that we're going to address in the next few sessions that we hear. So the next presenter is Dr. Sona Vasudevan. Sona, are you with us today? Yes, you are. Excellent. So great to see you and welcome. Dr. Vasudevan has a broad range of research interests, but primarily she is a director of a program called the Systems Medicine program at Georgetown University Medical Center. And we're excited to have her here because I think that she brings a unique insight into the role of information technologies in medicine, She deals with a broad cohort of international students. So any one of you that is really interested can benefit from understanding why she started this program. What does she teach her students? How is it done? How potentially you might want to register for a program like that? And where do students that finish that program go after they complete the training? I also want to introduce briefly a couple of speakers that will be picking up from the topic that Dr. Vasuban will be sharing on. The next speaker after that will be Alice Kamens. She's a strategic projects and program manager at Google Cloud, and she will speak about some of the strategic initiatives that they are undertaking at Google to make various training resources to be available to those who are interested to incorporate cloud into their skill set. Then we will hear from Dr. Adelaide Rhodes. She's a principal computational biologist at a company called Cultivarium, and she will speak about bioinformatics as a career option. So many of us are interested in research, but there's also the industry path, and so she will address that from her own personal experience. We will then go on to hear from several accomplished speakers from South America. And those will include Dr. Georgios Papas from Brazil, Dr. Gabriel Gatti, who is originally also from Brazil, but now works at Camp4 Therapeutics in the US, Mr. Silver Ceballos, who is in Costa Rica, Mr. Piero Gasco, who is in Peru. And we will tie all of these topics together in a group discussion where we have Dr. Raja, Gio Chavez, and Ms. Maria Victoria Luz, who will speak about some of these opportunities both in the US and in South America. So, Sona, I will unmute you now. Sorry about that. Okay, I believe you should be able to unmute yourself. Yeah.
Okay. You can all hear me, correct?
Yeah, we can.
Is the volume good? Okay, good. Thank you, Ilya. Thank you so much for the wonderful introduction, and thank you all the participants who are here. I think the previous sessions have been amazing, and I think most of the introduction and the foundations for my talk has been laid out pretty well, which is that everyone here probably will agree that bioinformatics is extremely important, and there's so much data, and there are lots and lots of challenges that all of us are facing. And what I'm going to be talking to you with in the next 20 minutes or so is to tell you the biggest challenge right now, in addition to all the data and processing of the data and everything else, we need to train the next generation. And in fact, we have to train ourselves on how to handle this big data and how are we going to mine meaningful information from all of this data and create meaningful resources for the next generation of students from different disciplines. And of course, Fine Biotechnology is leading in that effort, providing different resources and courses and everything else. So I'm going to share my screen here. Okay, I hope you can see my screen.
Yeah, I think you need to switch the screens. We can see.
Okay.
Presenter.
Okay, give me 1 second. Give me 1 second, slideshow. Is that better?
Your screen is not shown right now.
Oh, it's not shown. Okay, sorry.
Yep, now it's great.
Yeah, that's okay. Thank you. Complications with So many different monitors here. Okay, so today I'm going to talk to you about systems medicine, a new and evolving emerging discipline that we created at Georgetown. More importantly, I'm going to focus on how, why, and whom should we teach systems medicine to. So first, I'm sure all of you are wondering, what is systems medicine? Because it's not become a lingo that's commonly used by a lot of people, and it's getting popular right now. So systems medicine, to give a formal definition of it, it's a novel approach to medicine. It is the first step on the path to personalized medicine. And I'm sure living through the pandemic, all of us realized that each of us have different symptoms, different reactions to vaccines and therapeutics, and wondering why even within a whole similar household, we have different reactions to all of these different things. So systems medicine is based on computer models in which vast amounts of clinical data is used to analyze the health of individual patients. It also brings a humongous promise to further our understanding of diseases, mechanisms, and also boost the efficacy of treatments. And of course, at the end of the day, all of us want to cut costs of healthcare. And so it is actually one aspect of what we call precision medicine, practicing medicine precisely that reaches individual care. So practical definitions of systems medicine that all of us are very familiar with is it's a multidisciplinary field that looks at an individual as a whole. And it uses systems biology and computational sciences and the omics technologies to help solve biomedical problems. And of course, it is not necessarily the future, but it's also the current practice that all of us have realized that the summary of it is one size does not fit all. So I actually call this one size does not fit all medicine. So what does this mean? It means that, say, two patients who have similar symptoms, have similar findings or diagnosis, and the same disease, and given the same drug. But both these patients react differently to these drugs. So, of course, we all ponder why two patients with the same symptoms and same underlying everything conditions react differently to drugs. And of course, we all kind of know with all the data what the reasons behind this could be. So why does drug response vary among different populations of people, right? It's individual variation. Of course, it's all at the underlying genomic levels, genetic differences, single nucleotide polymorphisms that exist between two individuals, or single nucleotide variants that we call these single nucleotide changes. Of course, it also could be ethnicity, age, genetic factors, drug-drug interactions, and environment plays a big role. Of course, we are all now focusing our attention also to nutrition. And of course, also the focus has been on a microbiome. I'm sure everyone in this audience knows the importance of microbiome and the fascinating concepts and realization we have come to today, that we have more genes from these microorganisms that exist in each part of our body than our own genes as well. So they are actually taking more control over all of this. So there are different factors that are actually responsible for individual variation that exists. So why are we talking about all of this right now? And the reason, of course, we're all talking about all of this is, of course, we are in the midst of an era of big biological data. And I just put the pandemic in parentheses with the hope that we are going to say goodbye to it, hopefully soon. And so I don't want to feel like we're still living in the middle of this. But of course, the pandemic has also caused a lot of realization for us, and also generated a huge amount of data for us to a mine to understand these individual variations, specifically with respect to the disease COVID-19, and a revolution in medical practice in the 21st century. And of course, we can't forget where it all started. It all started with the Human Genome Project almost 17 to 18 years ago. And of course, at that point, all of us celebrated that we are going to say goodbye to diseases. And we also thought that we all understand every single diseases, because we understood how many genes the Human Genome Project generated, how many genes is there within all of us. So understanding all of those genes, we thought that we could say goodbye to diseases. But as you all may realize, after 18 years, we still realize that that's not the case. And now we know that lots of factors that are important in understanding all of these things, which I outlined in my previous slide. And so there's lots of data to understand specifically how genome, how can we go from genotype to phenotype, understanding all of that. We have different layers of omics and lots of data generated at every step of this ladder of omics, and also microbiomics. And also I just checked Wikipedia, and I think we have more than 200 or 300 different omic terms now. And this is something that I would do with my students. I walk around the class and ask every single student to come up with an omic term, and we have 4 or 5 times more omics now. So which means each one of those omics is generating a huge amount of data. And now we all have to figure out how to mine these data and how to generate the next generation of bioinformatician who can actually understand all of this, and to come up with a simplistic view of understanding how we can go from genotype to phenotype. So genomics, of course, is being used to integrate pathways, mutated genes, and drug-drug interactions for various different diseases. And genomics is also is a tool, a diagnostic tool now to understand molecular mechanisms of diseases. And all of these different omics terms has become a standard way of looking at everything in whatever discipline that you are working with. Of course, in the clinics, for physicians, they need to embody, they need to understand all of this data in order to be able to meaningfully treat their patients. And so the idea of precision needs to be there, and the idea of personalization needs to be there for the practice of medicine. So we also have realized systems medicine is already in clinics today. Big data used to learn about general population, to make clinical decisions in this and reproductive health, reproductive endocrinology, prospective parents, genetic testing before and during pregnancies, tumor profiling using genetics to tailor cancer treatments, by tracking mutations and traits of cancers in various tissues, and pharmacogenomics, of course, accessing an individual's perspective reaction to a drug based on their specific genetic makeup. Of course, this is actually already in use in the field of cancer, and we have come a long way. In fact, there are specific drugs that have been approved by the FDA to treat specific mutations that people may harbor in specific cancer types, for example, colon cancer, breast cancer, and all of those different things. So we have actually come a long way. And there is a revolution in medical practice. So now, instead of focusing more on curing diseases, we want to actually prevent diseases from happening, and also health promotion. So there is enough data out there. But the problem is, how do we mine this data? So we have to generate the next generation of scientists and biomedical informaticians who can actually understand this data. So this explosion of data has made us realize that we need educational programs to learn this new language. I call this a new language that all of us in any discipline needs to learn. And of course, it's an investment, and that probably pays the best interest as far as I'm concerned. So Georgetown became one of the leaders to create the first program in systems medicine, gave birth to a program, a dual degree program, MD-MS in systems medicine. It was successfully launched almost a decade ago in 2011. And the students are required to take 30 credits. Of course, it's a very rigorous curriculum. And it is specifically intended for students in medical school who have completed 2 years of their medical training. They take a year off, and then they get a master's degree in systems medicine, and they have a dual degree so that they understand the computational sciences, genomics, proteomics, and all of these omic terms, so that they can actually make an intelligent prognosis for their patients that they're going to see. So they will be a new type of physician who actually embrace the concept of all of these different omics. And of course, more importantly, systems approach to medicine, which is what we all have to learn. And of course, we also wanted to create a master's program, not necessarily for students in medical school, because this is applicable to any discipline in the sciences, in any discipline that people want to go to. So we created another master's program in 2016. And the program, again, the curriculum was intended for any student who is interested from any discipline of sciences, who's interested in pursuing a medical career, becoming a physician, or by becoming a biomedical scientist, who one with a grasp of modern computational sciences and omic technologies, so that they actually become the bridge to translate all of these discoveries from omic data analysis from bench to bedside. And of course, again, a systems approach to medicine and in biomedical research. So the curriculum of this program was not very trivial to create one that embodies different aspects that the students need to be trained in. And so it has different aspects. Biostatistics is important when you are actually talking about one single nucleotide polymorphism in a patient. We have to make sure that it is statistically significant before we actually come up with treatment modalities. And then biomedical informatics, this is specifically how do you mine electronic health records, electronic patient records, and all of those things to understand a pattern in a given population, maybe ethnic populations. And of course, at the end of the day, students need to learn journal articles and figure out how to interpret journal articles and understand a use of clinical data. And of course, microbiota, as I said, microbiome is actually very important, and genomic medicine. And how do you translate all of these discoveries from bench to bedside, which means translational. And then, of course, metabolomics is actually integral to systems medicine, because at the end of the day, anything that happens in the body in terms of cells and what they do is through a pathway, and the end of the day, the result of a pathway and reactions is metabolites. So it's very important to understand what these metabolites are doing and how do you mine these. Of course, the program has core courses that students need to learn, that they're trained in fundamental aspects, but also each student may choose to go into a different career path. There are students who want to become physicians. There are students who want to do a PhD. There are students who just want to do work in healthcare. There are students who just want to mine data and learn data analysis. So the electives are geared towards specific students' interests. As I said, systems medicine means precision medicine and personalized medicine. So all the curriculum is also personalized to each of these students. So if a student wants to do drug design, they can take a structural biology course that teaches them how to do drug design. Of course, if you want to get into policy, then they can take policy courses. And if they want to get into a lab, you can do lab courses. So all these electives are geared to a specific interest of these students. And if a student is interested in cancer, for example, they can take cancer. So all of this is tailored as the systems medicine program itself is personalized in terms of what it delivers. And the students can take other courses, epidemiology, microbiology, and emerging infection nutrition. And so the whole curriculum is tailored towards what the goals, career goals of each individual student is. Apart from the courses, one of the most important aspect of the curriculum is that every single student has a capstone or a practicum. They do a semester-long research that for most cases culminates in a publication. And they, of course, they present a poster and they're judged by judges in the field. And so this really drives the students' interest to carry what they learn in classes, in lectures, to actually applying it to a real scientific problem. So this really sums up the whole experience that each of these students actually has. The students work in a variety of different institutions, and we are very collaborative in nature, so they can work in national NIH, which is down the road from us, FDA, which is also down the road from us. Of course, students can also partner with other institutions. So students, these are all the institutions that students have actually done their internships in. Memorial Sloan Kettering in New York, and also students actually do internships in industry as well, in companies. Of course, to summarize the skill sets that the students learns from all of these is to actually analyze big data. They learn different aspects of next generation sequence analysis, RNA sequence analysis, single cell RNA sequence analysis, Journal Club. Thanks to Pine Biotechnology, they have offered an outstanding course to the curriculum on machine learning. And of course, students also know how to learn different aspects in the lab, and of course, microbiome. This is actually a summary of all of the things that the students learn. And there is actually something for everyone in terms of career. Student can go to medical school, nursing schools, PA or DO schools. They can pursue PhDs. They've got, some of the students have gone to industry, and of course, they can work in doctors' offices. And of course, the MD, MS students actually go and pursue and become physicians. And so there is something for everyone from the curriculum. And so where are the graduates now? So our graduates are in all different institutions across the US, pioneer institutions like Cornell, Sloan Kettering, and all of those things, University of Maryland, and all of those things. And then, of course, every student will ask, do we have scholarships to support? Of course, we do have. So we have an endowment called the Robert Stephen Ledley Endowment. We were given $100,000 for the program, and that generates some scholarships. And also we have other scholarships, all merit-based scholarships that can help you with your tuition, because Georgetown is expensive. Of course, it's not all about curriculum. The students also have lots of fun. We celebrate different festivals, and there's lots of conferences students attend. And at the end of the day, you can see smiling faces. So there's a rich curriculum, but also there's also socializing aspects to the program. We take field trips to NIH. Here, this is a field trip to look at the sequencing facilities. And then, of course, I want to summarize why Georgetown. The top 5 reasons to attend Georgetown: location. It's very, very pretty and beautiful here. And of course, traditions connect students from all campuses, past, present, and the future. And then we also have rich athletic history, as you all know, Hoyas, Saxa. And then, of course, we also have famous and influential people who have loved us in the past. And of course, you can be satisfied with minor things. Everyone should be should aim as big as possibly can, from Watson. That's his advice to all of us. And of course, our faculty and staff are incomparable. We have amazing faculty and who are always there. And of course, the program is first in the nation right now. And of course, you're all welcome to come right now to visit, because the cherry blossoms are in full blossom right now, and it is very beautiful here. If you want to learn more about the program, you can go to this website, systemsmedicine.georgetown.edu, or you can actually also email us at systemsmedicine@georgetown.edu. So I thank you all for your attention, and I'm very happy to take any questions you may have.
Thank you so much for your talk, Dr. Vasudan. We have a few questions for you.
Okay.
So we want to know who all are eligible to join the program there.
Okay, that's a fantastic question. Anyone who has an undergraduate degree in the sciences or even computer science majors are eligible for this program. And because every single course starts from basics, so you won't be lost in any of the programs. So we have actually had even one history major who has actually been in the program from our MDMS curriculum. So anyone with a basic interest in the sciences and has a little bit of background in sciences is eligible for the program.
Great, thank you. Another question we have was, so thinking back to when you were first learning and first introduced to the field of bioinformatics, what advice would you give yourself just starting out?
To figure, I mean, can you repeat that question? Let me get it.
Yeah, so if you're looking back, if you could go back in time and ask, give yourself advice when you were first starting out in bioinformatics, what would you tell yourself?
Okay, so I actually want to, currently today, when I started bioinformatics, it was an accident. After I finished my PhD in a lab, doing drug design and X-ray crystallography, and I'm a physicist by training, had no idea what even bioinformatics meant. I was accidentally put in a lab in NCBI, National Center for Biotechnology Information. So at that point in time, I did not realize the importance of bioinformatics. So when I turned my life back, I think that was the best accident that happened to me in my life. What I would have done is at that point in time, I would have gone and done a PhD in bioinformatics at that point in time. So I now am glad that that accident happened in my life because I can't imagine what I would have done without the training that I've actually had as I'm standing today, looking at the evolution in the field.
Great, thank you so much. Another question we have is, have you considered including some basic math courses in the program?
Yeah, so actually that is a very good question. And we actually do have, so the biostatistics course actually does cover a little bit of mathematical concepts in it. And in fact, all of the analysis that we do, all has to have a little bit of math. So we don't have specific math geared. I'm not sure what specific aspects that you're asking about. But most of the courses actually embrace mathematical concepts. So we do teach a little bit of mathematical concept across different courses, but not full-fledged math courses because of the student population that we actually have.
Great, thank you so much. I think that is all the questions that we have for you right now. So yeah, again, great talk. We will now begin with our next.
Yeah, before that, can I just answer one question that is in the chat? There's a one question, can a PhD guy do this program? Yes, absolutely.
Yes, great.
Yes, definitely, the answer is yes. Thank you again.
Great.
Thank you all for your attention.
Thank you so much, Dr. Vasuvadan. Okay, so our next speaker, I am pleased to introduce Alice Kamens. Alice leads the higher ed and public sector partnerships for North America at Google Cloud. Before Google, Alice spent time helping cities innovate their cities of service and City Smart, and began her career as an urban fellow for the city of New York. Alice holds a BA in Urban Studies from Barnard College and an MBA from Cornell Tech. So if you could all join me in welcoming Alice Kamens. Hi, Alice. Are you able to share your screen? Oh, sorry. I think you're muted still.
Perfect. I think it unfortunately requires the host to unmute me. So I've now been unmuted. Hopefully you all can hear me. Can I get an indication that you can hear me? Laura? Yep.
Yes, we can hear you.
Fantastic. Okay, it's great to be with you all today. Let me present. We'll get started. All right, I'm hoping you're all able to both see my presentation and to hear me. Is that correct?
Yes, we can see and hear your presentation.
All right, fantastic. Well, thank you all so much for having me today. It's great to meet you. I am going to share with you today some of the resources that we make available at Google Cloud for researchers. If you are a researcher, you're an undergrad or a PhD student or a faculty member, and you are conducting research with large amounts of data, you may be wondering how the cloud can help you. And so I'm here to share some of the resources you might want to check out as you either begin or continue on that journey. And I'll take you through some of what we can help in your own cloud research work. So I'll take you through an intro of me, brief intro, share with you some opportunities for learning and how you might want to actually build your own knowledge on what the cloud can do for research. I'll then take you to learning more about some credits we offer. These are credits that can be used to seed your cloud journey and get started in the cloud without having to open up your own wallet. And then talk through some of the communities we have where researchers are meeting and working together and talking about how they're using cloud for their research, which might be helpful for you all. And then I'll make sure we have some time for questions at the end. So I work at Google Cloud. I've been here for almost 3 years. And a large part of my role is focusing on how we support researchers in their work. So I've had the pleasure of working with a number of researchers over the past few years to help them use Google Cloud for their research. And I'm excited to share that with you all today. Feel free to contact me. My email address is here. I'm an open book. You're welcome to email me with any questions if you have questions after this session, or if you want to know more about how to use Google Cloud for research, I'm happy to answer any questions you have. So everything I'm going to share today is publicly available resources. It's all out there. So what I would actually ask you all to do, I know this is a bit non-traditional, is to actually open up this link in the tab on your computer or on your phone or however you're watching this, because I want you to go check out this page after we finish talking. So open up this link so you have it. Thank you, Elia, for putting that in the chat. I know it's non-traditional to tell people to look at something else while you're presenting, but I'll bring your attention back now to the presentation, but I want you to have this available and be a resource whenever you need it. Everything I'm going to share today can be found through this page, so keep it handy. Before I go any further, is it possible for folks to share in the chat, and maybe the OmicsLogic folks can read out? I'd love to just get a sense of who's in the room. And so if you wouldn't mind putting in the chat, if you're an undergraduate student or a graduate student or a PhD student or a faculty member or an industry, work coming from industry, it would be fantastic if you'd share those in the chat, or you can share them. You might be able to share them with me now that I'm presenter. I just want to get a sense of who's in the room. It will help me tailor the presentation a little bit. Just give everybody a second. Okay, great. I'm seeing bioinformatics graduate student, faculty member, industry, master's in molecular and cell biology, teaching assistant professor, focusing on, okay, great. So we have a really great mix of folks in the room. Thank you. It's helpful for me to understand that. And Elia is saying from YouTube, there's some undergraduate students. Great. Awesome. Okay, let's move along. So first up, let's talk a little bit about learning. And I just want to make sure I can see here what you all are seeing. Let's see. I think I can see that. All right, let's keep going. So I mentioned that we're going to start by talking a little bit about learning. And my thinking here is that there's probably a mix of backgrounds in the room on your level of Google Cloud knowledge or understanding. So we know that academic researchers and researchers in general are facing a world of constant change, new tools emerging all the time. Some of you on this call might already use Google Cloud for your research. Some of you may be new to it. And so we know that researchers don't always want to spend a ton of time maybe doing the same like training and certifications that an IT person would do. You have different needs. So recently we've done a lot more tailoring of our training offerings so that the training is actually very researcher specific and even domain specific. And I'll show you those in a moment. So we've created some new training pathways and resources for researchers specifically. Whether you're a researcher who wants to build a genomics pipeline or run a Monte Carlo simulation or create an auto-scaling HPC cluster with Slurm or host Jupyter notebooks, or whether you've never heard of any of those things and you're looking forward to learning, we have the training resources to help you do that. Moving into our Google Cloud Skills Boost, it's really our home page, home base for all training offerings at Google Cloud. You'll find everything you need here. And students and IT admins actually get an additional bonus, as well as faculty members. Anybody out there can have 30 free days of access to Google Cloud Skills Boost. And students and IT admins get additional credits, and as well as faculty members to distribute in their courses. And then I mentioned that we have these very researcher-specific training paths. I know this may be small and hard to see, but the paths here are called out. For example, if you are a genomics researcher, we recommend those training blocks on the right-hand column in line with genomics. So again, you'll find this all on that same page I had you open up under the section for researchers. Take a look at what's available in your domain and see if there's something that will help you understand how Google Cloud may support your research. For faculty who are looking to bring Google Cloud into the classroom, let's say for your undergraduate students, we want to make it very easy for you to teach Google Cloud to your students. So we've offered a free curriculum for faculty. You're able to access this again on that same page I asked you to pull up under faculty. It's a 10-module, 40-hour curriculum. You do not have to do all of that. If you want to take one module to integrate into your semester, I encourage you to take a look at this option so that you can use these lesson plans and use this content rather than having to come up with it on your own. And then one other thing I'll share, this isn't really learning, but I wasn't sure where to mention this in the presentation. I did want to share, we offer guidance for people who are applying for funding for cloud. So you may have had the experience of applying for funding from the NIH or the NSF for cloud. And in particular, the NSF has a process called the supplementary document. And so we have this resource available here. I will pop this link in the chat. This is a resource that you may want to bookmark if you're planning to apply to funding from the NSF anytime in the coming year. This will help you, give you some of the basics on how you might propose for funding for cloud. Happy to talk more about that or provide more hands-on support if you need it. And again, you have your email, my email. Let's see, I put that in the wrong chat. Here we go. All right, there you have it. All right, so now let's say you've gone through some of those learning experiences and you're starting to feel like, okay, cloud would be very useful for my research. I'd love to use it. One way to get started is through credits. We offer credits for researchers and also for classroom use. This isn't meant to be long-term funding. This is meant to help you get started and seed your research on Google Cloud. So again, using that same link I shared with you, if you go to the researchers section, you'll find access for this. Researchers are actually eligible for up to $5,000 in credits to use on Google Cloud. So the other credits I was talking about were for our training platform. This is to actually build in the cloud. And those applications are reviewed on an ongoing basis. There's no deadline, there's no time period. You can apply anytime, and you can apply for any topic. It's topic agnostic. And then for classroom use, if your faculty member would like to teach cloud to students, then there's also an opportunity to get a smaller credit for handout on a student basis, let's say as part of Google Cloud Computing Foundations course. So we want to make it accessible to get started, and these are some of the resources we have for that. So moving into communities, we offer a couple of different ways for researchers to connect with one another. We know that researchers are often learning the most from one another, far more than they can learn from anyone else. And so we have a couple of communities that help people connect on that level. So the two communities I'll highlight here, one is we have a researcher community for anybody who gets those research credits through Google Cloud, that up to $5,000 credit mark. They're immediately added to a community and a forum where people can ask advice from one another, find out if there's someone facing a problem that they've faced, and get advice from their fellow research peers about how they're using Google Cloud. So highly recommend that as a way to connect with others and with peers. We also have a research innovators community, and this is, it's almost folks who are really active users who are using Google Cloud quite a lot for their work. We had a 2021 cohort, and the 2022 cohort, I believe, was just selected. So we're not currently accepting applications. But again, if you go back to that link I gave at the beginning and go to the researchers section, you can sign up to be notified when applications do reopen, if that's something you think you'd be interested in. It's really a tight-knit cohort. They work really closely together. There are other opportunities for professional development and other benefits to be part of presentations and conferences. So it's a great way to get additional opportunities out in the world. And as part of that, I'll just share two examples from the 2021 Research Innovators cohort, which I think it's always helpful to hear examples of how people are actually doing this. This first cohort, 2021, had 33 Google Cloud Research Innovators. They were across 30 institutions and 8 countries in both industry and academia, and across all disciplines. So one example, one of the research innovators was Dr. Tapio Schneider, who's a senior research scientist at NASA's Jet Propulsion Lab. And his team has this product called the Climate Machine. It leverages recent advances in computational and data science to learn directly from Earth observations from space and the ground. And it helps harness huge amounts of data, much larger quantities than were accessed before, to create much better accuracy for predictions of droughts and heat waves and rainfall extremes. So he shared his learnings, how he was using cloud with this Research Innovators group, who also learned in turn from him. And then just one more story to share with you. Dr. Theodora Saz, who was also in the 2021 cohort, she's a computational scientist at the University of Chicago. She is actually using Google Cloud Solutions to identify and categorize images and analyze images in children's books as a way to understand what kinds of representation children are getting. So she uses AutoML, which has machine learning capabilities that can identify and categorize images. And they are using that to understand how and what kind of messages are children getting through books today. So really interesting, really broad range of topics. Really encourage taking a look at those communities if there would be of interest to you. So I'll pause here. There are more resources beyond what I've shared today. If you keep digging into that site, I'm also happy to meet or chat with anybody. If you'd like to email me, again, my email address, I'll pop it here in the chat. You feel free to reach out to me. And with that, I'll pause and take any questions you might have about Google Cloud for research.
Thank you very much, Alice. That was a great talk. All right, so some questions. First off, do you have any advice for those who want to get involved with Google or Google Cloud? Like, are there any qualifications that you say stand above the rest?
That's a great question. I think there are homes for many different types of people from so many different types of backgrounds and experiences at Google. I think my advice is pursue what you're interested in and bring that interest and passion to anything you interview for, whether that's at Google or elsewhere. I think if you're specifically asking about, is there anything qualifications-wise you're looking for research credits or anything like that? In terms of our programs, we have a really broad range of research projects that we accommodate through the Google Cloud Research Credits. So there's not a lot of particular requirements. I'll pop the link for that. And you can take a look. It actually lays out some of the eligibility requirements. So let me send that through. You guys can take a look.
Perfect. Thank you so much for that. Another question, what kind of resources do the cloud credits cover?
So the cloud credits, let's say you apply through that link I just sent in the chat, and you are granted $5,000 in research credits. You'll be able to use that on Google Cloud Console for any product that you need. So there's no limitations. It's not like you can only use it for BigQuery, or only use it for one or another product. It gives you access to the suite of Google Cloud Platform solutions.
Great, thank you. Another question, can working professionals try out Google Cloud and get credits?
So working professionals should go through our traditional Google Cloud free trial. And I can pull up the link for that. There is a free tier. Let me pull that for you. The offering that I put in the chat before is really targeted at researchers and with the knowledge that researchers often may need a jumpstart in terms of accessing cloud. But those who are working for professionals, anybody can get $300 in free credits to get started. And actually, that will take you pretty far. That will be enough to help you figure out if this is going to be useful for you. And at that point, you can figure out, okay, how will I be able to use this? And is this something that I want to pursue?
Great, thank you. Another question, which universities are now using Google Cloud for genomics specifically, if you know?
That's a great question. I don't know off the top of my head, but two other colleagues of mine are going to be presenting from Google Cloud later on in this symposium, and especially my colleague Jesus will be able to tell some really rich stories from universities that are using genomics. He actually works much more closely with customers and helping them access and use Google Cloud. But one thing I will pull up for you. I know I'm sending you guys a lot of links. If you give me just a second, I'll share with you a couple of different case studies from researchers. It might take me a second to get that together. And I also had a question in the chat sent to me via direct message. Do you allow training facilitators who run bioinformatics events that train researchers to apply for GCP credits for those events? So I think right now, the way that you can get GCP credits to distribute is if you are a faculty member or an IT staff member at a university or a researcher. If you're outside of that domain and you're not actually working for a university, you'll probably want to either access 30 free days of Google Cloud Skills Boost or figure out if there's another way to do that. Reach out to me and we can figure out via email if there's a good way to help you. But yeah, typically the resources I was presenting today are typically designated for university affiliated folks.
Great. Well, thank you again for speaking. If there are no further questions, I think we will move on to our next speaker. Thank you again, Alice. That was a great
Thank you so much. And I will put one case study in the chat of a team that is using Google Cloud for genomics work. So just popping that in here.
Perfect. Thank you again. Okay. So our next speaker is Dr. Adelaide Rhodes. So Dr. Rhodes has more than 20 years of experience in analyzing big data at the molecular, organismal, and ecological scale. She holds a PhD in zoology for NCSU and an MS in biotechnology and bioinformatics from Johns Hopkins. As a bioinformatician and strategic consultant, she has assisted dozens of universities and government researchers to analyze cancer, crop science, bacteria, virus and plant genomes, transcriptomes, and disease variant datasets. She has developed advanced workflows for secondary and tertiary analysis and has written custom scripts in Bash, R, MATLAB, and SAS to interrogate large datasets for meaningful signals. She also has a strong publication record that spans multiple disciplines. Recently, she has helped researchers develop pipelines for data-driven discovery, such as HAIL, GWAS, and CNN, using a variety of workflow languages like WDL, CWL, and SnakeMake and Nextflow on HPC and commercial cloud computing resources like AWS and GCP. She also provides consultation on AI and ML genomic opportunities for data-driven discovery in the health and life sciences. So now I would like to turn it over to Dr. Adelaide Rhodes.
Hi there. Should I share my screen for my PowerPoint?
Yes, please. Are you able to do that?
Yeah, let me share screen and pull it up. I wasn't sure about the meeting format, so let me do that. Oh, great. Can you guys see my slide?
Yes, we can. Thank you.
Okay, good. This is going to be a little different. And thank you, Alice, for that presentation. I'll just say that I use Google Cloud. You might hear a little bit of that when I talk about what I've been doing. And yeah, so what I'm going to talk about today is, wow, that sounded really impressive when Laura read out all those like qualifications and what I do, but I didn't just start out that way. So my talk today is a little bit of a motivational speech about anybody can get to where I am if you just put your time and effort into finding ways to kind of get involved and improve your skill sets. So I do have to say that because I work for an NGO now, like the views and opinions are only my views and opinions, and they're not a representation of any organization to which I currently belong or I have previously belonged. Okay, so I'm gonna walk you guys through my little bit of my life story and then give you at the end talk about lessons learned. So in order to kind of give you guys a bigger picture, I'm just gonna talk about being a scientist and then switching from being a scientist to becoming a bioinformatics faculty member at a university, and then realizing that wasn't a fit, and then moving on to working in bioinformatics full-time, mainly with cloud content. And then of course we all kind of experienced the pandemic. So what does that do to our careers? How does that look like? And then is this an opportunity, not something that hurt us? Okay, so just to give you an idea of where I was 20 or so years ago, I studied copepod crustaceans, which are at the bottom of the food chain in the oceans. They're very important species for fisheries. There's been 75,000 described species, and I spent most of my time looking through a microscope at these organisms and trying to describe them. And then I later got into DNA barcoding. That was my gateway into bioinformatics because I was generating a lot of sequences for unique organisms that were being collected here. You can see Sherry Reed and another, oh, that's me down below in the lower right corner. I used to go out and collect benthic species, sort them, look at them under a microscope, but it would be really nice to know if this had ever been observed somewhere before. So this was near the beginning of the Barcode of Life project. So I contributed a lot of marine species to that effort. Basically 18S or COI cytochrome oxidase 1, species sequences. So this was actually back in the day, like we're running them in the lab, had a Sanger sequencer to put them together. And the frustrating thing was like, I would go onto the databases and I'd be like, and no one's ever seen this before. and that pretty much happened for most of the species I was working on. I was cataloging them to be of service, but a lot of them were unique observations. And then this happened. So I was working in Florida around the time of the Deepwater Horizon oil spill. And what's happening here is that we had to collect a lot of data really quickly. So this kind of pushed the boundaries of what was being done in our lab to include a lot more thought about how to collect a lot of data about a lot of different species over several years. And I was put in charge of not the copepod crustaceans, but the Tenea crustacean observations. And you can see here on a slide, I had to look back in time. I wanted to know what had people observed previously so I could understand what I was observing now. Once again, just validating whether there were new species in this region or not. So it did involve data, but it didn't involve any genetic data. But I did want to do genomics because we were working in a very interesting and unique environment, the deep sea. So deep sea environments are really different. Not sure how much you guys know about it, but there are coral species at the bottom of the ocean that do not require photosynthetic organisms, the zooxanthellae, to live. There's a lot of symbiotic organisms, things that because it's very sparse, there can be a large distance between species or little species islands. You know, things have learned how to grow up together. So there's just a few pictures here of the pretty corals and the commensal species, the brittle stars that hang on the corals, but there's so many examples of that. So it's a really unknown environment. And so getting a really good list of species that were at the different sites was really important. And as you can imagine, being very good at organizing the data, being able to access the data, you know, tens of thousands of observations, plus observations on the conditions, the biochemical conditions at the bottom of the ocean were important too, because we were kind of trying to build data layers, like looking at the species, looking at the chemistry, looking at physical conditions, looking at where the plume was flowing. That all was very complicated and resulted in quite a few papers. So what I realized through this process, and where am I going with this, is that I really like the data part. And so I did get a chance to do some more barcoding type genomics as a side project during this time. So I really realized two things about myself, that the parts I liked about my job, believe it or not, as a marine biologist, did not involve going out to sea. They actually involved the part like where we had a bunch of data and a lot of spreadsheets, and then we had to tell a story. We had to say, this is what happened here. This is what we need to pay attention to. So looking for patterns, trying to come up with hypotheses and then testing the hypothesis all on a computer. It's fascinating stuff. So that's when I decided to make a career change. And it didn't happen overnight. I did go back to school, even though I had a PhD. I chose the Johns Hopkins University program in Biotech with a major in bioinformatics because, well, first of all, it was one of the only two that were available online, 100% online at that time. The other one was Stanford, and that Stanford was double the price. And the other thing was for the amount of time you had to invest, it was very comprehensive. Like you took molecular biology, you took computer science classes and all that. So you have to understand my PhD was in biology, didn't mean that I knew how to do bioinformatics. So I really appreciated that program. And so as a result of getting a master's, I actually was able to get a professor job over at Oregon State University. So that's what the little beaver guy is there. And so I ended up moving over to Oregon State, and the best way to learn is by teaching. So even though I was a very new bioinformaticist, I taught classes before. So I thought about, what do I want to learn? And then I started teaching things I really wanted to learn about. And I taught them several times a year to graduate students, undergrads, and then other people like myself, basically a lot of people who came up through school, finished school before any of this existed. So I had to, construct classes. I'd had to understand how to do the data analysis. So it was a great way to learn how to do more. And so that's when I also realized what is my favorite programming language. It's Bash. So I really do like Bash, and it's just the cleanest and stuff. But I did have to know R and Python. So being in a school, teaching, volunteer teaching, mentoring, for those of you out there who feel a little bit of the imposter syndrome, it really helps you get past that, because you're transmitting, you may only be a couple steps ahead of somebody, but you are a couple steps ahead of somebody. So by transmitting that information and getting involved in projects, I was able to kind of overcome that. And then the other thing that I got to do, which was really fun, and if you notice at the beginning, the little guy holding up the sign is a beaver, that was the mascot of the school. So I was actually put in charge of a really cool opportunity to teach students through generating hands-on experience with a genome assembly project. And of course, we picked the beaver to do the genome assembly, which meant that the students and other professors that were working on the project, we were all involved from the very beginning to the very end. Data collection, data analysis. What did we learn from having another rodent genome? Because of course, mice and rats are fundamental organisms for medical research, but there isn't a lot of diversity out there at that time for other genomes that you could look at for other answers. And so there's a few features of the beaver that aren't present in other rodents, such as the castor sac. They make a castor oil, and the fact that they live most of their lives in dens in the water, so they do have to be able to hold their breath a very long time. And another feature about beavers is that they are ecosystem engineers. So their brains may develop differently. So I was in charge mainly with the transcriptomic part, and then cataloging. We didn't really find any new genes, right? We just found homologs, but there were long non-coding RNAs that were very interesting that might have shown how beavers evolved over time to take advantage of the environment they lived in. And so that resulted in this paper about pan-tissue transcriptomic atlas methods to find long non-coding RNA. So this was the first paper where we actually built a new pipeline, something that hadn't been done before, in order to quickly screen for long non-coding RNAs in non-model organisms. So just think about the fact that, like, if you haven't had a lot of chances to get involved or learn, this is an opportunity, like maybe on your own campuses, at your companies, like there's always a way to get more experience. So volunteering or finding opportunities to help other people do their projects is a great way. My job was in the core facility. For those of you who don't know what a core facility is, it's basically a service that is provided by the university. So anybody studying anything can come into your office and ask for help, and the university subsidizes the salaries and the supplies for people in the facility. So I'm just going to flip through a few different examples. And so these were not research projects I initiated. These were ones where people came into our facility, asked for help, and it was my job to try to get them the best answers I could. So we looked at viruses and omycetes, which are slime molds. Another project I got involved in with Molly Burke's lab was a molecular yeast evolution pipeline upgrade. So that was an automation project. I also was co-author on a paper about canine human bladder cancer comparisons. That was very interesting because we not only got to do a deep dive into canine cancer, we actually looked at human cancer and combined data sets. So that was really comprehensive study that was very challenging. So I learned a lot from that, and I also got into a little bit of the modeling. And another one was looking at pathogen-associated molecular patterns, so basically innate immunity in bugs. So I did more than one of those. Stink bug was a more well publicized paper that I was on, but there were, like, as you can see, there's like couple dozen people on the paper. So you can get involved, you can publish. You don't always have to be the person initiating the idea. Like if you're willing to pitch in and look for unique patterns, or you're asked to because it's your job, you could certainly do research. And then I did continue my own research looking at copepods. And so I was using reduced genome methods, to be rad, to try to find biomarkers at a time when on the right of the screen here, you'll see like a little map with pins in it. That's the coast of Oregon and California. And what's happening is there's a warm water front that appears every few years called the blob. They really do call it the blob. And so the idea there was, well, if there's this large patch of warmer water, could copepods from southern regions be migrating into Oregon and sticking around? Now, who cares, like, right? But copepods are the main food of salmon. So if they're not recognizing the species of copepods that have come into the area because of the warm water, are they going to have problems maintaining their body weight and living through their maturity? Because salmon come from rivers in Oregon and California, and they go out to the ocean and they live in the ocean for a few years. And then they need to have all this energy stored up so they can make it back into the rivers in order to, be able to spawn and pass on their genetics to other salmon. So it was a really interesting project. Still went out to sea a little bit and generate some novel data that kind of showed that, yes, there were some stratifications in the populations that hadn't been seen ever, before, and that was enabled by genetics, genomics, and big data. Oh, and I just throw this picture in here because we were also having the pyrosomes. There were like a million things called sea pickles. So we'd throw out our nets and we'd pull them in, and instead of getting a bunch of fish and flounder and stuff, just FYI, we got these things called sea pickles, and they were a big deal that year. And that had a lot to do with global climate change, right? Because this warm water area, other species were coming in and taking advantage of it. So I could have been happily content to work on other people's projects and everything, but I had also been getting really fascinated with cloud computing and using cloud to answer questions. So this was about 7 years ago. I mean, now we all use cloud, and cloud is accessible. We just had a presentation talking about cloud, but I really, really wanted to go in that direction with my career for some reason. So I ended up moving to Boston, and my first job actually did involve Google Cloud platform called Terra. If you're not familiar, it's at the Broad Institute. And the idea of a Terra is that it brings together all the resources you need to reproduce a study. So you may know the pain of reading a paper, a very interesting paper, and you're like, oh man, I could totally use that idea. And then you start reading through the methods, and it says produced by custom scripts, that are, and then you'll call the lab and they'll be like, oh yeah, those custom Perl scripts, disappeared when our grad student left the lab. We have no idea how they actually did the analysis, right? Or there's a data set, there's like a spreadsheet, and it's maybe just one of the pieces of metadata that was used to generate the final product, but it's a very important part, but that's not in the supplementary materials. You can't find it again. And then you call the lab and they're like, we don't know, that was like on a book drive, and then we had a fire, and then we don't have that anymore, right? That happens all the time. That's why cloud is so important, because it gives you a place to put all your stuff. And Terra is cool because you can put together your data sets, you can put together your methods, and then you can actually run really high throughput workflows using things like the workflow development language, or CWL. So you can use your free credits. Like I heard the person before me, Alice, just talking about that. You can use your free credits to play around with Terra. It's mainly biomedical research, but there's a lot of really cool, interesting stuff on there, and you can read it for free. Like you don't actually even have to sign in to get a lot of value out of it, because you can flip through the pages and see how other people did the program, or how someone wrote their WDL, or you can find Dockers. Like you might need a Docker for Tidyverse or something, right? Like people have written that like over and over again. So that kind of stuff's available in Terra. Oh, thank you for putting the link there, Laura. It's terra.bio. But anyway, I don't work there anymore. I really did enjoy my time there. But I really wanted to not be stuck in one type of role. As a scientist, I kind of wanted to explore and do more diverse things. But I'm also just putting this up here. This was one of the products out of my work at Terra was like a synthetic BAM generator. But you can see here this arrow. If you are not familiar with the FAIR principles of data science, I really feel like it's worth reading up about because there's lots of opportunities. So once again, if you're starting out and you're not quite clear which direction to head in your career, but you do want to make a contribution, FAIR is a way to kind of get involved and know like how to present yourself, how to present your data, how to make things reproducible. Because if people can find your stuff and use it very easily and reproduce your research, they're more likely to cite you. So if you're still on a science track, just thinking a little bit about a project before you start it is going to help you. And so I just gave a talk to my coworkers today about being FAIR. So it just means findable, accessible, interoperable, reusable. All right. Don't make a spreadsheet and then don't put it in supplementary in your papers, in other words. But data was getting bigger. And like I said, I wanted to get more challenge out of my work. So I went to a third-party partner for Google Cloud. So I actually trained people. So I wanted to get beyond just doing a thing. And so once again, I wanted to learn how to do it better. And for me, my philosophy is if I teach somebody how to do it, I can learn how to do it better myself. So I'm very fascinated. This happens to be a Google schematic, but there's AWS corollaries. There's also, Azure. I'm not trying to influence any kind of product decisions, but basically you can build up from, I know how to use a spreadsheet to, okay, so I can take that spreadsheet and put it into a database. So now I know how to use BigQuery, and I know BigQuery, oh, wow, that's going to enable me to ask interesting research questions through machine learning. And then, well, maybe I need a model for this thing I'm trying to do. I'll go over to the Google Brain and look for some advanced protocols, right? So I like the versatility and the flow of Google, so I tend to default there. But like I said, there's really great choices on all the cloud providers. And this is just an example of kind of a product that I came out with, showing the power of cloud, because we are generating genomes at a faster rate than anybody ever anticipated. And even just knowing where to put them is a challenge. So cloud, it's all about like, how do I store a large amount of data? How do I access that data? And then what can I say about the data after, everybody else has done all this really important work, putting the data where we can get to it. What can I do with it? So those are the kinds of questions you should ask yourself as you're going through your career. Like what parts do you like? Where do you want to be? And then like, I really got into Kubernetes and Nextflow and all that through those experiences. And one thing I found I lacked in, and I went back and got some more training through Coursera, et cetera, was how to present the data back to the researcher. So I spent a lot of time thinking about these interactive tools such as RShiny or Plotly or Observable, right? There's a million of them popping up now because that is a common need. You could be somebody who specializes in that. Like if you particularly don't like any of the database stuff, or you don't particularly care about the infrastructure, just knowing how to communicate is so valid. And these types of things are very easy to learn. And there's tons of free documentation out there, Coursera, Udemy, GitHub itself. Like people put up very nice and web pages and share like how to build these like visualization and interactive tools. And then that resulted in me like doing some work where you can find these videos out there on the internet if you care about, but like trying to fill in those gaps. Like I'd be like, man, nobody knows how like the NCBI is on the cloud, right? Like everyone keeps hearing that NCBI is on the cloud, but like, how do you actually like go to the cloud, get the data and use it for something? That's the gap. That's the gap that I keep trying to fill. That like, yes, it's on the cloud. Cloud is great, but if you can't have a concept of even what that means, you're not even going to be able to take advantage of all these cool, freely available free resources. Because even though Google and Amazon and Azure cost money, they all have free tiers. And I heard a little discussion about credits. There's ways to get credits. Like when you sign up for Google, you get like $300 right away. There's ways to kind of dip your toe in the water without spending any money at all, but getting the benefit of thousands of researchers' data. And so I've seen this a lot with the pandemic data, like a lot of that was made publicly available. And there was a kid here, like his high school project was making a dashboard for the data that just kind of updated people, and it got a lot of buzz. It was really popular. And then now he just went out and made a dashboard for Ukrainian refugees, like people who want to share their houses, connecting them, because this data exists. There's just a gap in, like, how do I get to it? How do I find it? How do I use it? So that's a really good opportunity. And then, as I mentioned, there was a pandemic. I didn't build a dashboard, but I did other things. I spent a lot of time like on trying to develop skill sets through hackathons, and by volunteering once again to teach through the Bioinformatics Virtual Coordination Network, which has a bunch of free lessons like on RNA-seq, metagenomics, genome assembly, gene annotation, things like that. All right. Oh, this is my last slide. So just a little thought about the different categories of bioinformatics. As you can see, it's a really diverse world. Not everyone's going to be like me, where they have a short attention span and you're going to jump from one to the other, right? But that's my style. That's who I am. So I would say to you guys, be who you are, figure out which part you like, invest time in yourself, always be building your skill sets. Like I said, you don't have to spend a lot of money. There's so many free things out there. Networking, this group is great. There's always so many cool opportunities on the LinkedIn coming out of the group that is sponsoring the symposium, reaching out to people like a little further ahead, like 2 steps ahead of you. Because like I said, I'm never, like put off by somebody asking me for help, because I'm always like, oh, that's a great opportunity for me to learn this thing better, because now I'm going to have to explain it to somebody how you do this thing, right? So think of it that way if you're shy or you're afraid of asking for help on a particular project. But yeah, that's about it. That's all I had to say. This is me. This is who I am. And I guess I have a few minutes to get some questions. And I didn't, okay, here we go. Let's see. I did see that Sona said that you do diversity and ecology jellyfish. So hi, Sona. Yes. And then
actually, it's not me. I'm sorry. It was I posted it on behalf of Gisele.
Oh, thank you. Thank you.
Because I think he didn't have or
thank you. Thank you very much for posting that.
And yeah, but I must say, now that I'm online, I know this is out of my turn, but extremely inspiring talk. Amazing talk. Thank you.
Thanks. Yeah. And then there's one here, question 1. How do you compare working for academia versus working for the industry? That's a great question. I'm an impatient person. So sometimes for me in academia, things didn't move fast enough. But I also don't have all the energy I used to when I was younger. So the problem with industry, sometimes it moves a little too fast. So I'm looking for the Goldilocks job all the time, where it's not too slow, but it's not too fast. In academia, the resources are limited, but that means your creativity gets an opportunity to do the work. That's why I know about so many free things, because as an academic, I had very little money. So, I had to accomplish big ambitions with nothing. So I hope that answers that question. Question 2, how do you decide what kinds of research projects to focus on to advance your career? That's a great question too. And because I'm the type of person who says, do what you love, I also like to love what I do. So my career decisions a lot of times have been led by, I just don't love this anymore. This is not something I see myself doing like for 20 years. And so then I'll just go to the next level. But that's not very practical for those of you who, may not have the ability to move around like I can. So I would just say, look for opportunities in your company. There's always somebody who needs like a hand or help. And you never know, just like by volunteering a little, spending that extra hour with a project per week, showing up for a meeting that you weren't necessarily invited to, but you heard about and got permission to attend. Those are ways to kind of edge yourself over. You don't have to make leaps like I do. You can just edge yourself over. And then, of course, taking the trainings online are very helpful because then you show up and you're now the expert, like maybe in your group, because you spend a little bit of extra time on Coursera or in the Pine Bio, platform, learning how to do a workflow. And it's needed all the time. Like other people just don't have the time to look. But if you have a little bit of time to look, invest in yourself. I only got 2 minutes left, but question 3, how has bioinformatics changed over the past decade? It's just gotten bigger and harder in the fact of, storing data, but it's gotten more interesting and impactful because you don't have to collect the data to be the person to say something about the data. So that's pretty cool. Let's see. Sorry, I'm going to skip. Manor says, I'm a student of biochemistry. I love to learn more and more about bioinformatics and genomics. Yes, people, please feel free to reach out to me through LinkedIn. I'm not sharing my email because I don't check my email enough to be a great person, but LinkedIn, I see it pretty regularly. So do that. And then last question from Elias is, what is a major skill students struggle with on their path to mastering bioinformatics? I think it's confidence. I don't think it's even a skill. I think it's just believing that you are. So a lot of these changes I made was me saying, I am that now. I am a bioinformaticist or I am a data scientist. Nobody waved a magic wand over my head and made me that. So that's why I'm saying students struggle with that. So just be the thing that you think you are, and you are. So, and then the BVCN is just a freely available set of lessons on GitHub. It's called the Bioinformatic Virtual Coordination Network. I do link to it in my LinkedIn profile. You can see it in the featured pages there. There's links and there are videos and basic lessons. It's all free. It's all freely available. Just how to do metagenomics. It's mainly metagenomics, but there is some transcriptomics. But for beginner bioinformaticists, it's a nice little set of lessons that we wrote. Let me see if I can find it real quick. I didn't come prepared to share that, but let me stop sharing and I will put it in the chat momentarily. Ooh. You guys still see my screen?
No, we do not see your
screen. Okay, good. Okay, so I'm sorry, I didn't want to drag you through me looking for this. So, okay.
No worries. Well, thank you very much, Dr. Rhodes. That was a great presentation, very informative. I think inspiring for a lot of young bioinformaticians. So next up, we are going to transition to some speakers from South America. So we are going to begin with, let me share my screen. We are going to begin with Dr. Georgios Ioannis Papas. So it is my pleasure to introduce Dr. Papas to you. He has a bachelor's degree in biology from the University of Brasilia and a PhD in biophysics and computational biology from the University of Illinois at Urbana-Champaign. He has worked with computational prediction of protein structure there. He also joined the faculty at the Catholic University of Brasilia in 2001, moving to the fledgling field of plant and microbial genomics and teaching bioinformatics for undergraduate and graduate level courses. Since 2012, he has been an assistant professor at the University of Brasilia, heading the computational genomics group and teaching courses such as bioinformatics, molecular biology, introduction to Linux, and programming for biologists in R. So please welcome Dr. Papas. Hi, Dr. Papas. I think you are still muted.
Okay, here we go. Can you hear me now?
Yes,
can
you
hear
you.
Okay, great. Well, thank you so much. I will start sharing my screen here. Just a second. And Can you see my presentation? I guess
Yes.
so. Could you give me a feedback, Laura? Okay, so my name is Georgios Papas. I work at the University of Brasilia, and I'm going to present to you some challenges and perspectives of bioinformatics teaching here in Brazil. Okay, so first of all, Brazil, I'm really far away from everybody. We're located here in Brasilia, the capital of Brazil, far away from everything. And Brasilia is a nice city with very modern architecture. So I'm gonna start off like Dr. Rhodes, giving you my personal experience in bioinformatics, even before bioinformatics was born. I'm not that old, but I'm from the 80s, you see? So the 80s was really nice. And in the 80s, I started programming with computers, so to speak. Okay, so what we had was this machine with 16 KB RAM. We didn't have monitors, we didn't have hard disks, floppy disks, anything else. But it was really fun having being initiated in, let's say, computers, even before college, okay? But in the 80s, there were several things missing, okay? First of all, Linux, several programming languages that we use today, like Python, Java, R, World Wide Web. We didn't have this. We didn't have cell phones, GPS, MP3s, and so on. But it was really fun. So moving to the university, since computer science was such a new field, my other passion was biology. And of course, I got a formal education in molecular biology. So undergrad degrees, a degree I got at the University of Brasilia, and I have training in all wet lab techniques to characterize biomolecules, anyone, DNA, RNA, proteins, okay? And even by that time, I did Sanger sequencing by hand, okay, running really big gels, like a meter-long gels with radioactively labeled DDNTPs. And in recollection, the wet lab was a very essential experience for me in my formation, mainly to get the notion of the complexity of biological systems. Okay, so, but exactly at that time, as you can see here in 87, I started programming and doing research at a lab that worked with protein secondary structure prediction, okay, based on amino acid sequences. And by that time, I started learning BASIC. Myself, I learned C, and I improved in terms of hardware, and I got this computer with a 48 KB RAM, okay? But it was really fun. Then I moved to Illinois, University of Illinois at Urbana-Champaign, where I exclusively did work in computational biology. And for that, I was working with a protein structure, trying to categorize structural motifs. And by then, everything was programming, computers, and it was really nice. And my computational biology, this is very important. And it was in the 90s. And at that time, in fact, I was one of the first persons in the world to use a web browser, because it was developed like next door to me, the Mosaic web browser. And I recall we had like 15 links worldwide. That was all we got in terms of websites. And in my evaluation of the web browser, I said, oh, okay, no, this won't work. Bad decision. But okay, since last century, I never set foot in a wet lab anymore. I was strictly a computational biologist. So in recap, what I can say, I can say that I had a biological background from theory to practice, and continues computational to use in terms of programming languages, operating systems, algorithms. But this is important. No formal academic training. It was all by myself, reading through paper books, okay? And what happened? In the 80s and 90s, in terms of biology, we didn't also have many things, such as automated DNA sequencers, genomics, a holistic view of biology. You see, we had alternatives, so gel-based Sanger sequencing. Genomics was only in dreams, okay? And biology at that time, last century, wasn't what I can say holistic. It was, we had an individualized view of individual components. Oh, this protein, this promoter, and that's it. And what we didn't have was this term, bioinformatics. We used to have computational biology. We still have computational biology. And everything we did was computational biology. And then, of course, the genome era of biology began. And it was like the big data era of biology. What happens is, let's say it started at the end of last century, as all of you know, in 95, with this first genome published. But then in the 21st century, we have explosion of genomics. And this is a new trend, a new paradigm for biology itself. So at least to begin with understanding complex organisms, we need to have the DNA blueprint of the organisms in order to try to relate phenotypes with genotypes and several other functional aspects of biology. And this is going on, okay? So we even have this very interesting project, the Earth Biogenome Project, in which they want to sequence everything in the world. And this will be some very doable in the next decades, okay? Meaning that we are gonna have lots of data, let alone the data that we already have. Okay, so I really like this paper here that I want to share with you, Teresa Atwood, from the beginning of the century, where they say that bioinformatics is immersed in a field of high throughput techniques that generate vast amount of data, but data can only be useful if we can turn in data into knowledge and knowledge into understanding. So I'm gonna highlight the key words of what I'm saying: data, knowledge, understanding. Okay, and of course, what we have is when we are in the high throughput, not just genomics, all the omics techniques that generate vast amount of data. Of course, the new biologists should be inserted in this new reality, and the biologists should be proficient in doing some computational analysis. On the other hand, we have bioinformaticians exclusively linked to this field. And in this case, okay, excel in computational analysis of data. Why? We have to turn these data analysis into knowledge. But there's a big problem here. There are differences between these two profiles in terms of overall goals, the perspectives of what I can do with the data, and of course, in the training level, okay? And to turn data into knowledge, okay, this new breed of professionals have to know algorithms, other areas, statistics, aesthetics, as Dr. Rhodes told about how to present the data. Oh, this is very, very important also. But also we need biology. And to transform knowledge into understanding, here comes our focus, is biology, okay? And with that, I should quote Wally Gilbert here, in which he says that biologists must be computer literate. And this is a new paradigm in the whole biology, okay? And in this new biology, for the 21st century, the author say that the new biologist should not be only a scientist that knows little about several things. We need a deep knowledge of one discipline, let's say biology, one of the biologists. Okay, we have several biologies, okay, and we need working fluency in several other sub areas, okay, and what are the challenges of teaching bioinformatics? So I've been teaching bioinformatics courses for 20 years, for the past 20 years, since 2001. And after 2 decades, I'm still working on it, okay? It's not straightforward. Why? Because it is very difficult to create and adapt a curriculum that will fit the needs of all student aspirations. Okay, this is there's a big diversity in this aspect here. But anyway, I divided all my courses based on student background. Okay, and essentially I have courses for undergraduates, graduate students, and for students interested in a bioinformatics career path. So I organize the courses like this with the for the background, the audience. So my audience is biology and biotechnology majors, and in fact, biological sciences, even agronomy, some medical students, and so on, and so forth. And the objectives here are just to provide a general introduction of bioinformatics and bioinformatics tools. And then we have courses for graduate students where the objectives are reflect the students seeking proficiency in using computational tools to analyze their data. And the final thing are for research. And of course, again, for only those who want to become bioinformaticians. Okay, and so for undergraduate level, it's a single course, a 60-hour course. It's a required course with theory and practice. Per semester, I have about 40 students. And now, in terms of graduate courses, I teach multiple courses, and I will get into this later. And for research, it's personal mentoring. So I was happy to see that ISCB developed, let's say, what a bioinformatics core competence would be for several levels. So they had competencies for a bioinformatics user, a scientist, and an engineer. And this is, if I don't expect you to read that, but this is exactly what I have here. So I divided into tiers. So at the undergraduate level, the major focus is given to study biological databases. Okay, and why is that? In which databases? Normal literature, nucleotides, proteins, everything related to proteins in terms of sequence, domains, enzymes, biological pathways, genomes, variations in phenotype, and data quality assessment of the databases, and of course optimized querying the data, and of course some basic tools. Everything is online here for this course. Okay, and why this operation? Basically because of, again, Walter Gilbert, when he said, oh, a new paradigm in biology emerged in that biological data. And for that, the starting point of biological investigation will be theoretical. And with that, I can present you a simple, let's say simple, in 4 lines, a question that can be asked. Let's say, how do I know all the protein coding genes from DNA repair system of the fungus C. neoformans? This is a very complex biological question. And of course, you can answer with using bioinformatics tools, but just doing canonical database surfing and filtering may not be the answer, you see. So we have some, what I consider optimized alternatives to using and querying that databases, and which it's kind of really simple. Okay, it's stuck here. Just a second. Okay, I'm having a problem with this slide here, but essentially it's gene ontology, ensemble, and biomark. Okay, but here we go. Gene ontology, ensemble, and biomark, and we can do this in less than 15 minutes, okay? So this is one of the goals. So in terms of the overall appraisal of the students, this discipline has had very good reviews from the students, because they see bioinformatics tools as essential for their careers, even though they might not be bioinformatics. Active points are the lack of textbooks. I'm writing one for them, and but for my perspective, when they are doing studies and using the concepts given, the basic concepts given in bioinformatics. The negative aspect is that I'm having a relatively low number of students actually engaging in bioinformatics research. This is low, it is low. At the graduate level, I have one market division of courses. Okay, so what happens is that I teach several different classes, reaching NGS, Introduction to Linux, R programming for bioinformatics, okay, and pipelines for bioinformatics. I'm not going into the details here, but what are, in my perspective, what they need to know. First of all, NGS, they need to know the data. Linux, it's the work environment for computational analysis. Programming in R, for instance, it's our work tool. And pipelines in bioinformatics, it's our workbench where I can mix and match several bioinformatics programs to produce complex analytical workflows, okay? I had some decisions. Okay, you might ask, why you were teaching R instead of Python? For me, since the students are only coming from biology background, I think R, it's a little bit easier to learn in like a 40-hour course, 45-hour course, okay? And in terms of pipelines for bioinformatics, the major goals were students, since the background is just biology, they should be able to install and run published pipelines. And what happened was that I was trying to teach them how to program in Nextflow or SnakeMake, but the total lack of programming skills wasn't a nice experience. So we had to halt this thing. Okay. So here I come with an example of the richness of how just simple,
I'm so sorry to
let's say,
interrupt.
Linux. Yes.
Oh, Dr. Pappas, we are running low on time. Do you think you could wrap it up in the next few minutes?
So
thank you.
I'm just gonna skip this and for the research level bioinformatics, what happened? I need, okay, personalized mentoring, okay? So we work with several plant genomes. We published this eucalyptus, the common bean genome, and we are working now with Brazil nut tree genome. But we also work in my group with several other species, with bacteria, fungi, with all omics technologies and all NGS technologies is a showcase. And it's very difficult to adapt to these technical changes. And this poses both opportunities and challenges regarding bioinformatics education. And so essentially, we have been developing several pipelines which need several tools, individual tools. So Nextflow, Docker, Shiny, R, and this is not trivial to teach in regular courses along the way. So just to finish up, my experiences were in terms of gender imbalance for graduate and undergraduate courses, absolutely no imbalance. But for research, I still observe a gender imbalance. And let's not forget that the mother and father of bioinformatics is Margaret Dayhoff. So we should close this gap. And just to finish, the new biologist needs several skills and working fluency in programming, statistics, and knowing the data, but deep knowledge into the biology is, let's say, what I consider the most important thing in the moment. Okay, with that, I'm going to skip the other slides, and I will finish up my presentation here with that. Okay.
Great. Thank you so much, Dr. Pappas. That was a very awesome talk. No worries. Okay, so we have a few questions for you. The first one is, where do you think bioinformatics is going next?
This is a very interesting question because since, let's say, I saw it growing from the beginning, and technology simply is molding, is shaping bioinformatics. So we started with, okay, we cannot have genomes, we cannot sequence DNA, then we can sequence short, then we can sequence long, and then bioinformatics, and this is a very important point, bioinformatics will change next semester, you see, we are tightly linked to technological advances, new biological discoveries, and bioinformatics is involved into that. So it's an ever-changing field. It is challenging, but it's really nice. Why? Everybody needs bioinformaticians, everybody.
Great, thank you so much. I think we have time for one more question. What are some bioinformatics careers that are available in Brazil specifically?
Okay, so in Brazil, we have huge problems now. And unfortunately, career-wise, not for bioinformatics, Brazil should be an exception in the world. It's not because we are short in bioinformatics careers, we are short in science careers because economic and political difficulties. And what we're doing with bioinformatics, all my students, they are moving abroad. So all my PhD students are moving abroad. One is now in University of Florida and the other in Denmark. We cannot, Brazil cannot absorb. And this is detrimental for future development of the country, unfortunately.
Okay, well, again, thank you so much. And that was an awesome presentation.
Great, thank you so much. I will unshare here.
Thank you.
Okay, thank you so much.
All right, so it is my pleasure to introduce our next speaker, Dr. Gabriel Goltzer-Gotti. So Dr. Goltzer-Gotti was born in Caracas, Venezuela, and spent most of his childhood years on a Caribbean island off the coast of Venezuela called Margarita Island. His passion for genetics brought him to obtaining his biology degree at the Universidad Simón Bolívar in Caracas, where he subsequently graduated with honors from the Masters in Biological Science program in 2012. He continued his education at Tufts University in Massachusetts, where he got his PhD in 2018. Up to that point, his focus was to apply genetic tools on non-model insects to various questions in the field of taxonomy, population genetics, ecology, and evolutionary biology. He did both the experimental work at the bench and computational analysis in R, Python, and used several other bioinformatics tools. At the end of his PhD, Gabriel decided to pursue a career only in data science, focusing on genomics, epigenomics, and transcriptomics. He continued his training doing a postdoctoral fellowship at Harvard Medical School and the Massachusetts General Hospital Cancer Center, working with renowned scientists both in bioinformatics and the cancer fields. After a year and a half, he transitioned to the biotech industry, working at Camp4 Therapeutics as a data scientist, continuing his journey of uncovering mysteries in biological data in the field of gene regulation. So it is my pleasure to welcome Dr. Goltzer. Thank you for coming.
Thank you, Laura, for that introduction. So I'm gonna go right away and jump in. So the title of my talk is From the Caribbean to Massachusetts and How NR4A1 Regulates Expression and Suppresses Replication Stress in Cancer. I'm going to use the Mass General Cancer Center and Harvard Medical School template because this project was my main project as a postdoc, although I no longer collaborate with them. So Laura already mentioned where I'm from. I'm from Venezuela, just north of Dr. Papas, who just spoke. Specifically, I grew up in a Caribbean island, and this was kind of the setting that I spent my weekends. So you can imagine that growing up here will get you drawn to the ocean and be a surfer of some kind. But when I was in high school, I didn't want to be that, not even a marine biologist. I wanted to be a geneticist. And that drove me to study biology because there was no like genetics degree per se in Venezuela. And when I started going through all the biology courses, I was more passionate about analyzing lots of genetic data over everything else, over like doing PCRs or spending time in the lab. So I was so passionate that I joined the only lab at the time there that worked with tons of genetic data. And that lab was working with this insect right here to fly. This ruler scale is a millimeter scale, so it's like roughly 5-6 millimeters long.
Dr. Bolzer, we can actually see your slides.
Oh, sorry.
No worries.
I forgot to do this. Okay. So yes, so these guys, can you see the laser pointer now? Yes.
Yes.
Yes? Okay. So this guy here, pretty small sandfly, and yeah, I had to go to the jungle, tropical jungle, to capture them. This is me under this light trap here. And the interest of these insects was that they transmit a disease parasite called Leishmania right here in this microscopic picture. So all this work in the field and the lab to extract DNA to get this. And this is Sanger sequencing, not used at least on a broader scale because we have now next generation sequencing, but I spent tons of time analyzing this data. And this kept me passionate and going into bioinformatics. So for the next steps, I went to do my PhD here in Boston and joined a lab that was kind of working with insects as well, and with genetics. And coming from this climate to this climate was quite a transition. And this is the insect that I work in during my PhD. So it's the European corn borer. You can see the caterpillar here in the stalk of the corn and the adult form here. So also extracted DNA, doing analysis, and uncovering some interesting evolutionary biology and population genetics questions. This journey more on the Q&A section. So I will go to the title of my talk, which focuses more on NF4A1 and cancer. So before I start with this story, I'm just going to go and state a disclaimer that I'm not the typical bioinformatician that you might find in some labs in the industrial or academic setting that just like, oh yeah, give me the data and I churn it out. And I don't talk to the people at the lab. I'd like to collaborate a lot with the lab and bench people and go back and forth with them, design questions, like try to get the experimental design right. So when it makes sense, when the data comes in, I have a better idea of how to analyze, and we don't have to go back and forth afterwards and then say, no, we cannot repeat the experiment. We already did it. We spent $20,000 on it. So I prefer to be in the early stages on it. And this project, I had the pleasure to collaborate with Hongshan Guo, PhD, who was the postdoc fellow at the time in this project. So another disclaimer here, this project has been recently published last year in Molecular Cell, so you can find it there. And you might find the same figures or not find the figures that I'm showing you here because they were intermediate and they work better on a slide than on a tiny space on the figure panel on the paper. So jumping right in, I should have started with cancer. So 1 in 8 women in the US will develop breast cancer specifically in her lifetime. And if it's caught early, as you can see in these bar plots, you have a pretty good chance to survive and overcome it. So the problem is that when it gets stage 3 to stage 4, it starts to do metastasis and the cancer cells can go to other tissues or organs, organs like the brain, lungs, ovaries, major organs. So we want to understand how the metastatic process works. And this gets us to circulating tumor cells. So in the early stages, when you have a primary tumor, then this tumor can shed into the bloodstream cancer cells. And these are called circulating tumor cells, or CTCs, as I'm going to call them from now on. And they can land in another tissue or organ and evolve into a secondary tumor, therefore creating what we call metastasis. So why are CTCs worth studying? Well, although you have 1 CTC per billion of normal blood cells, patients with CTC positive samples in the blood have worse prognosis. And CTCs may provide a real-time, non-invasive approach for tumor characterization and personalized therapy. So studying these CTCs can further understanding of the metastatic process and identify key drug targets to prevent it. The lab that I collaborated with, the Harvard Maheshwaran Lab at AGH HMS, has CTC derived from patients cultured in vitro. And with a multi-omics approach to understand the transcriptomic and epigenomic programs that drive these CTCs to produce secondary tumors, they started this project in 2017, 1 year prior that I joined as a bioinformatician. So this is the initial experiment model. Dr. Google injections, these CTCs that are derived from patients, for breast cancer human patients, into these mice. And of course, we have imaging with bioluminescence markers to see where they are, what other organs or tissues they land in, and they form metastasis. And they cell-sorted the cells, separating mice from humans, and in throughput sequencing, single-cell RNA-seq, ATAC-seq, ChIP-seq. This is kind of a panel image of the time progression of the mice. And they injected 2 CTC cell lines, BRX-142, so from patient 142, and then BRX-82 from another patient. And you can see that starting when you inject into the heart, it goes all over the place, but it starts to concentrate in some major organs, in this case, the ovaries, and then later also in the brain, and also at some stages of the experiment, the lungs. So we dive right in the bioinformatics of it, transcriptomics. So if you see on the right, the U map here of the transcriptomics, you see that I draw 3 clusters. And this first cluster has like the pre-injection, so cells before being injected into the bloodstream of the mice, and initial samples just after the injection, cluster here, and then in later stages, from the first 2 weeks, they cluster here. So and then afterwards, so transcriptomic programs change over time. And when we compare in volcano plots the pre-injection per we found this little guy here, this gene, NR41, which is in the title of the talk. So I will try to explain the logic of why we focus on this guy. First, you see it's highly enriched when comparing day 30 or more samples and pre-injection samples. We also looked at the chromatin accessibility using ATAC sequencing. And here you have a correlation plot of the samples. We looked at overall samples, how do they look in similar or different to each other. And you might notice that the higher correlation is among the pre-injection samples, which are highly identical here and here, depending on the cell line. And there's a huge difference between those groups and the other groups, regardless of the tissue or the day after injection. So there's a change in transcriptomics and epigenomics here. Looking at the ATAC-seq more in depth, you can see in this heat map to the left, each column represents a gene, and the color is the relative accessibility comparing pre-injection and after injection, so after day 2. And you see that the genome becomes more accessible in these genes here at the bottom of these heat maps compared to the pre-injection. And we have NR41 here and another guy that I like to call the usual suspects in this paper, FOSB, EGR1, BTG2, June. You can also see them here in this plot to the right, which is a scatter plot when we want to compare both cell lines and the pre-injection and day 30 and above accessibility. And here you have no change in the center of these plots, and when you go to the corners, you have more open chromatin, or see these genes again popping up. NR41, FOSB, FOS. What are these? These are called immediate early genes in cancer. These genes are activating transiently in response to a stimulus or stimuli, and they are standing response mechanisms activated at transcription levels before protein translation. These are around 40 genes that include NR41, FOS, FOSP, BTG2, MYC, JUN, among others. We annotated the regions that gain chromatin accessibility. And we also did chip sequencing of NR4 using an NR41 antibody, comparing pre-injection and then the post-injection ovary mets in this case, and brain metastatic cells in the case of BRX82. You might notice here this highlighted tracks, section of the tracks here. You notice that, for instance, NR41 gains accessibility, so the ataxic signal is increased on the gene, not before the promoter or the promoter proximal region, on the gene body region. It happens also in FOS and in FOSP. And the ChIP-seq also gives us NR41 binding to the same region, roughly at the same time points. And NR41 ChIP-seq on both cell lines gave us a binding at mostly gene body regions, which is kind of slightly odd if you look at the literature for most of transcription factors. Here I have two heat maps on the left of each cell line, and you notice that NR41 is at the top because that gain accessibility, it's one of the top genes that have increased gene expression after the injection. And after doing including ATAC-seq, post and pre-injection ratio, and also transcriptomics, we found the top 6 genes are these genes here, and the one with the greatest scores is NR41. So we decided to focus on what NR41 doing in the metastatic process with this CTC. What do we know about this gene? It's also known as NR77, orphan nuclear receptors of family group A, member 1. Is described to have putative ligands. These orphan receptors members remain to be identified, so not much information about it. The gene ontology often cites DNA binding transcription activity and sequence specific to DNA binding. And the latest paper at the time was Liu 2019, describing it as a key regulator in T cell dysfunction. It has paralogs, NR4A2 and NR4A3. So the first question after focusing on NR41 was, is this exclusive to CTC? So we repeated the experiment with another cell line, which is not CTC, it's also a cancer cell line. It's a triple negative cell line called NDA NBT231, which I'll call M231 for short. And basically did the same thing, done a lot of sequencing, and then plotted pre-injection versus liver. We see NR41 popping up again, gaining chromatin accessibility, exactly at the gene body. Most of the gene body targets, majority of gene body targets. So it's kind of repeatable. We also look at the gene expression, and there's all. This is not exclusive to CTC. One is to have an in vitro model to further explore this mechanism, and we compare different kind of stimuli. We try X-ray, UV, heat shock, and measure the expression of some of the M231 cells and also MCF Kinase cells, and we found that the serum extravasation model mimicked what we were seeing on the CTCs and the M231s injected into the mice. The model is basically you have cells growing in serum baseline, then you start them for 24 hours, you remove the serum, and then you replenish the serum and take samples 30 minutes, 1 hour, 3 hours, and 6 hours after replenishment. Then you do a lot of sequencing. So looking at the data here, ATAC-seq, again, the plots comparing basal to 1 hour after replenishment for both cell lines that we tried that were not CTCs, NR41 gain accessibility. So we have the same pattern here. And the only common genes that gain accessibility between the two cell lines, well, NR41 and NR43. So we stick to NR41, we look at the expression, and you see in this bar plot that the expression keeps ramping up and then going down. So NF401 also binds to gene body regions when we do ChIP-seq. Most of the regions that it binds, 77%, it's gene body, and there's no promoter regions, which is kind of odd. So comparing our NF401 ChIP-seq done from MCF10A cells here, looking at the gene FOS and gene D, and comparing E2F1, which is a transcription factor well characterized. D2F1 is binding at the promoter or just before the promoter regions, but NF401 binds to the gene body and downstream of it. We also look at the gene ontology and GSCA and found that one of the pathways that were enriched is transcription for RNA polymerase promoters. So that prompted us to look at the RNA-pol2 chip data from these samples. And we see an overlap on it. We just see sometimes the peak of the typical peak at the beginning of the promoter region of RNA-pol2, but you see that there's overlapping between the NF401 and the RNA-pol2 in the same regions on these two genes and in others. So looking at the seed maps, we found that NF401 binding precedes the high expression. We can divide the genes into group 1 and group 2, the ones that have binding at the beginning of the serum, the baseline experiment, and then at 1 hour. So those will be group 2. And just before we start the cells, we have NF401 bound to this group of genes, which has FOS, gene B, gene, immediate early genes. And after the serum starvation ends, we start seeing expression increase in these genes. So we look at these three aspects on group 1 and group 2 genes. We have NF401 protein binding here on red, then green we have gene expression, and on blue RNA-pol2. And when we have high protein binding of NF401, we have RNA-pol2 traveling ratio, which is a measure of how RNA-pol2 is transcribing normally, or its pause, is really low, which means the RNA-pol2 is paused, and transcription is low. Once NF401 is removed by the stress of starvation and then replenishment, transcription and RNA-pol2 go up. So it's kind of, NF401 is kind of, for these genes, is stopping RNA-pol2 for transcribing. And we can see that when we compare the genome tracks here. So this is kind of our model so far. NF401 is kind of blocking RNA-pol2 when we have high coverage at the gene body. And then when we remove NF401, low coverage at the gene body, RNA-pol2 transcribes normally. And we have more transcripts. We measure that with RNA-seq. And so this is kind of like putting a plug on a sink and filling it with water. The plug is NF401. You remove the plug, you have a lot of transcript, a lot of water pouring down the pipe. This effect with RNA-pol2 is also found on BRX-142 and BRX-82 on our in vivo experiments. So everything lines up so far. So wanted to do, Hongsheng managed to CRISPR one, so creating NF401 knockout cells. And we measure, again, ATAC-seq, chromatin accessibility. Almost no genes being outliers in chromatin accessibility, almost no change. Expression on the knockouts of NF401 of the immediate early genes is through the roof. It's the equivalent of when you add the serum back in the mechanism, the stress is no more. We have a high expression of NF401 on our cells. And this is how the knockout cells on this panel look like using immunofluorescence. And this picture gave us a picture, it's redundant to say, of what's happening. To see all these multiple nuclei. So the loss of NF401, the knockout, induces all genome stabilization. So the cells go haywire. So what happened to the mice if we inject NF401 knockout cells in vivo? Well, we have the M231 knockout control cells. We have this size of the tumors. Of the knockout cells, the ones that are CRISPR out, the tumors are smaller. So the tumor mass is significantly smaller once we don't have NF401 in play. And this also holds on with CTCs. Hongshan couldn't do CRISPR on the CTCs, but he managed to do shRNA constructs, so knockdowns, not knockouts. And we again see smaller tumors on the knockdowns compared to the control cells in the same organism, in the same mice. So if we do the opposite, the overexpression of NR41, it favors self-proliferation and tumorigenesis. So a construct to overexpress NR41 gives us bigger tumors. So what is the mechanism behind NR41 and RNA-2 pulsing in breast cancer metastasis? We found out using S9.6 strip qPCR that there's formation of OR loops that kind of stops RNA-2. So DRIP-seq utilized a sequence-independent but structure-specific antibody to massive parallel DNA sequencing, and we have more OR loops at the beginning when NR41 is binding, and then after the replenishment of the serum, we have less R loops. And this is confirmed also using another type of sequencing using ProSig, which measures activity of nuclear transcriptional processing on the EEGs. We have EGR1 plus B having high ProSig signals. So this is a summary of all of these projects. So under basal conditions, or NR41 is here blocking, acting as a traffic light on red, a red sign, blocking RNA-2 and creating these R loops that pauses transcription of immediate early genes. Stress to the mix. This NR41 is released, and then we have a lot of RNA-2 transcribing these immediate early genes. So when this stress signaling kind of give us, if IGs can be regulated by NR41 and it becomes adaptive proliferation, but if we do uncontrolled NR41 uncontrolled replication stress, there's DNA damage and mitotic defects, as you saw in the picture, therefore making the cancer less adaptable to our body, meaning smaller tumors. So this insight on NR41 has become a field of research in the lab after I left, because it has, you can imagine that they can have a lot of application of how to stop metastasis by controlling NR41. And with that, I'll acknowledge, of course, all the people involved in this project, mainly Hongshan Guo, Dr. Guo, who did all the amazing work at the bench and gave me terabytes and terabytes of data to have fun and analyze all of these data that I showed, and more that I didn't show, because of course there's experiments that don't yield any conclusive evidence. And of course, the Daniel Haber and Shamala Maharsora and the PIs of his lab, and Michael Lawrence, my postdoctoral mentor, which is the head of the computational lab in the MGH Cancer Center. And with that, I'll be happy to take questions from talk or for professional questions, advice, career, etc.
Great. Thank you so much, Dr. Goldsworth. That was very good. We have a few questions for you. So number one, how easy is it to transition from bioinformatics to biology or biotechnology or medicine back to bioinformatics and vice versa? Are there skills that are transferable?
Yes, I am a firm believer that we have to have a strong biology background. There are a lot of bioinformaticians that go in from computer science or even math that don't have that biology background. So having a biology background makes the transition easier. If not, if you have mainly computational skills, you'll have to make an extra effort to understand the biology behind what's going on in front of your computer monitor. So going back and forth, what some people already talked, how you're willing to learn all these tools, all these computational tools, or in the case of a computational person that wants to go into biotech and bioinformatics, how willing are you to understand how DNA replication works, how molecules work. That will give you a lot of insight and make the transition easier.
Great, thank you. What was your role in this project as a bioinformatician?
Well, I was a lead bioinformatician, so I got the data from the sequencing facility to the plots and the paper that you saw on the slides, and you can see. Of course, the project had already other bioinformaticians working tangentially, but when it started the fellowship, they gave me this, and they, especially, I remember this, Mike Lawrence told me, this is kind of a fishing expedition. We want to see what's in this massive amount of data. And the role that I play, as I say, is going back and forth with Dr. Guo to figure out what interesting questions could be asked from this experiment. So that's how I describe my role in it.
Great, thank you. I think we have time for one final question. So how useful was the experience you had working on this project for your current career?
Well, I think that so first of all the undergrad, master's, PhD gave me the basic skills to jump into this postdoctoral fellowship. But this project was basically what I think it landed me my job in industry, because I was working with insects, as you remember at the beginning of my talk. I was working with insects. So some people in the industry are like, oh, well, but you don't talk or study about humans. So they're a little bit hesitant. Once I made that jump to human, and DNA is DNA, it's the same for bases in the case of DNA, same for bases in the case of RNA, regardless of the system. But that jump and all these new technologies, I don't know if the people hearing or making the questions are familiar with ATAC-seq, PRO-seq, DRIP-seq. I wasn't familiar with those until I started with this project. So basically that gave me the skills to go into a more fast-paced environment in industry and work with the technologies that are just being developed in a matter of months, and be able to implement them into the insights that we need in the company.
Great. Well, thank you again. That was a very informative talk. I really appreciate you taking the time to come and speak with us today.
All right. Thank you for having me, Laura.
Of course. All right. So moving on, I am happy to welcome Mr. Silver Ceballos. Mr. Ceballos is a biotechnologist by profession from the Tecnológico de Costa Rica and graduate student in microbiology at the University of Costa Rica, with almost 10 years of being involved in biological sciences, working on several research projects. He is currently working at the Center for Research in Cellular and Molecular Biology, together with the Host Microorganism Symbiosis Research Group, led by Dr. Adrian Pinto-Tomas. Aware that the fourth industrial revolution is biological, he has professional interests in microbial ecology, bioprospecting, symbiosis, secondary metabolite biosynthesis, and synthetic biology. He is the co-founder of Rosalind Inventions, a STEM education company dedicated to creating tools and programs for school and college youth, teachers, and professionals in the natural and health sciences. So please join me in welcoming Mr. Ceballos.
Thank you very much, Laura, for having me here. I have the opportunity to show a little bit about our work in bioinformatics teaching. So to start, I'm going to share my screen. Could you confirm, please, that you are seeing my presentation?
Yes, we can see your presentation.
Thank you very much. So good afternoon, everybody. The title for my speaking today is Breaking Barriers for Scientific Education in Latin America. I'm going to introduce myself really quickly. As Laura said, I'm a biotechnology engineer from Tecnológico Costa Rica, and right now I'm finishing my master's degree in the University of Costa Rica, specifically at the Faculty of Microbiology, in which I work in a research group dedicated to the study of symbiosis, or interactions between insects and bacteria, specifically bacteria that are capable to produce antibiotics. Right now, I'm working with fungus-growing ants and its symbiotic bacteria, which is called Pseudonocardia. I'm also a very creative person. I loved music, like doing it for a long time, and something that characterizes myself is to how art or this creative can combine with science, and I'm going to show you how it looks like in here from Costa Rica. Also, I'm a co-founder of Rosalind Innovations, in which I develop as a lead developer. I'm in charge of designing some of the scientific programs that we have, and also about all the technical specs that comes from the challenge of designing and executing those programs in here. And to start, we are a STEM education company dedicated to promoting science in youth through experimental STEM programs, completely 100% experimental. These programs are focused to high school students, for example, and also university students. We have a few experience with kids also. And before the pandemic starts, we were developing a lot of workshops in which we engage youth students with, for example, activities such as isolating microbes or doing experiments with microbes and also with fungi. But then the pandemic starts, and as you all know, we have like a lot of trouble with presentiality. So basically, our model just got disrupted. We no longer have the availability of young students or professionals also. So we have to transition for a more virtual program that works for us and also for the persons. And here we come to with idea to develop a scientific program that is dedicated to teach bioinformatics for public of Costa Rica primarily, but we are looking to expand it into other countries as in Latin America as well. And I'm going to share with you some of the background and also challenges that we have in our country, which I hope that many of you also could familiarize with those challenges. First of all, bioinformatics is not a new field. We all know that. It's a field that has more than 20 years in developing, also aided by the emerging of new sequencing technologies and computational capacity. But it feels very new to us. We, for example, many bachelors in life science here do not have bioinformatic courses in the programs. And if they have just one course. Also, there is a lack of well-trained professors in bioinformatics, and there are very few researchers that actually does or employ bioinformatics in their research. Also, both teaching and learning bioinformatics could be very difficult, especially because of some of the backgrounds topics that are employed to teach bioinformatics, and concepts in cell biology or biochemistry and molecular biology could be very distant to many early students, which do not have a proper background. So basically, we have like this idea to design a program to engage beginners and early stage students and professionals with bioinformatics. In a way that it would be like a first step that could derive in more learning opportunities, maybe, for example, from platforms such as Coursera, or maybe some international programs that are also available. But we want to focus in here with these public, just beginners and early stage students. So we have like various challenges that we need to overcome in order to make like these persons not only to like bioinformatics but also to understand it and want to continue to pursue like knowledge in this field. So we designed a program that is basically conformed of 4 things. First of all, is synchronic sessions in which we have like a masterclass. We view some concepts, review tools. We also view case studies and some analysis that we do with those tools. We also engage with activities and some homeworks in which we have extracurricular activities dedicated to strengthen some of the techniques or maybe some of the activities that we already see. We have lectures also to expand knowledge. We have evaluations, and we have also a mentoring style in which the participants are allowed to make questions, and we deliver a very quick answers to them to guide them through our program. And also, we use Google Classroom as a platform to unify all this. Unify, for example, like the classes, the videos, the extracurricular activities, the lectures, and it had given us like very good results with the public that engage in our program. Also, there are some things that we wanted to make to introduce in our program because of personal experience with these tools in the past, but also because of their potential. And I want to introduce Benchling because it's like a cloud-based informatics platform for life science, and it's a very good one. I have been following Benchling since its start. I don't know, it's like 8 years ago, maybe. But it's great, all the capacities that it has right now and the upgrades that have been also introduced. It's great for managing, for example, biological data. You could have like folders in here and also upload a lot of biological sequence, and you can visualize very easily, create multiple sequence alignments, and it has other features that works very well as preliminary efforts. And also we have like some online bioinformatics tools, such as the French portal of phylogeny, in which we have like tools for trimming or doing multiple sequence alignments, and also to create phylogenetic trees, which are very adequate for our purpose of teaching bioinformatics. And also we employ this third open tool, which is ITIL, or the Tree of Life, International Tree of Life, for visualization of phylogenetic trees. So basically we are like combining different tools to employ these programs. Google Classroom for the resources, but also Benchling for the managing of all the data that is given to the students, and also a set of tools that are publicly and anyone can use them. They're open source, and also these tools that is well known and used in the visualization of phylogenetic data. So I'm going to give you right now like a really quick overview with topics and the case studies that we use to teach bioinformatics, which I hope that could inspire many other initiatives. This and also, so it has worked really great for us. So first of all, like we have this program in space of 4 sessions. The session number 1 is pretty focused on databases and management of biological data. We use a case study, which is the virus that is the cause of the pandemic. And also, what we do pretty basically is to explore mutations on the spike protein. So NCBI has a really great portal to look on information about this SARS-CoV-2 virus, and we focus specifically on spike sequences, in which we review a question such as what is bioinformatics, what a biological database, and what is the virus and how does it look like. So basically we obtain a lot of sequence from this virus that are publicly available and comes from different parts of the world: Latin America, United States, Europe, Africa, Asia, also Australia. And we can use those data to explore mutations, and we can upload the data to Benchling. We can very quickly observe its sequence, a lot of residues, and also identify mutations in here. So that is like our first approach to the use of benchling. Prevalence is it's a basic but very robust skill that we teach in this program, how to get all the advantage of a platform such as benchling. We then have like a second session, which is called genetics and evolution. And we in here we start by doing some more advanced analysis. In here we try to obtain sequence. We focus primarily on the searching of sequence in databases, how you obtain those sequence, you upload them, and you manage them in folders. But in here, we advance a step further in, for example, in constructing multiple sequence alignments and how to visualize those alignments. And we use a case study based on foodborne pathogens. And why foodborne pathogens? Because it's really easy to maybe understand the issue here. I think well, it's a very important issue around the world, but also I think everybody has one in life, like an experience with diarrhea. And also we explored that this thing that diarrhea is like a very critical disease around the world, and it's caused by bacteria. So we try to also to explore further these microbes that are cause diarrhea, such as the coliforms, for example. So we use questions such as how to construct a database that we can explore later, how to, or what is like also multiple sequence alignment, how we do that, and what is the evolutionary origins of coliforms, those bacteria that are well known for causing disease and are well transmitted in food. So basically, we try to also to obtain a lot of sequences from coliforms, from the gene bank database. We also construct and curate a database, and we engage in activities such as constructing multiple sequence alignments in MAFT, which is a pretty famous tool, and also to start looking at preliminary phylogeny. So in here, we also try to expand, on the relationships of Escherichia and Shigella, for example, which are two species that causes some debate because some microbiologists believe that they are one species that just have like a speciation event, and other microbiologists tend to argument that they are different species. And also to look at other types of bacteria that are pretty interesting, just as Yersinia pestis, which is a pandemic bacteria in the past. And we engage in here, like doing some starting phylogeny, like starting to grasp concepts about how phylogeny works and how to also interpret phylogenetic trees. And this case also has like a really cool activity in which the students try to obtain different sequences from other organisms and try to include those sequences into the database, try to reconstruct again multiple sequence alignment, try to reconstruct another phylogenetic tree, and then compare the new phylogenetic tree with the older one to see how does it differ and what does it happen, what does happen in the analysis. And we have two other sessions dedicated exclusively to explore phylogenetics a little bit more. And for this, we use a model organism that is called Clostridium difficilis for a variety of reasons. First of all, because it is a very important pathogen here in Costa Rica. And also it's internationally is well known as a model of organism because it produces various toxins which play a big role in pathology. And also, well, in countries such as United States, like it is a very big problem, not only because of the fatalities of the disease, but also because of the recurrence, which produces itself in an economic cost. So we explored questions such as how the toxins look like from a phylogenetics perspective, or are toxins from Clostridium difficile derived from Costa Rica are similar to other toxins from isolates around the world. And also in here, we have a pretty interesting feature in which we employ genomes from Costa Rican isolates that have been isolated from an outbreak that happened into like 2010, around 2010. And a lot of from those isolates, like a few of them got sequenced, and the data is like publicly available. So this is something that we want to like some kind of signature to employ sequences that are derived from our country. Not also because it is like important, it's publicly funded by the state, but also because it is very important for us to engage in science outreach and to let people know about some efforts that are done in some of our universities. So we employ genome data from these Clostridium difficile isolates to look for toxin sequences and then managing those in benchling, for example, but also use them to construct phylogenies of the toxins. And we also include a database, like a basic database with reference organisms such as outgroups in order to improve our phylogeny. So this could be like a very good figure of how the phylogeny of these toxins look like, in which isolates of Costa Rica tend to group in these clades and these ones here. And and also in the last session, we tried to create a typification of Clostridium difficile using multiple sequence alignment, which is a very common technique used in epidemiology. And so we basically teach the students to how to obtain housekeeping gene sequence from genomes and how to use them to construct multiple to construct the sequences that are employed later in this type of analysis. And we try to answer if these genomes, the phylogeny of these genomes using MLST looks similar to the toxin phylogeny, which we already know that we have differences. The phylogeny, the genome phylogeny is very it's very similar, the isolates, but the toxins of those isolates is very different. So it's a very nice result to illustrate how some features of genomes, such as toxins, have a different evolutionary path as the genome or the general chromosome. It has different evolutionary pressures, and that is something that is really, really interesting to learn. So some conclusion about our experience is that we have approximately like 1 year to teach this program. More than 50 students have passed through Let's Make Bioinformatics. Also, we try to offer this program to university students and professionals from different countries, which are interested, for example, in pursuing master degrees. And also, as the professor or instructor of this program, we have seen that the implementation of case studies and model organisms improve both teaching but also learning. Like it's more concrete to illustrate some how bioinformatic analysis can be employed in different cases scenarios in real life. And also, well, always the alumni share like a desire to continue learning bioinformatics, so it's like a good sign for us. So that's like the summary of this program that we share with you, and also that is continually offered to students here in Costa Rica. So thank you very much for your time.
Great. Thank you so much, Mr. Ceballos. That was a very informative talk. All right, we have a few questions for you. Number one, with so much antimicrobial resistance happening because of the overuse of antibiotics, do you think bioinformatics can be useful to track and overcome this challenge?
Yes. First of all, I think it's like a global phenomenon. We are starting to obtain isolates that have resistance. Also, we have, as a country, we have different efforts to survey for microbials that present resistance. Also, well, I personally might participate in a big research project that is focus on discovering novel antibiotics. Basically, we have a very big bioprospection operation that isolate and obtain microbes from insects and then test them against a panel of clinical pathogens in order to look for interesting isolates that are then transferred to a university in the United States. So it is like a great necessity and all around the world to not only to survey for resistant pathogens, but also to look for novel sources of antibiotics. And what was the second question? Excuse me.
Yeah, so the second question is, what are some of the ways that students from South America can find mentors that can expand their horizons and help them gain insights into specialized topics like bioinformatics?
So some of these bioinformatics analyses are well known or applied to both this case of antimicrobial resistance, but also in epidemiology, in research also. I believe universities are making an effort to include bioinformatics in their programs, but it's a long way to get to that. So I believe, for example, one of the purpose of this course is just to bring a starting point. And I think that is very important if you want to engage in bioinformatics, like to have a good starting point in which you like can have like a little grasp of the concepts that are, or the concepts or the techniques or the tools that are taught in bioinformatics, so you could advance in other types of programs. And I think programs such as the ones offered in Coursera are like a very good second stage to explore like more different tools, and also start in coding, which is like the final I think it's the final objective. I myself do bioinformatic analysis in clusters and use like the coding stuff. But if you are an early stage student, like it's going to, maybe you're going to get afraid of the coding, and maybe that also it turns you down. So that's like the barrier that we are trying to break. That it's coding is not I know it's kind to be difficult, and you have time and a lot of practice, but it's easier to start coding if you have like a good grasp of what's going on.
Definitely, yes. Thank you.
Thank
All
you,
right.
Laura.
Well, thank you again for speaking. That was a very informative and wonderful talk that you just gave.
Thank you very much, and all the persons that are here in the symposium. I hope you enjoy all the lectures.
Definitely. Thank you again. All right. So it is my pleasure to announce our next guest speaker, Mr. Piero Barangasco. Mr. Barangasco has a bachelor's degree in bioengineering from the Utah University of Engineering and Technology. He's very passionate about life sciences with a multidisciplinary approach. He is interested in scientific research about synthetic biology and its application in the environment and space travel, currently working in fungal biotechnology and part of the IRMA, an astrobiology project that will fly to the Tiangong space station at the end of 2022. He was also selected as one of the 100 leaders in biotechnology in Latin America by All Biotech. So please join me in welcoming Mr. Baran.
Thank you, Laura. I will share my screen. Sorry about that. Okay. Can you see my screen?
Yes, we can.
Okay. So this talk is a little about my previous experience and also how I ended up teaching bioinformatics and computational biology. So the title of this talk is Computational Biology and its Applications, Education and Training in Latin America. So let's first start with the beginning of my journey into computational biology. So I was a bioengineering student in Biotech University in Peru. So as part of my courses, I was in first contact with bioinformatics. During my first year of undergraduate students, as I didn't have access to a laboratory, I was doing some coding in Python, like trying to simulate some experiments, doing comparison between DNA sequences, basically learning. So after this first year, then I switched to wet lab. So I was with no contact with bioinformatics until I started an internship at Vanderbilt University. So it was in the year 2020. So when I was in the middle of this internship in Vanderbilt, all this situation with COVID-19 started. So the university was shut down, and I basically had to start working from home. So at this point is when my point of view in biology, biotechnology in general changed from not only doing wet lab, but also doing bioinformatics. So I will try to explain how it was this journey. So what I was doing at Vanderbilt was investigating the role of STK38 kinase in Wnt signaling. So the way it works is when there is no Wnt signal in the environment, what happens inside the cell is that a protein called beta-catenin is phosphorylated, but this phosphorylation complex in which the dominant protein is this one, APC. So once beta-catenin is phosphorylated, it quickly degrades. But when there is a Wnt signal, this Wnt signal fused with this Frisled protein, and Frisled kidnaps this phosphorylation complex. So beta-catenin survives, and then beta-catenins switch into the nucleus of the cell and can turn on some Wnt target genes. So what they saw before I came to Vanderbilt was that the response after a Wnt signal is directly correlated with the amount of STK38 that is present inside the cell. So the wild type has normal levels of Wnt response, but when you increase the amount of STK38, there is also an increase in the response to the Wnt signal, and in the opposite, when there's a decrease in the amount of STK38, there is also a decrease in the response after a Wnt signal. So they wanted to know how is STK38 working. They actually figure out that STK38 is doing something downstream the phosphorylation complex. So it's in this part of the pathway. So as I'm a bioengineer, so I basically design new technologies, new biotechnologies, and this is a research in basic science. What I did was working on synthetic biology and designing and testing a synthetic gene circuit to perform a knockdown on STK38, a programmable knockdown. So what I did was a CRISPR system, a synthetic circuit that uses CRISPR protein, a modified Cas9 protein called dCas9 that cannot cut the DNA, but it can bind to it using the guide RNA. So the way this system works is with the this protein LTTA, an inducible protein with tetracycline or doxycycline. So in normal cases, the system is off, so there is no production of the dCas9 protein. But when there is a signal, tetracycline in the medium, RTTA can bind to this promoter that is a constitutive promoter with, an inducible promoter with a TET-O operon. So RTTA can bind to this promoter and then activate the production of this dCas9 protein that is fused to a CRAB domain. This CRAB domain is a repressor domain. So this dCas9 acts as a repressor for our target gene, in this case, STK38. So controlling the amount of tetracycline that is in the medium, we can control the amount of STK38 that is being produced inside the cell. So this was basically my work before the pandemic started. Then when I had to work from home, the first question I had was how to continue my work from home and how can I learn computational biology since it was all what I can do. So by this time, I didn't know anything about computational biology. So I had to ask some friends I had on two Peruvian communities, the Society of the Peruvian Society of Bioinformatics and the local group of the International Society for Computational Biology. So they told me about how can I perform bioinformatics in order to continue my research in home. So the things I was doing was basically protein docking. So I first tried to improve my skills doing some docking with proteins that I knew that actually docks inside the cells, that are GSK3 and beta-catenin, because in this complex, the protein that performs the phosphorylation is GSK3. So it worked, and it also shows that the active site of GSK3, that is painted in red, is in contact with the phosphorylation site of beta-catenin. So I decided to continue and test STK38 with all the proteins that are downstream this phosphorylation process. And what I found out was a really strong interaction between STK38 and beta-catenin in silico. So one of the hypotheses my PI had was that STK38 was not acting as a kinase, but helping beta-catenin to switch from the cytoplasm to the nucleus. So I painted in red the active site of STK38 that is marked with this lysine 118, and it shows that this contact region is far from the active site of the kinase. So in silico, it shows that STK38 is not phosphorylating beta-catenin. So it's like a step more into proving this hypothesis that my PI had. So after this experience, I was really interested in computational biology. So when I returned to Peru, I took my first steps into protein engineering. I joined a project between my university and Cleveland Clinic in the USA. So it all started as a synthetic biology project. We want to produce synthetic extracellular matrix. So we have two systems that we wanted to test. One is using collagen and spike capture system. The way it works is we use two input signals, IPTG and AHL. And these input signals activates the production of one of these proteins. IPTG controls collagen spike tag protein, and AHL controls collagen spike capture protein. So we can control the amount of both proteins that are produced. And spike tag and spike capture really want to meet each other. So they basically bind and fuse. So we can create a giant matrix of a lot of collagen proteins that are bind together. And the other system is kind of similar, but it uses elastin. So as we were still in a pandemic, the first work was done in using bioinformatics. So I was in charge of that part by doing protein engineering. So I was modeling some collagen's protein here. As collagen are three coiled chains of collagen peptide. So this is a normal collagen protein simulated, and this is collagen after simulating it with a spike tag signal at the end. So it kind of denaturalized the structure, but it's still together. So maybe it will work in the laboratory. And there are no laboratory tests until these days, but we want. They are expected to occur soon. So after this experience, last year I started my IGM journey by joining and founding an IGM team called Btec, and it was my best research experience. It was really complete. I did a lot of synthetic biology and also a lot of computational biology that I want to show you. So the first thing was, well, let me explain a little bit about the problem. So in Peru, we have a lot of losses in the food chain production. So nearly half of all the food that is produced in my country actually go to waste. So we wanted to solve this problem by producing different proteins that has antioxidant and antifungal activity. So we first designed this protein by doing some research, reading some papers, and selected different parts, different domains of interest, and mixing them into a lot of possible protein candidates. So after this, we simulate them. We use a lot of softwares that uses artificial intelligence, de novo protein simulation. So we obtain these models. These are the best models. This was performed with Modfold. And also we did some docking, so we can actually test if the antifungal proteins bind to the chitin, that is this molecule that is in the cell wall of fungal cells. So with this, we then design the expression cassettes that we will use in our project. We won this competition, and now we are going to Boston, to Ginkgo Bioworks, in order to continue with the experimental part of this project. But it didn't stop there, because as you know, I'm an engineer. So I always thought about engineering problems. And what I asked myself was, how can I study the expression of these synthetic genes? How can I put numbers into DNA sequences? So I researched about genetic circuit modeling and systems biology. So what I did is the engineering behind this project. So the basic way of modeling gene expression is by modeling it in two steps. The first one is transcription, and the second one is translation. So this simple model only works with cell-free systems because cell-free systems only has these 2 steps and also are free of stochastic problems that are inside the cell, because as you know, in cells, there are normally just 1 copy of the genes. So in cell-free systems, we have multiple copies of the genes without the problems that carries using alive cells, but with all the benefits. So this was a normal modeling, but I decided to go 1 step forward and actually applying engineering tools to analyze these 2 simple equations. So I did a system analysis by applying a Laplace transform to this equation. So I need an input and I need an output. The output is obviously the protein production, but the input, I need to create one because there is no one here. So I create this one called, let's say, the opening of the gene. So it basically has a value of 1. So it's a step response of 1 when the system is activated. And then I can perform the system analysis. So doing Laplace transform and isolating in one side the output and the input, we finally reach this final equation in which we can observe that the protein production over the input of the system has this transfer function with 2 poles here and no zeros in the numerator. So then we can, this allow us to do a dynamic analysis of a system. So we are not only plotting a curve to match some point we obtain in experiments in the laboratory, but we can also make a dynamic analysis. So these transfer functions not only works with stem responses, but with whatever input we have. So we can change like this function here, not only for a step response, but for example, when there is an inductor or a repressor present in the system, this value can change. So we can actually use the transfer function to predict the behavior of the gene expression by only taking the input, right? So we don't have to create new equations. We don't have to repeat the same experiments in the lab. We just need to run 1 experiment in cell-free system to obtain this dynamical constant of the system. And then we can predict the behavior of the gene with whatever input you want to test here. So for example, this is a graphic I extracted from one paper, this one. It's from a cell-free gene expression. So the graphs are actually really smooth. There are no stochastic processes here, like in whole cell genetic experiments. So I can extract the data with simple code. So I can extract this curve of this graphic, and I can perform this dynamical analysis and obtain the kinetic values of the constant. So what we wanted to do is to do this when we will be in Boston, so we can test how our genes are behaving and try to predict how they will behave when doing all the escalation of the process and producing this chimeric protein. And also I teach myself a little bit more of system biology, so we could also model other processes in our project, like how the presence of 4 antifungal proteins can affect the growth curve of fungal cells, in this case penicillium. So this graph actually uses real values, real experimental values of these curves of fungal growth. So we can predict, at least in silico, how our system will behave, how our chimeric proteins will kill the fungal growth. And we can also find this value, 1 micromolar of antifungal protein that can completely block the growth of these fungal cells. So now, like 4 years ago, I joined the DIY bio movement, the democratization of biotechnology. So everything I learned, I really love to teach. And as I was learning bioinformatics and computational biology, I wanted also to teach it to more people. So I was called by a startup that was doing this, that was teaching some classes in biology and biotechnology, using a lot of professors. I joined this project, even though I didn't even have a bachelor degree by this time. But well, I decided to first teach about synthetic biology and how you can design synthetic biology systems. Then I decided to also teach bioinformatics and how to model mathematically biological systems. So it was a really good experience because, as you know, when you search for information in bioinformatics, it's usually all in English. So this was all taught in Spanish. And in Latin America, it was good because not all students in Latin America know how to speak in English. So this course has more broad audiences. And what really surprised me is that my classes were focusing on undergraduate students, but nearly half of them were in graduate programs. So it was nice to know that graduate students really wanted what I was teaching, because bioinformatics is not usually taught here in Latin America, or it's not usually taught in Spanish. So it was a really great experience. They learned, I also learned, because I had to practice a lot in order to taught these classes. And finally, what I'm currently doing is applying all what I learned and all what I taught in astrobiology. So the last project I joined is IRMA project, or IRMA. It's a joint project between the Mars Society Peru chapter, the Mars Society España, and with the help of the United Nations Office for Outer Space Affairs and the Chinese Manned Space Station. So the problems we want to address here are two. First, there is high launch costs when you want to run biological experiments in space. So there are a lot of economic challenges to perform research in space. And there is also a very limited capacity for experimental instrumentation in the space stations, not only in the International Space Station, but also in the Chinese Space Station. So there is also a lack of equipment. To solve these problems, there are also some things you can do here on Earth on normal gravity. So you can actually simulate low gravity, but it's not too accurate as using real space. So we decided to solve this problem by designing a complete autonomous system that can run biological experiments in space without people in a small space and send the results back to Earth. So this is IRMA, or Instrument for Recording Microbial Activity. The first test we will do in the Chinese Space Station is to analyze the growth and the differences in gene expression in two model bacteria. The first one is E. coli, that is the model bacteria for growth, and Pseudomonas aeruginosa, that is our model for biofilm formation. So these two are pathogenic bacteria. So we have a concern with them as they are also, they can affect the health of the astronauts in future space missions. And as they are also model bacteria, we can actually do a good research about them. The previous research that has been done in space with them that have been done in liquid cultures. So we are using solid cultures that represents well the medium inside our guts because they are a solid surface in which the bacteria can grow. So I am currently working and in charge of the biological part of this project. So what we're going to do in that biological part is first planning the RNA seq analysis of these two bacteria, the E. coli and Pseudomonas aeruginosa. We are only focusing on pathogenicity islands because we want to know the differences in the gene expression of these genes that can increase pathogenicity in these bacteria. And we are also doing some simulation and training of the cameras that will be inside IRMA because as we want to analyze the growth of this bacteria, we need to train the cameras to detect the shape of the colonies and to measure the velocity of growth of these colonies. So what you are seeing here are stochastic simulations of how bacteria are growing in a solid surface. And we are also sending to the Chinese space station a strain of E. coli that is genetically modified. So it has a synthetic genetic circuit that takes arabinose as input and GFP as output. So we want to know how microgravity changes the potential of synthetic biology in space. So we will be also using this tool I showed you before, the dynamical analysis of gene expression, to measure how microgravity can change these dynamical values of the gene expression inside a cell. And finally, we are actually writing a manuscript on how to use COG database. COG is cluster of orthologous genes. So it compares the sequences that encodes the same genes but in different species. And we want to understand the thermophilic archaea evolution. So we are using these COG databases in order to analyze which genes are preferred to evolve or to change over time when archaea were developing to withstand different harsh conditions here in Earth. So I cannot show you results of this project because the manuscript is not already published, but we can talk about this project. So if you have any question or any commentary about this presentation, now is the time.
Great, thank you so much, Mr. Baran. That talk was very informative and very interesting. Thank you again. I think we have time for one question. Do you think that you would have gotten interested in bioinformatics if the pandemic never happened?
If the pandemic never happened, maybe I will still be only doing wet lab work. The way I switched to bioinformatics was this critical point, the shutdown of all the universities and public spaces. So it changed my life, and it changed my life for good. And most of my friends that are currently working in bioinformatics actually switched to bioinformatics thanks to the pandemic. So I think even though the pandemic was bad for the world, for economics and everything, it was actually good for research.
Definitely, that's a very interesting perspective. Okay, well, thank you again for speaking with us today. I'm going to go ahead and introduce the next discussion. So next we have a group discussion from a group of panelists. So here we have Mr. Elia Brodsky, the co-founder and CEO of Pine Biotech. We also have Dr. Raja Guthrakuta, a professor of astronomy and astrophysics at UC Santa Cruz. Dr. Japoliano Chavez, a postdoctoral scholar at the University of Chicago. And Miss Maria Victoria Luz, a bioinformatician from the University of Brasilia. So they are going to be discussing the SIP program, how they all got involved in the field of bioinformatics, and if they have any advice for new bioinformaticians. So please welcome.
And thank you, Laura. Before we jump in, I know that Raja, you're going to introduce us to the SIP program, but before we jump into that, I just wanted to highlight a few takeaways so far from all of these wonderful presentations. So in the scope of today, we heard from people in India, in Africa, in South America, in the US, people that come from life sciences as well as computer sciences. And we heard how there is a common challenge of introducing, because this topic is kind of central to our symposium, introducing bioinformatics at an early stage. Many people don't realize the significance early on, but later on, as they continue to pursue their research interests, they kind of rediscover the significance of this skill set and go on to apply it to a variety of topics. We saw people that apply it to studying insects, studying plants, bacteria, eventually pursuing cancer, biotechnology, big data, cloud computing. So it seems like the trajectories are very unexpected. But what they all share in common is that early exposure to bioinformatics, appreciation of the role of data in biology, and eventually learning how to utilize these skills to pursue what they really enjoy, what they love, what they care about. So that really emerged to me as a common trend. Another important trend that I saw was really the diversity of what we call bioinformatics. I mean, it ranges from cloud computing, like I mentioned, all the way to mathematical modeling, like we just heard right now, to analyzing kind of simple data and very, very complex data, right? So it's really a term that encompasses a diverse set of directions and challenges that it seeks to address. And my final takeaway from this is that there are so many efforts to try to introduce these topics early on. And a major question that arises for me is, yes, we are here because we're all interested in bioinformatics. What about people that maybe are outside of the circle? You know, people that don't necessarily, tend to enjoy research or pursue research. You know, how do they get exposed to these things? And how do we make that early interaction fun, meaningful, and leading to something effective later on? So with that summary, Raja, over to you. Let me just make sure that you can unmute yourselves. There you go.
Oh, thank you. Thank you very much. I know Laura mentioned that I have about 12 minutes to speak and a couple of minutes of Q&A after that. I have a slide set. Thank you for inviting me here. I'm an astronomy professor at UC Santa Cruz. What am I doing in a Biotechnology Research Symposium? Let me explain. I got connected to Gio Chavez, who was a grad student at UC Santa Cruz when we got connected, because we were involving high school students in our research. Gio connected me to Elia, Pine Biotech, and to this Omics Research Symposium 2022. I have a few slides to share with you. So let me see if I can share my screen. There we go. Are you able to see my slides now?
Yes, we are.
Okay, thank you. I will talk not so much about SIP, but the CREST umbrella of programs and initiatives, and I'll tell you what CREST is in just a moment. I'm a professor at UC Santa Cruz. I have the misfortune of being department chair. I can't wait for that to end. And I've been running the science internship program for high school students. And 7 years ago, I spent 8 months at Google, and they really helped me scale up some of the outreach work I was doing. This work has been done in collaboration with my colleagues at UCSC, Sarah Schneider, Christy Tall, and Carolyn Roosevelt. What I'll talk about in a moment is CREST, which stands for Creating Equity in STEM. Santa Cruz is a beach town. I hope you can see the shape of the crest of a wave here, but it's also the letter CREST. And it's an umbrella that is, that sort of brings coherence to a range of things we're doing here at Santa Cruz. And I'm really grateful to you for allowing me to share this. This is, our bold title is Creating Equity in STEAM, where STEAM is science, technology, engineering, art, and mathematics. Are we going to create equity in my lifetime or yours? Probably not. Are we going to create equity with one person working on this? Probably not. But the whole idea is for us to join hands, And that's what this symposium is about. Join hands, work together, and work towards this thing. This is a bright light in the distance, but it can guide us in our work. And that's what I'm hoping to talk about. Why do this? There are folks from India on this call, so I thought I would mention something about the Indian cricket team. I'm a huge cricket fan, and the Indian cricket team in the 60s and 70s, when I was following them, were constantly losing. In Brazil, on the other hand, the Brazilian soccer team was constantly winning. There was some difference in the formulae. The Indian cricket team, when after every loss, the captain or the best player on the team, the person who had put in their best performance, would be interviewed by the television or newspaper in those days. And even though they had all grown up in India, they spoke the Queen's English. They spoke perfect English, growing up in India, playing cricket. Now, there's something wrong with that picture, right? Because you don't need to speak English with a perfect accent in order, the ability to do that and the ability to fling your body into the air and pluck the ball out of thin air with your hands, or to good hand-eye coordination, that has nothing to do with the ability to speak English. So this changed for the Indian cricket team in 1983, where for the first time they had a captain who could not speak a single sentence of English. He was an embarrassment in front of the camera and the television. They had translators for him to translate from his native language, his native Indian language, which was Punjabi, to English. But guess what? The Indian cricket team won the World Cup in 1983 with this captain. I don't think this is a coincidence, right? When you open your talent pool so that you're not just selecting people who can speak perfect English and they're part of an elite section of Indian society. When you open your talent pool wider than that, you're going to get better talent. And the Brazilian soccer team realized this a long time ago. If you look at almost every Brazilian soccer star has not been from an elite background. Almost all of them. Very few have been from elite backgrounds. So diversity and excellence are not two separate things. They go hand in hand. Other thing I realize is we are working to diversify our institutions, but that requires working outside the institutions. We can't just expect that people who will apply to our institutions as undergraduates, graduate students, or faculty are going to be representative of society around us if we don't do the groundwork, if we don't bring ourselves, our university work, to the outside world. And this is true for corporate organizations as well. We have to work outside to diversify the inside. And I want to say that we are going through a pandemic, COVID-19, but we're also in an information pandemic where the value of evidence-based thinking, the value of the notion of expertise has really eroded. And while there's a vaccination, I just took my 4th shot yesterday, my Pfizer shot for COVID-19, and they give it to the elderly first, because that's who's at risk. For the information pandemic, where we've eroded critical thinking, the people who are most at risk are the young people, because they're going to live with this disease the longest. So we need to address that, and programs like these, like Gio and I are involved in, are the vaccination for young people. And it's a long game. It's not a short game. It's not something that's going to address the immediate needs of the community. It's not going to immediately address hunger or domestic abuse, poverty. It's not going to address those things right away, but education is a long game. Okay, with that, I'll get off my soapbox and talk a little bit about like a pattern I see. When young people are going to engage in STEAM, the most logical thing for them to do is first gain access to the people and the process. And I've underlined people and process here, really, because very often when science is reported, you see the gee whiz results. You don't see the many things that fail to get to that. So people and process are important. For me, that access happened by going to a planetarium as a kid, or by watching David Attenborough's Life on Earth. Then you need to build the skills, and then you can apply them in a controlled setting where the mentor has already gone down a path and they send other students down the same path, where they can control the amount of instruction they give them. And then finally, there's open-ended research. And the thing that Gio and I worked together on in SIP involved open-ended research. By open-ended, I mean the mentor has not solved the problem before. There's no recipe to solve the problem. And you're engaging young people in that process of learning from productive failure. So creating equity in STEAM is something we're trying to do. And the words that run across are what we're trying to achieve. And the words that run up and down, critical thinking in the context of open-ended research under close mentoring is how we're trying to achieve it. So the goal and the method, goal across, method down. So we first want to encourage young people to dip their toes in the water. This is the access piece. And I've been running something called Shadow the Scientist. Now when we get time on, I'm an astronomer, so I use telescopes in Mauna Kea, telescopes in California. Instead of going to the telescopes, we can't go to the telescopes anymore because of the pandemic. What we do is operate the telescopes from home using VNC, using Zoom to connect people. And what we've started doing is encouraging young people to join our Zoom call to see how we work. This is dipping your toe in the water. This is seeing, do I like the temperature of the water? Do I like the people who are already in the water? And then if you like this, then you have to learn the swim strokes. And this is something we've been running a Python and research tutorial where you learn the techniques. Geo has started this wonderful R in the context of research in computational biology. The idea is to teach young people the tools, the computational tools, and really embedding this in research. What we do here is we have code that's directly relevant for a science project, and you can remove lines of code and ask the student to write them back in to make the code work again. And what you're doing there is you're teaching them both about coding, but also about the application of that coding. Once you have this, then you're ready to take a deep dive. That's what the science internship program is. It's open-ended research, 10 weeks. Today is our application deadline for our high school students. So it's too late if high school students are interested. Today is the deadline, but we have over 2,000 applicants. We have a second program that's in collaboration with Professor Annette Lee at Arizona State University. That's at the intersection of art, science, and culture. It's a different kind of deep dive, not just into one science, but into an intersectional area. Then once you do this program, after this plunge, what do? We have a network called the Sphere Network, which stands for STEM Programs for High Schoolers Engaging in Research Early. It's a way to connect programs so that we're not each reinventing the wheel. It's a way to create a community of practice for mentors who engage high school students in research. We have an Alumni Buddies program, so near peer mentoring. We follow the trajectories of our interns. We offer this to teachers in the science internship program. That's what TSIP is, so that they can take this work and spread it to their students in schools. We have a peer-to-peer college and career network, and we have community college students in SIP. So that's the end of my slides. That's my 12 minutes. I'll stop here and see if there are any questions. Thank you.
Okay, I actually do have a couple of questions. So the first one is, what is that critical thing that can lead a student to go from following a tutorial to starting with open-ended research?
I can tell you what I've found, but others should chime in. I found the first time I was doing research was when I was a grad student. But for younger people, for high school students and college students, what I've learned is the first time they're participating, when they feel like they're, if not equal partners, but at least partners in the enterprise. When the mentor turns to them and say, what do you think of this part? Why are we seeing this trend? That's a critical moment. That's a critical moment where you know that you're not just doing this for someone else. You're being asked to think about something. You're close to an equal participant in this. That's what our students have told us. That when we felt like we were participating, but in a way that where our opinion was valued as a scientist or engineer, that tipped the scales for us. That gave us the confidence to really feel like we have ownership.
I think, in other words, Raja, a good way to put it is the feeling of community, feeling of being part of something. Maybe my way of putting it is to feeling that you're part of something bigger. For instance, the one interest that I have that motivated me for doing this type of research that I do is the possibility of caring people, of helping people get cured for the diseases that they face. And so that's my motivation. That's the one thing that makes me never give up on the drawbacks that I kind of face daily on a day-to-day situation. because the result of the effort that is going to be put is going to be much better than the whatever is costing. So I think that from the beginning, not from right now, I think the opportunity that I had on SIP, and that causes me to strongly appreciate that, is the fact that this experience of belonging that I have as a mentor can be shared with the students that come to join the program. And because of the philosophy of SIP, which is in my mind is a thing that you have taught us, you have pioneered, because of the philosophy of SIP, that becomes something that is offered to the students. I mean, this mindset, the possibility of feeling the belonging to the reasons, to the cause of research, is passed on to the students very clearly, in my opinion.
Maria, did you have any? Sorry,
yeah, please go ahead,
Maria.
Maria, please go ahead.
Yeah, and I'll just briefly mention that Maria is a student. I think, can you unmute yourself, Maria? Yeah. Oh.
Hi, everyone. I am Maria, and I'm a student of biotechnology in Brazil at University of Brasilia, and nice to meet you all. And I will put my presentation here right now.
One second, Maria, we will get to your presentation. I was wondering if you just had anything to add about, how does a student go from just learning, doing what I'm told, to participating in some research? What do you think?
Oh, my experience was very different because I had classes at university about bioinformatics, but it wasn't my focus at all. I wanted to study plant sciences and everything, but then I started to compete with synthetic biology, and I faced this challenge to learn by myself all the tools that I haven't learned at university. And it was a very interesting moment, of doing things by myself and going to the internet and researching. And then I tried to talk to professors that are experts in the area, but it was a very challenging experience in bioinformatics. And it's still hard for me.
Yeah, I wanted to add, since we are coming from the same background, because I didn't have any bioinformatics at all as an undergraduate. So I didn't even have the inspiration, the where to go to about the bioinformatics. But as you said, if you either if you're exposed to or not, I think that motivation that drives is what really can get you to accomplish the goal. So you said that from the beginning you were interested in plant science, which for me, I was really not interested in plant science. But now we come to the same place. We're interested in the same thing, field. Exactly in the same field. And this brings something that I learned a little bit with Raja, which is we are from completely different backgrounds. He's from astronomy. I'm a biochemist. These are very diverse, complete in the sense that he is looking to the cosmos and I am looking to the microscope. These are very opposite fields, but at the same time, we come to this area where the knowledge of programming and of coding completely intersect.
Okay, so Gio, did you have slides that you wanted to share?
Yes, if I have the time to.
Okay, so let's do this briefly. I have a few more questions. Yeah, go ahead, Raja.
Sorry, I was just going to say one very quick thing to address your point earlier. I think the way we train our students, they're trained to self-betterment is what they're. If you do well, if you study, you'll do better at this exam. Self-betterment is how we teach in schools and colleges, whereas research is about betterment of the world. It's not about improving yourself. And I think that's a very important switch.
I have to say, you have very interesting insights. Both I liked your cricket analogy, and this was also very interesting. Thank you.
Thanks.
So I should
okay,
just go ahead
Gio.
and
Yes, it's all yours. Yes.
All right, so I guess I gave the title of my talk, Computational Programs to Understand and Heal Diseases, and I guess I already gave you an introduction for that. Why do I hold this worldview? So one thing that happened in Brazil very recently, and this motivates a lot of my personal engagement in this whole story, I think, is the fact that the federal government just announced the intention to sequence, I think their number is 1,000 Brazilians. And the reason for that is because the government now wants to understand the genetic backgrounds of the Brazilians and how that can inform the public health system. So for a Brazilian, I grew up with this notion that Brazil is an unequal country, that there's a lot of need in terms of resources. So this particular event here for me is really emblematic, because we are now at a situation where we are thinking about sequencing the people, but honestly, we need to develop ways and tools and human resources to allow us to access that information, understand that information, and make a good use of that information. So I think here we are talking about fields like big data. My field is the field of DNA sequencing, and we are basically talking about how can we make this information benefit an entire society, or as much people in a society as possible. So we have this notion that there is a lack in representativity in many sectors of societies, in many groups of societies, but unfortunately, an inequality is present everywhere in life, and it's a fact of life. Even in the USA, which is the richest of the countries, there is inequality. And I also learned this when working at the science internship program, because what we do in the science internship program is we try to help as much students with problems in their resources as possible. And also in the United States, several institutions deal with the same problem, and they, in many cases, they do the same type of approaches to the problem. But I don't want to talk about a lack of representativity that may be a little bit subjective. I want to bring the attention to this field, the field of genetics or genomics, because I was reading this very interesting paper that stated that around 10% of the human genes, or 10% of, you can argue as 10% of the human genome, to be well studied, and for instance, has allowed us to develop drugs to treat, let's say, only 10% of the conditions that come out of genetic problems. And so this statement that I am highlighting here in yellow is a state that this group published. And basically they concluded that in 2015, 50% of all the publications came from only 16% of the known genes at 1991. And the information that needs to be addressed here is the fact that we were only able to study these genes because there was research going on. And even though there was research going on, there is this inequality in the amount of research that is done for some groups of genes, and other genes are not researched at all. That happens possibly, I want to mention, because we just didn't have the time or the investment in researching all of these other genes. But again, we come to the same point, which is the lack of representativity of all of the genes, or all of the genomic regions that are present in the human genome. And because of that, just because of that, some people are going to lack on the possibility to have their health problems cured or treated. However, there are legitimate reasons for this lack of representativity in many of these studies and in many of these areas. So here, first I want to acknowledge this problem that we have in the genomics field, and then I want to address that from the perspective of the two diseases that I have studied during my PhD and now during my postdoctoral study. One thing that I noticed we for sure need is innovation. We need to think differently. We need to think out of the box about all of these problems. And in my experience, building the community is something that is not necessarily innovative, but I think I learned at SIP that that's the innovation that we kind of need. So now, moving a little bit to the diseases that I studied. So Huntington's disease is actually what we call a monogenic disease. It's a disease that is caused by a condition in a single gene. This gene, I haven't put it here, but the gene is called Huntingtin. And the gene, it's a neurodegeneration that is caused by a CAG expansion. This CAG, for those of you who may not be familiar, is a sequence of bases that are expanded in this condition. And that expansion in the DNA causes the protein that is encoded in this gene to have some different chemical properties. And that's the source of the neurodegeneration in Huntington's disease. Because the genetic of Huntington's disease is so simple, I would say simple in the sense that it is one gene only, and that this one single gene has this dramatic impact in the health of a person. It was well studied in the 20th century. And then what we had was the Huntington's disease was actually one of the first diseases for which we had these genetic consortia that were established in the past century. On the other hand, a disease like neuroblastoma is a polygenic disease, which means many genes, many factors contribute to the establishment of a pathological condition. There is one example of this MYC-N amplification that occurs in neuroblastoma, but I'm not going to talk a lot about that. So here I'm showing kind of where the disease happens in the body. So with this comparison of Huntington's disease and neuroblastoma, it is what I want to say is that it is legitimate for this lack, let's say, of genetic consortia to study certain types of cancer, not compared to Huntington's disease, because remember that I said that the genetic consortia for Huntington's disease were established much before cancers, because studying the genetics of cancer is more complex in the sense that there are multiple genes affecting the condition. In both cases, both in Huntington's disease and neuroblastoma and cancers, this genetic information needs to be stored. And when I say needs to be stored, I am talking about the sequenced genetic information. The sequenced genetic information, the genetic information that you know is written in the human DNA, it needs to be stored. And I think that there is a gap in the availability of programs that allow people to have that experience where they are going to learn how to analyze this genetic information. And I think that's how SIP and the programs that in Pine Biotech also, OmicsLogic, fill this gap is by providing the tools, the environment, the community that is required for people to have that exposure, to have that experience. As I mentioned, I didn't have any bioinformatics background as an undergraduate student. And in my mind, that has not changed. There is some minimal exposure of undergraduate students to the field of bioinformatics. In many cases, that's not a mandatory exposure, but I feel that I think I cannot emphasize enough how much I believe that that exposure is completely necessary, because DNA is information which can be analyzed through computational algorithms and so on and so forth. So that's the motivation for the SIP and the Recombio outreaches that I have been involved. This picture here is showing an outreach talk that I did at the Instituto Federal Baiano, from which I am an alumni. So we have these. It's also a set of tutorial, but really here what I do and what I try to do is to help the students have a sense of what installing a program is, what running that program is, and what does it really the student in the Recombio approach, really the student has a feeling of what it is to interact with the machine and install programs and then starting doing their analysis from scratch. I think that's something that I value a lot, doing the analysis from scratch at this point. So as far as the mentoring perspectives in SIP and Pine Biotech, that's completely valuable, but I really value also the possibility of people engaging and understanding how the research that we do is important, and here are some illustrations of that at the SIP program. The other thing that I think is very useful, and I guess I think I now want to address a point that Elia brought as far as why do we want to go that route. So I didn't mention to you, but my mother was misdiagnosed with an infectious disease when she actually had a cancer. So clearly at that point, there was a problem either with my family or with the medical system or with the government that financed the medical system, because just by not having the appropriate diagnosis, there is some problem going on with either the educational system or the medical system. That's one of the reasons why, in my mind, we cannot really differentiate education from medicine, because we need to have education is the process that allows the development of the tools that are going to enable us to treat people. So what I'm trying to say is that in a situation like this, where you have a diagnosis problem running in your family, you need to I was faced with the problem that not necessarily we have the best diagnostic systems in the world, and if we have it as good as it possibly could be, we also needed to make that diagnosis available to as many people as possible. So this is why I think I need to mention this idea, this concept of liquid biopsies. Because in neuroblastoma, what happens is that the tumor needs to be accessed in the patient, but in the other hand, you need to have certain growth of the tumor in order for you to have access to what's going on with the tumor and the cells. That's why with a biopsy from the blood of that patient, we get a chance to look at many other interesting biomarkers, I would say. So here I am bringing the examples of cell-free DNA, cell-free RNA, nucleosomes, exosomes, cancer-free cells. So we can basically collect these markers, these biomarkers from the blood of a patient, and we can basically interrogate, we can basically study, we can basically understand and educate ourselves about the progression or the biology of these diseases. And my understanding is that you are not going to understand that if we do not understand how to do the analysis, if we do not understand how to interpret the genomic data, how to collect the genomic data, how to analyze it properly, how to develop the strategies that are necessary to treat the patients, to diagnose the patients properly. So this is why I think that understanding the bioinformatics tools is at this point in time, I think it's crucial. I think that in the future, at least, obviously, the societies that are more financially privileged, we're going to have these biobanks that store information on the blood of people, on tumors of people, and this is going to be very useful information to have and to have access to. And there's going to be many opportunities in my mind to study that and to help treat these conditions in people.
So, Gio, I'm going to just jump in here. We have a few questions for you, sure. But for the sake of time, I think that this is a very critical topic for us to continue and explore, and I'm sure that we will have the opportunity to do this soon. But as a way to kind of bring this together, Maria, similarly to yourself, right, is a student that got exposed to bioinformatics almost by chance and has a very fascinating story of how that eventually turned into something very practical and meaningful to her. So before everyone leaves, I wanted to give her the chance to share her story briefly. And then if we have a few minutes at the end, we can address some of the questions that we already have from the audience and kind of bring your story to connect to what she has to say. Is that okay?
Sure, absolutely.
Okay, thank you. Okay, go ahead, Maria.
All right. So I am, as I said before, I am Maria. I am a biotechnology student and I work as content writing intern at Biotech. And I will tell you part of my relation to bioinformatics here and a little bit about my history and also give an example of how we should explore this area being undergraduate student or college and high school, for example. So bioinformatics, it was part of my underdegree curriculum, but Professor Papas was like one of my biggest inspirations because he teach us how to learn about NCBI, protein data bank, this kind of thing that are right now very basic, but this is my way, the way that I start to learning bioinformatics. And but my biggest goal in university was study plant sciences, as I was saying before, specifically phytochemistry and work with the biodiversity in Brazil. But I wasn't aware that my degree was so much than this. I work in communication, in administration, and in bioinformatics. So then appears this great opportunity to create and to lead an IGEN team. So our goal was to develop a microbial fuel cell to generate electricity from leachate. And this was beautiful passion of mine because leachate is a local problem of Brazilian where I live, and the whole problem was this leachate leaks, and we wanted to treat these persistent pollutants there and also take into consideration the other that are present in the city because of the Brazilian sanitary landfill. So this way, we listen in also the Brazilian sanitary landfill and the population, and bioinformatics was used a lot because the whole project was in silico. And I wasn't so familiar with the tools that we need in the same way as my teammates because they had different backgrounds far away from bioinformatics. For example, my friends are from pharmacy, agronomy, administration. So to achieve our goal, we construct a genetic circuit in silico. We start learning independently, and that's the message that I wanted to send to you, because you don't need a degree in bioinformatics or a post-graduation and master's degree to enjoy the benefits of bioinformatics. But you have to be proactive to go ahead and learn something by yourself and discover your passion. And then after you discover your passion in science, you can introduce bioinformatics. So we applied computation in synthetic biology, in environment, in energy, because our goal is to generate energy using microorganisms to poor communities in Brazil, because we were born and raised in low-income families. And we also took into consideration like the social part. So we produce materials for awareness about the environment. And this was also a point that made us to be winner of gold medal in this competition. Arjun competition is like the biggest competition of synthetic biology in the world, but we participated in this version from Latin Americas. And this was a project that was very important to us because we wanted to bring for our people things that are worldwide, you know? So we became the first Brazilian team and the only Brazilian team that was finalist in this competition. And we also won this best project in energy, environment, and biodiversity track. And we were nominated for best human practice and local hero. So we saw how important was bioinformatics for our community. People that don't even know what synthetic biology is, what a molecule is. So we bring all of these things together to make our place a better place to live. And because of that, we achieved many things. We had many opportunities to present our work, including at the campus party, which is the biggest geek fair in the world. And the last few days, there was a version in Brasilia. So we also won some incentive grants and scholarships, and we are now changed like forever because we continue to learn each day a little bit and about this field, such as the OmicsLogic courses that I began to watch and to study. So I recommend for all of you to start in some place, even if you don't know where you're going to apply this knowledge. But when you find the right place and the right opportunity, you will use bioinformatics. I am 100% sure. So this is my contact. You can talk to me anything and ask me some question, and that's my presentation.
Excellent. Well, thank you very much. We're already way over time. We have scheduled this to end at 4. So, you know, taking additional time from all of the people that participated. But I do have an important question, and I want to start with Raja. You know, you bring years of experience to this. Why is it that the academic world is so appearing to be maybe closed off to people that are just coming in? Why does it seem to be so like such a big gap between people that are doing actual serious important research and students that are just coming up and getting involved?
It's a great question. I don't know. I think I don't think there's a proper recognition of the amount of brain power. I think it's two things. I think it's tempting for us to think that you have to have gone through all of these years of training. It's tempting to think it's a linear process. It's not. People can, people talk about the STEM pipeline. It's not a pipeline. You can plug in at any point, right? You can, the work I do, most of my day can be done by a trained monkey. That's the sad truth, right? It's very possible for young people to do this work without having gone through this long winded training. That's a sad truth. People don't like to acknowledge that. I don't know why. They don't like to acknowledge it. And I think they're not aware of the sheer amount of brain power in the, let's say in the, 12 to 20-year-old or 12 to 18-year-old, population in the world. It's taking a person from that self-betterment mode to world betterment mode is hugely motivating. You just have to reorient that person's thinking and they become a powerful force for good when you do that. I've seen this happen myself, but I think more and more people are turning to it, Elia. I don't think it's a lost cause.
Excellent, thank you. Well, my second question is for Gio. Gio, you mentioned that Brazil just has this goal of sequencing 1,000 genomes. Just today, or maybe yesterday or the other day, there was an announcement that the All of Us project has released 98,600 genomes that they sequenced in the past, I think, couple of years. So, the goal of the All of Us sequencing project is to introduce more diversity. And I think 42% of the people were white, as expected. So what do you think the world is missing out on when the numbers are so disproportionate, between countries that, have such efforts and countries that maybe do not have as large numbers as you showed in Brazil?
I think that, for the Brazilian part, I think that the actual interaction between the diverse chromosomes that compose humanity, that there is a peculiar case for Brazil because there is high degrees of diversity in Brazil because of the colonization. We had this admixture of these different populations, mainly notably from Europe, Africa, and American Native Americans. Of course, there is a matter of income. Only those countries, I would say, that have the resources to afford this can do it. And for good or for bad, that's going to be these countries with huge populations like Brazil, the USA, China, and India also. But certainly the other populations that are underrepresented in the world, with smaller populations, like small countries in Africa, small countries in Asia. There's representation of those populations in other countries. In many cases, that I know that to some degree, these genetic consortia, they do what they can to increase the representation of these populations. And unfortunately, in many cases, you're not going to be able to see the interaction of these chromosomes as you are going to see in Brazil. But in a way or another, this information can be available, or this can be analyzed. I think one thing that is necessary is the interest to analyze. And by interest, not necessarily I am meaning the interest in only investing in this type of studies. I am meaning the interest for people to go out there and download the files and put their hands on downloading these files, which per se is a strong effort to do.
Excellent, thank you. My last question is for Maria. So Maria, I know that you showed us you do like a million different things, right? You write for different companies, journals, you have like 10 different jobs. What is the one thing, if you didn't have to, what would be the one thing that you would apply yourself to if, you could choose from anything? And what do you think would be the benefit of just that single focus if you had the freedom to do that?
Well, my purpose in life actually is work with bioeconomy. So I want to help other companies with my knowledge to make all of different parts of the world more sustainable. And I think that all of the tools that I am working on and all these jobs, like in pre-acceleration of startups or bioinformatics or communication, are going to be some like of soft skills for me to make the world like a better place to live, you know.
All right, great. Well, it was a pleasure talking to all of you. Thank you again for joining and sharing. I think there's so much that we can get from all of your conversations. And I think if we had just a little bit more time, we would learn a lot more. I want to briefly mention that tomorrow we have our second day that's actually dedicated to industry and practical solutions, right? And so what I mean by industry is we're going to be hearing from people that work in biotechnology industry, in pharmaceutical industry, and they're going to share their insights about the role of bioinformatics and the significance of bioinformatics in that field. We're also going to hear about novel ways to use and simplify bioinformatics so that it's much more accessible and much more easy to interpret the data. And so if you're planning to join, we're going to have people like Anka Maria Ciobanu from the Pistoia Alliance from Massachusetts. She's going to be speaking about Pistoia Alliance's new Women in STEM Leadership pilot program, as well as their program on the efficiency and effectiveness of R&D for big pharma. We're going to have representatives from the Tower Bioinformatics Research Center speaking about innovation with cloud computing and different types of analyses. We're going to have several speakers from NASA, from the Blue Marble Space Institute of Science to talk about astrobiology and what's happening there. And we will have prizes for those who presented today in the poster session. So we're going to have prizes, so be sure to join that if you presented. We're going to have some more people speaking from the National Association of Idea Principal Investigators, Google, from the Precision Medicine Lab here at Louisiana State University, and also speakers from the Puerto Rico INBRI program and from the INBRI in West Virginia. So again, thank you everyone for joining today. Thank you, Raja, Gio, and Maria for your insights into this problem, and we will see you hopefully tomorrow.
Thank you, Elia.
Thank you so much.
I learned a lot from you, Maria and Gio. Thank you.
I too learned from you. Thank you.
Take care.